I'm the director of the Duke Margolis Center for Health Policy, and on behalf of the center, I'd like to welcome you all to today's public workshop on utilizing innovative statistical methods and trial designs in rare disease settings, which is being convened under our cooperative agreement with the FDA. We are pleased to be joined for these discussions by a wide range of leading experts from across government, academia, industry, care delivery, and patient advocacy for what I think is a very timely discussion on some important and potentially addressable issues related to rare diseases. Our purpose today is to further a broad discussion on rare disease drug development by considering a set of important topics on trial design, analytic approaches for clinical trials, and other steps that can assist in learning about what works in these small patient populations. The meeting will be a chance for leading experts to discuss their experiences with these techniques and to help identify a set of actionable next steps for advancing drug development in the rare disease setting. Throughout the day, we encourage meeting participants, all of you, to consider opportunities for increased collaboration, to take some of these ideas from different stakeholder perspectives and try to find ways to work across agency, across industry, across different groups, and with the agency to support these efforts in drug development. The goal is to predictably and consistently utilize these novel approaches across development programs to help get better treatments to the 30 million Americans that suffer from a rare disease. That's a big number for diseases that are defined to include less than 200,000 people each in the United States. But collectively, this is 10% of the U.S. population, most of which have no treatments available at all, and many of which certainly have room for improvement in the therapies that might be available. Now, in a few moments, we're going to hear from the FDA about recent experiences in rare disease drug development and where they see some of the potential opportunities for additional work and progress. We're then going to turn to the major three topics of the day. The first is a session on the use of early phase data in phase 3 trials, of building on what's learned through a variety of methods and data sources for those phase 3 pivotal trials. The second session will focus on the use of patient registries and natural histories in rare disease settings. A third session then will explore how master protocols can be leveraged in this space. So these are a range of techniques which all have in common trying to make the most out of the limited data available often for these populations, and to try to push forward on ways to develop better data, better evidence to support more efficient drug development. After these three sessions, which will have some discussion along the way, again, from a lot of these different perspectives represented today, we're going to have an opportunity for comment from the public. So we want to hear from all of you about your ideas to build on the topics discussed today. And then we're going to close with a very interesting session, a synthesis discussion that will bring a range of perspectives together to look forward. Based on what we've heard today, what are the priority action items for everyone in this room, and as well as those who are watching online, to take away as next steps. So before we get started, I want to go over a few housekeeping notes. First, we are being joined online, and I want to thank again everyone who's joining us through our webcast for participating as well. As we go through each session, as you'll see on the agenda, we're going to begin with presentations, and then those will be followed by a panel discussion. We've also got some time set aside in each of these sessions for comments and questions from the audience. And for those of you who are in attendance, we have roving mics for you to use during the day. So we'll get time, a little bit of time during each session for those comments and questions. I want to remind everyone here and online that this is a public meeting. The event is being broadcast, so everything is on the record. And for those of you who are joining from our webcast audience, we encourage you to participate as well by sending questions to margolis.rare@duke.edu. margolis.rare@duke.edu, and our staff will get them up to us to include them in our session discussions. For those of you who are in the room today, we have coffee, beverages located just outside. Please feel free to help yourself. Lunch is on your own, so we'll take a break for that. We have, I think, a restaurant list as well. Plenty of restaurants very close by in the area. And then finally, just a reminder that although this meeting is being convened under a cooperative agreement with the FDA, this is not a federal advisory committee. There are not going to be votes. There's not going to be a consensus determination or anything like that. The meeting will be a success if there is an exchange of lots of ideas and open discussion about how to move forward on these important issues for developing better evidence and supporting drug development for orphan diseases. Okay, so that's a little bit of framing. What I'd like to do now is turn to Laura Lee Johnson, who is helping to lead the efforts on this topic at the FDA. Laura Lee is the acting director of the Division of Biometrics 3 in the Center for Drug Evaluation and Research at the FDA, and she has some opening comments for us about FDA's approach on these issues. Laura Lee, thank you very much.
Thank you, Mark, and welcome everybody. Thank you to everyone who's joining us today, whether you are online here in Silver Spring or, unfortunately for some, probably waiting on 270. So I want to start today to remind everyone that in clinical research, patients are really our most precious resource. So as was just mentioned, the Institute of Medicine estimated that 30 million Americans, or roughly 10% of the US population, suffer from a rare disease. But when we think about those small samples, it might be that they're 30 people, or 500 people, or 30,000 people, and the global prevalence may vary. So when we're thinking about study designs and the types of analyses we can do, it's not simple enough to just say we've solved this for rare disease. We have to think about all of these other factors. Those small population sizes pose significant challenges to the traditional research and development process. So what I'm hoping that we focus on today is thinking about reducing the burden for patients, their caregivers, clinicians, and others, then reusing and recycling. You know, we talk about this as a phrase other places, but realistically, that's where we've been moving, is that reusing data that's collected for other reasons. But that also means perhaps changing the data and what's used and collected and thinking about this with really more of a holistic approach. So a lot of times we have a very specific mission. I worked for more than a decade at the NIH. For my individual study, I collected the data for my study. When I work with clinicians, they collect the data that they need and they do it in the way that they are going to use it. But if we're thinking about reusing and recycling that data, we have to think a bit broader. And so I grew up the daughter of an engineer who always taught me to try to over-engineer. Think ahead. You know, don't build the bridge for the traffic you have, build the bridge for the traffic you expect in 20, 50, 70 years. We probably can't foresee where we need our data in 70 years, but we can think about the broad uses of that data. Because again, in this space, every single piece of data from every patient becomes very precious. So this is a fancy way of saying plan ahead. When you're starting a study, data collection, registry, et cetera, think ahead to what all that data could be used for. And you'll hear some examples of that from our speakers today. Think about collaboration. We all are not going to just collaborate today, but hopefully continually collaborate. And we're seeing this more and more with each other, with regulators. And while FDA only has so many resources, we would much rather talk to you early as you're designing perhaps a prospective natural history study, even if there's not a therapeutic yet. Because eventually, when that therapeutic is there, that study is going to be better positioned if there's a need in order to help support many of the innovative trial designs that we're talking about today. So it might be that that data is for an external control. It might be used to help design the study. There are a lot of different ways, and if you think about and listen to how they talk about standing up some of these platform trials, you may also experience that. Think also about the international collaboration that's going to be necessary. We're not talking about that today, but don't forget asking for parallel scientific advice, perhaps from the EMA and FDA, as you're doing some of your early planning. So hopefully today's discussions are going to inspire us all to be creative, to think about the regulatory intent in the future for all the work that we're doing. What data are we going to need? What are the clinical endpoints and the variables that are going to be necessary by all the different partners down the road? To give an example, we're now starting to hear about cooperative groups out of the NIH and other groups that are considering using, and some of them already using, CDISC data standards, for example, even for observational trials, because what they're thinking about is that end in the mind in future where that data may be sent to the FDA. But going back to the statistical methods and study designs, some people think conventional methods will never work in the rare disease realm. But many times, again, it's time to be creative. If you've never questioned what will work, by the end of today, you will have some interesting answers. We've so far had 2 statistics think tanks that led to today's public workshop. Now, I want to thank Lissa Lavange, who's here in the audience, and 2 others who were previously at FDA who really started this off. They were champions across disciplines, and although they're no longer at the FDA, they grew a much larger reserve while they were there. And this goes all the way to the top and across many different disciplines, some of which you'll see here today. There's strong interest in facilitating and accelerating rare disease drug development. There are a lot of different approaches for rigorously assessing efficacy in the rare disease setting. So what are the good strategies for using all the information there is in a single study? Thinking about what measures make sense. For example, maybe traditionally you measure sight using an eye chart, but if your study subjects are blind, or they're going to probably be blind during your trial, that's not the outcome that may be of interest. What is it that you can use to get at the concept? 6-minute walk test, for example. If you have people who are not going to be ambulatory, how are you going to measure what changes are important? So today we're not going to discuss all of these methods, but it's something to also think about as you're thinking about your trial designs. But there are a few other ways to think about efficiencies. For example, again, rethinking and standardizing measurement and collection methods for these clinical endpoints and biomarkers that are used both clinically and in our trials. How do we prevent the missing data? How do we become more efficient, for example, using group sequential designs? Think about the creative randomization strategies and also the community engagement and education that can happen. Many times we say, when we hear, patients won't accept this. But if you go talk to the patients, they are willing and they're very excited and sometimes your best advocates for excellent trial design. So we want this meeting to be about solutions and innovation. Keep everything positive because it's very easy to say, well, there's a problem. There are plenty of issues in small sample designs, but we need solutions. So recently I was at a meeting and the pediatrician, he was like, my patients deserve evidence, not just anecdote and conjecture. So what this meeting is about today is about giving them that evidence in a rigorous manner. There's a lot of leadership in trying to bridge some of these old divides that have happened, and there's a lot of great potential. So we're going to stay focused. Mark and Greg will make sure of that. And we're going to think about how we're going to rigorously create evidence for those patients, their care partners, so they're not only relying on anecdote when they're trying to determine care. There's a lot more dialogue to come, so I want to echo Mark and say success today is the exchange of ideas and open discussion. If you have a concern, please voice it, but also try to provide a reasonable solution or path forward. So if you have a concern, please, that's part of what we need to work through, but also give that path forward, the potential solutions. Thank you to all the patients who have come today and their care partners to provide patient perspectives. We also have perspectives from bioethics, various clinical trialists and other clinicians, a variety of other hats are here today beyond statistics, because the most beautiful study design and analysis isn't all that worthwhile if people won't enroll in the trial, or nobody will put the trial in the first place, or it doesn't answer the reasonable clinical question. So this is a true cross-disciplinary work. Thank you to everybody, and welcome to Silver Spring.
All right, Laura Lee, thank you for getting us started. So this is a moment in the session. We were very pleased to see so many of you here, as Laura Lee said, from some diverse perspectives. We do have some seats, and while I'm talking, you're not listening to somebody that's really got some good substance. This is a good time. If those of you who are already seated don't mind, may need to let a few people slide in. Again, it's really good to see so much and so diverse interest in the topics that we're presenting today, going way beyond statistical experts only involved. Okay, we are going to start our first session now on using prior data from early phase trials to inform phase 3 designs. And we have 2 initial presenters in this session, and I'm going to introduce both of them now, and then we'll hear from them. Then I'm going to introduce our reactants for this panel, our panelists who will provide some perspectives on what they've heard from these initial presentations. And then, again, some discussion. We want to hear from all of you as well. First up is Karen Price, who's Senior Research Advisor with Eli Lilly and Company. She's going to discuss Bayesian borrowing of historical data for confirmatory clinical trials. Then we're going to hear from John Scott, Deputy Director, the Division of Biostatistics in the Center for Biologics Evaluation Research at FDA, who will present on incorporating historical controls in phase 3 designs. Karen, thank you for being with us today.
Okay, thank you so much. What an honor to be here today, and a great start to what I know is going to be a wonderful day. I'm going to be talking about Bayesian borrowing for confirmatory clinical trials, and I'd like to start by acknowledging some of the individuals listed here who were instrumental in getting this deck of slides together for us today. So my outline, I'm going to start with an overview of the Bayesian approach, really more motivational than anything else. I'll be going through a few methods for formally borrowing information and also talking about data sources that could be useful in constructing priors. Have a couple of examples and then some conclusions. Okay, so the key messages for today, we're all here because we recognize that rare diseases are in desperate need of innovation. And in fact, I love the introductory comments that we've had that we really need to be able to squeeze every ounce of information from each and every single piece of data that we have in this setting. And the Bayesian approach provides a formal framework for borrowing that historical data. It offers an intelligent and complete use of the data. It's important to note that using a Bayesian approach in formally borrowing information is not a weakening of the standard of evidence. But if we do ignore that information, then it can be detrimental to our ability to make the best decisions that we can. And there are best practices to ensure that there's a transparent understanding of the impact of borrowing. And I think as we think about solutions, as we've talked about today as a solutions-oriented meeting, really understanding those best practices and aligning on those would be important for us moving forward as we allow the formal incorporation of data into trial designs. And then thirdly, Bayesian methods can improve the design and analysis of studies for rare diseases. So because this session is focused on borrowing, that's what I'm going to be focusing on. The other sessions will talk about other innovative trial designs. Certainly aspects talked about here could be utilized in those settings as well. Okay, so just briefly motivating the Bayesian framework. So one of the really nice things about a Bayesian approach is that it emulates the way that we naturally think. We all learn from previous experience. Personally, when we encounter everything from day to day, perhaps we know certain roads to take if it snows, or different types of restaurants that we might like to go to from our personal experience. But as we move into scientific and business decisions, much of the information that we have is in the form of data. And every time that we see results from a clinical trial, we naturally think about other data we've seen, maybe from a compound in a similar mechanism of action, could be earlier results, could be preclinical results. Whatever it is, we bring that to our decision-making process, but we do that in our heads, and we determine on our own how much we weight that piece of information. What we really need is a mechanism in place to formally synthesize that information, and Bayesian methods provide that platform. Pictorially, we can think about this as what we knew, which is our prior, what we see now, that's the likelihood, and then we update that with what we now know, the posterior. And this is, of course, cyclical as we learn over time. Okay, the value of a Bayesian approach. As I mentioned, it emulates how we naturally think, and it facilitates a process of continual learning so that we're always updating our knowledge. It is very useful in the sense that it enables us to answer questions of interest, so we can talk about probabilities of the questions of interest, such as what's the probability that this treatment is better than another treatment, or that this treatment's effect exceeds a clinical threshold of interest. We'll see some of those examples later. As mentioned, we can formally incorporate prior information. Okay, sorry about that. There we go. There's great flexibility in modeling and prediction. And if nothing else, I wanted to for sure mention that it is also completely transparent. So a lot of times I think one of the biggest criticisms that we hear when people utilize a Bayesian approach is the fact that there's a prior. But it's important to remember that no matter what, you cannot hide from the prior. And so we'll talk about the transparency of that and understanding the impact of that prior to inferences, the posterior inferences, and it's there for everyone to see. Okay, so I want to talk then about approaches for borrowing. Typically, when we think about approaches for formally borrowing information, they're bucketed into a couple of different methods, one being static borrowing, the other being dynamic. There are a few listed that are typically considered more static borrowing, pooling, single-arm trials, power priors when the discounting parameter is fixed. Dynamic priors include things like hierarchical modeling and mixture priors, and the examples will focus on power priors and mixture priors just to show a couple of different approaches. And because we know that we can borrow on the control arm and or the treatment arm, the approach for borrowing can differ as well, and we'll show that in a couple of the examples. Oh, all right. So briefly, an overview of potential data sources. I will touch on the potential role of expert opinion in this setting. Most of those are formal data sources, natural history studies, summary level information that may be available in the public domain, could be individual patient data, PK/PD modeling, and preclinical data. And importantly, just recognizing that when it comes time to borrow, we need to be thinking about the relevance of this historical information to the new data in terms of the indication, the patient population, time since collection, and things of that sort. I have one slide here on the potential role of expert opinion, and then we'll move into formally using data. This is something that there is a large literature on the topic and certainly would not be considered for establishing substantial evidence, but I think expert opinion plays an important role in as we think about how do we link sources of data, how do we link endpoints together. When expert opinion, if it is utilized, it's important to note that there are formal, well-tested protocols that should be used and established upfront before the elicitation happens. We don't need to necessarily use the prior as is. It can be downweighted, utilizing some of the methods we'll talk about. But as I mentioned, it is something that we've done some to help us understand relationships between endpoints or doses or populations and kind of hone in on and improve on the designs. And it could also be used to help inform on the relevance of that historical information. So, for example, borrowing of adult data from pediatrics, there have been some examples of using expert opinion to help us determine how much should we weight that adult data in that instance. And then before getting into the examples, just some things on how much to borrow, which is always the key questions, thinking about what data is available to be included. Right now, we typically would simulate operating characteristics, and we'll look at a couple of examples. Thinking about things like prior effective sample size and the prior probability of success, and assessing prior to posterior sensitivity are all good practices. We may think about what is the medical need, the unmet need, in terms of how much to borrow. Then a final note on borrowing before we get to the examples is that I think there is also a perception that borrowing is intended to be a way to always favor the sponsor, but in fact that's not the case, that it is really, it could dampen the effect, but again is getting at how do we best estimate this effect, and is a focus on estimation, utilizing all of the information. Okay, a couple of examples and then I'll conclude. So this first example is on borrowing historical controls, primarily to showcase some of the methods and the ways we might think about this. And then the second one will be borrowing on both the control and the treatment. In this example, we have previous data on the control group. We had a trial where the historical rate was 60%. And let's suppose that we're designing a study for 70 on control and 140 on the new treatment. I won't go into detail on this plot, but what we wanted to do is showcase a couple of different approaches. This side is the power priors on the left-hand side, and on the right-hand side you have mixture priors. So what you can see is for a power prior, you have this discounting parameter that ranges A0 from 0 up to 1, where 0 in this case is no borrowing, 1 is full borrowing. And so you can see the increasing, that prior is increasing in its steepness, and would be borrowing more in that case. A mixture prior, on the other hand, is going to be, in this case, what we've done is we've mixed with that same beta distribution with a non-informative prior, and so you get these sort of heavier tails out here. And as we'll see, because this is dynamic, when the data comes in, it's going to borrow more from the point that it's most closely associated with. The next plot that we have is just to show how we might look at operating characteristics. There, oh. Sorry about this. There we go. All right. Okay, so what's going on here, again, is it's very busy, so just to kind of talk through one example, what we've got is, this is the power priors, these are the mixture priors, again, increasing the amount that we're borrowing. If we just take, for example, if our discounting parameter was 0.5, we've looked at some known scenarios of truth to look at, as that control rate increases, what happens to the probability of meeting whatever we've declared as success. And so you can see in the power prior approach here, these probabilities continue to go up as that control rate goes up from the new trial. On this side, though, one nice thing about a mixture prior is this begins to plateau as the control information deviates further from the historical. So these would be the types of things that we might want to look at ahead of time to understand how that borrowing is affecting what might come in for our current trial. And then we may also want to look at a few example results. So what we're showing here on the left-hand side, this is for a power prior, would be we've got the prior is in blue and the likelihood scaled is in red, and the resulting posterior is this purple distribution. And you can see with the power prior approach a couple of different outcomes for the study of interest, and how with the power prior, it's essentially a weighted average between these. But when we compare that against a mixture prior, the dynamic nature of the borrowing allows it to follow. This is the posterior here now, putting more weight closer to the data since it's deviated away from what we had historically. Okay, one more example then to go through in some conclusions. This one is dynamic borrowing of adult data to pediatrics. In this case, suppose that we are considering a pediatric rare disease trial. In 50 patients, we have 40 active and 10 placebo. The primary endpoint is a binary response variable, and as we've talked about already, we want to utilize all of the information that we have available to us, so we've conducted a network meta-analysis of studies that are available, and we did have this drug of interest in one of the adult studies. One of the things that we're going to do is declare success, in this case, using a probabilistic statement, the probability of effect, whatever effect, in this case, it's the difference in log odds, exceeding some clinical threshold, in this case, 0.4, and we're going to declare success if that's greater than 80%. One thing that is important to note here is this probability can be based on the medical impact of disease or patient prescriber input. And so is a nice way to be able to think through how certain we want to be on that, on our probability statement. And then the next two slides just go through then these network meta-analyses showing the placebo effect by studies. Here, the average control rate was at 0.4. We also had some historical on the relative drug effect, and the drug of interest rate is at 0.5. And so then we wanted to think about how we might borrow. This is a plot of prior effective sample size. And so what you can see, what we're going to just do in this one is just show the mixture prior. So as a mixture ranges from 0 up to 1, where 0 is full borrowing in this case and 1 is no borrowing, you can see the prior effective sample size for the drug of interest with full borrowing is around 42 or 43. For the placebo, it's about 56. And then you can, at any point along that mixture distribution, identify how much you're effectively borrowing in your new analysis. And then we wanted to look at some outcomes similar to how we did last time. And there's a few points to be made here. One is that this example, what we've done is we have 4 out of 10 placebo, which is in line with the historical rate. And we wanted to see what happens as the drug of interest deviates and has different outcomes to ensure that we aren't misled. As you can see here, the prior probability of success, as we mentioned, that's our success criteria, is about 68%. But when the data come in particularly less than placebo, that probability drops to about 2%. We can look at an example here where the drug of interest performed very similarly to the adults in the pediatric patients. So this is at a 0.5 rate for the drug of interest. And here our probability went to about 85%, which would have met then our success criteria. If we had not borrowed, we're down around the 60%. So again, able to declare success, get information to patients, and improve our ability to estimate that probability of interest through the borrowing. And then, of course, if we observe better, that probability continues to go up. Okay, so in conclusion, as we've said, we are in desperate need of innovation here and need to leverage all sources of information. Hopefully the presentation today has shown that there is a great flexibility in methods for borrowing and that we can incorporate patient or caregiver preferences and set thresholds accounting for that unmet need. And I think importantly, though, what it's going to do is it requires us to shift our thinking from having a historically maybe 2 studies at p less than 0.5 to more of a continual learning via the Bayesian approach. Thank you.
Great. Thank you very much. And as I said, next we're going to hear from John Scott.
Thank you.
Sensitive.
Okay, I'll be sensitive. I'll be sensitive in every sense of the word. So it's always a pleasure to talk immediately after Karen. I've done this a few times before because her talks are excellent and comprehensive and give me not a lot left to do, which I like. But I'm going to be giving just a few general comments on the topic of borrowing from early phase data from the perspective of someone who helps statistical reviewers decide how to evaluate these proposals at FDA. So I probably don't need to tell anyone here these first two bullet points. When we're talking about doing clinical trials in a rare disease setting, there are sort of two really salient points. The first is that the need is very great. We're often talking about serious and life-threatening conditions, and we're often talking about serious unmet needs, areas where there just are not drugs available to treat very serious conditions. The second bullet point is that the practical trial constraints in rare disease populations can be very high. The most obvious way in which that's true is that you simply don't necessarily have a lot of patients to participate as research subjects in studies. And there are other ways in which there are practical constraints in a rare disease setting that we don't have in larger diseases. I don't know if it's controversial for me to say that I think statistics is a moral activity, but I do. And I think that we have a moral obligation, especially in this setting, in general, but especially in this setting, to use information as wisely as possible. And that can definitely include innovative trial designs and things such as the borrowing techniques that Karen talked about in her talk. But we also, of course, have an obligation not to make erroneous conclusions of effectiveness, not to leap ahead so much that we make reckless decisions to approve products, for instance. So there are a lot of different ways external data can be used in late-phase trials. The first is, it's not just common, it's essentially required to have data to help you plan a late-phase study. We have to do sample size calculations for late-phase studies. Typically, it's considered required to have some kind of empirical basis for effect size assumptions that go into that, and that all requires some form of external data, usually from phase 1, phase 2 studies. External data are also often used as comparators in single-arm studies. I think maybe the second session today will be talking more specifically about that topic. I'm going to stay away from it and stick to the two things Karen talked about. Maybe it's one thing, partial borrowing of control data from early phase studies or other external sources, and partial borrowing of both control and treatment or experimental data from early phase studies. I think those two topics obviously have a lot in common, but they also have really important differences, and sometimes it's helpful to keep them sort of separate in our minds. So Karen touched on much of this. There are some general considerations when you're thinking about whether you can do partial borrowing and how you would go about it in a given situation, general scientific considerations. The first are the things that Karen had a little footnote about exchangeability. I'm calling it scientific comparability or clinical comparability of the data sources. There's a lot that goes into that. Are you studying the same population in the late phase study that you were in the early phase studies? Do you have the same endpoints? Are they measured in the same way? The investigational sites, it may not be exactly the same, but are the regions the same? And if the regions aren't the same, are there substantial differences in background care that the patients are receiving, which can make huge differences in comparability? The second point I wanted to raise is prospective planning. I think it's non-controversial that if you're doing a late phase study and you want to borrow information from an early phase study, you plan on doing that at the beginning of the late phase study. I think most people would agree on that. But I think it's also helpful to think about planning even before you acquire that early phase or other external data. If you have an overall development plan strategy that includes borrowing, it's going to encourage collecting data in a way that fosters more homogeneity, and it also can help avoid some blind alleys where you might run a risk of fooling yourself. A thing that has come up is people have run a phase 2 study and want to borrow information into the phase 3 study, but they only want to borrow information from the highest performing subgroup from the phase 2 study. And that's not impossible, but it does raise questions of misleading yourself with regression to the mean and other issues. And then finally, the last point I wanted to raise is when you're thinking about borrowing data and borrowing information, I think it's a perfect goal that could never be achieved to use all relevant information in that exercise. But we should be thinking broader than just an individual early phase study in many cases. So let's say you're a manufacturer and you ran a positive, very encouraging phase 2 study. You want to borrow that data into phase 3. But let's say there were also 10 other manufacturers who ran phase 3 studies against the same target, and all of those phase 3 studies failed, should that not also, in some sense, go into your calculation? I don't know exactly how you would do it, but I think it's worth considering. Okay, so type 1 error considerations. Karen sort of touched on this and danced around it. I'm going to be a little more direct. If you're doing this kind of partial borrowing, if you are talking about controlling the type 1 error probability, the maximal type 1 error probability of the new trial at the conventional 0.05 rate in the strict sense that we usually mean, you're eliminating the purpose of doing the borrowing. That's fundamentally a self-defeating strategy. So if you were agreeing that borrowing made sense, I think there's a necessary way in that you're agreeing that the type 1 error considerations need to be considered a little differently than in most other settings. And I would go a little further, and this gets into some technical topics, but when you're doing Bayesian analysis with informative priors, what type 1 error is itself is a pretty murky concept. And we sort of take for granted that we know what we're talking about when we talk about it, and I don't think we do a lot of the time. There's a lot of literature on alternatives to type 1 error in Bayesian settings, other kinds of error rates, like the average Bayes error of a decision, or things like the maximum posterior probability of the null hypothesis in the rejection region. And I think broadening out our consideration to other kinds of error rates is a good thing for statisticians to be thinking about. And I want to stress, I'm not talking about relaxing standards. I'm talking about putting our standards on an interpretable scale where we can really weigh the benefits and risks of a decision. Speaking of benefits and risks and decisions, on thresholds and decision making, so Karen talked about ways you would think about how much information you would be borrowing from prior data. And I don't think she implied that that was easy, but I think it's very hard. I think it's a really complicated thing to even think about. So we can talk about prior effective sample size, and prior effective sample size is really intuitive. You're saying how many patients worth of data am I borrowing into the new study? But how do you go about approaching the question of how many patients worth of data should you borrow into the new study? Those two scales aren't necessarily immediately comparable. Similarly, I'll often hear someone say, yes, we'll borrow, but we don't want the prior to account for more than 15% of the posterior distribution. Again, I mean, that's on a quantitatively interpretable scale, but I don't know what that means in real life. And I don't know how you would come up with 15% or any other number. So it's difficult. People also talk about calibrating to type 1 error probability. That's fundamentally a fairly arbitrary exercise where you set your type 1 error probability to be. Dynamic borrowing helps somewhat in the sense that you're letting the data guide how much you borrow, but you do still have to calibrate the decision rule. And that calibration requires tying it to something you're trying to optimize, some goal. And what that goal should be is often quite murky. And so I would like us to try to think about ways that our decisions can be calibrated to outcomes, calibrated to patient values, provider values, and public health needs. And that's a tall order, and it requires some decision theory concepts, but it's fundamentally, I think, the only principled way to really make decisions. Okay, finally, this is mostly just a plug for tomorrow. There's a lot of complicated regulatory interactions around these kind of proposals. Any kind of really novel and complex approach is going to be a learning process, both for applicants and for FDA, and it'll require more planning than usual and a lot more discussion than usual. And I wanted to, probably most people in the room already know, but tomorrow on FDA's campus, we're having a public workshop on complex and innovative trial design, where we'll be talking about a lot of topics related to, especially this session, maybe the rest of the day too, including alternative trial design possibilities, the use of simulations in trial design, and FDA's soon-to-be-launched pilot program for evaluating novel and innovative trial design proposals. So people who are free tomorrow, please come up to White Oak. It'll be a barn burner, I'm sure.
That's right. Okay, John, thank you. John, thanks for that very complimentary proposal, what we heard from Karen. I would like to ask our panelists to come on up, and I will introduce them while they are doing that. First, we're going to hear from Lissa Lavange, was introduced earlier and I think known well to many of you in this room. She is currently a professor and associate chair and director of the Collaborative Studies Coordinating Center in the Department of Biostatistics at the University of North Carolina, but until recently at FDA, very much involved in many of these issues. And Lucas Kempf, who's acting associate director for the Rare Disease Program in the Office of New Drugs at the Center for Drug Evaluation and Research at FDA. Gary Rosner, professor of oncology at the Johns Hopkins University School of Medicine and professor of biostatistics at the Bloomberg School of Public Health at Johns Hopkins. Roy Tamura, who's associate professor of biostatistics in the Health Informatics Institute at the University of South Florida. And Gigi McMillan, graduate program coordinator and patient representative, the Bioethics Institute at Loyola Marymount University. So I'm going to hear from them in that order, and I'll start with Lissa.
Thank you. Now, am I reacting to the Carolina basketball game yesterday or the talks we had this morning?
Take your preference, but you might want to stick with today's topic. Yeah.
So thanks to Mark for moderating this session and Karen and John for giving two great talks. I am not at the FDA anymore. I've almost stopped saying we, but this meeting is important to me. It was a couple of years ago, Rich Mashitsky and Jonathan Goldsmith and I put our heads together to figure out what we could do to kind of break loose the rare disease drug development, encourage more innovation, show sponsors in particular and patients that FDA was serious in trying to facilitate and accelerate development of drugs for rare diseases. The quote from our center director, Janet Woodcock, that I repeat over and over again is that just because not many people have your disease doesn't mean that you don't get drugs, good drugs. So we did have two statistics think tanks. Many of the people you see on the panel and speaking today participated in those. The idea was to just have a no-holds-barred discussion of proposals, anything wild, don't hold back, what could we do in rare diseases? And we had a lot of very good discussions with some esteemed colleagues, statisticians. We were focusing on design, endpoints, analysis, but also our clinical colleagues. We had participation by many people at FDA at these think tanks. And when we were getting together last year to plan the public meeting, we had a common feeling that, okay, we've talked about these innovative things. Let's have a meeting where we put the proposals up there and have a public vetting of them. It's too late to just talk about the challenges. We all know what the challenges are in rare diseases. So the purpose of today isn't just to have one more public meeting about the challenges in rare disease drug development, but to actually say, could we do this? Could we do this? Would FDA take it? Would sponsors be comfortable with this? Is this something that will help the patients? Will we get credible evidence that we can make regulatory decisions from these different approaches? And so that's our goal. So even though I'm not in charge of this meeting, I had to start out with with what I hope that we accomplish today, because I'm very excited about being here. All right, so that being said, the first session is probably the most complicated topic that we're going to talk about today. And I think the thinking was, the thinking was that we would throw the hardest thing out there first, early in the morning when everybody was fresh, get you to think about power priors and posterior distributions and so forth, and scratch your head a little bit, because it is a bit complicated. But the idea is that we're in situations often with rare diseases when you just simply cannot enroll enough people for a fully powered randomized controlled trial, the gold standard for all of our drug development programs. And this is a place where we're looking to substitute something for patients. And that something could be from a number of different areas, and we'll talk more as the day goes on about sources of information that you can substitute. But the topic from this morning's session is, can we substitute early information on the same drug in the same drug development program? And this is a stretch for FDA. We typically want your phase 2. FDA typically wants the phase 2 program to end with evidence of efficacy and good words on safety, and then you sort of wipe the slate clean, assume the drug doesn't work, go into phase 3, and use your typical frequentist thinking to make a decision based on the type 1 error. which assumes the drug doesn't work. Well, this is turning that upside down. You know, we don't have that luxury necessarily in rare diseases. So can we come up with a good, strong, rigorous way to use that information we already have on this drug, or maybe drugs similar to it in the same class, or maybe drugs with the same mechanism of action, maybe some preclinical information, some prior source of information about the drug under study to inform in a rigorous way what's happening in the phase 3 trial. And can that let us get away with fewer patients? That's really the bottom line. Can we have a smaller trial in phase 3, one that has a better chance of getting an answer to the question, does the drug work, than running what's often an underpowered trial? And Karen talked about the way to do this. There is a lot of math. There's a lot of decision points along the way. It does involve the prior, and the prior is sometimes thought of as a dirty word in regulatory settings. And, you know, I think we want people to react to this. If, again, we try to go to the prior to get information that we can't get by just enrolling more and more patients in a rare disease. Does FDA use priors when they review drugs? Of course they do. They use priors in a very informal way. I always talk about it like this. You know, I have to weigh the evidence I have and what I already know about the drug and come up with some regulatory decision. Well, Bayes methods enable you to formalize this and do it in a transparent way. Karen made that point, and I think it's important. It is a way to sort of put your money where your mouth is. What do I know about this drug? What do I believe about how it will work in this population before I ever start the study? Put that down on paper. Get agreement about it. Worry about the applicability to the next trial. Talk about how much you're going to discount it and so forth. It's almost like a cost-benefit analysis. What is the cost of me not using this prior information? Maybe missing a chance to answer the question that I most need to answer. Now, as John pointed out, this is new stuff in a sense, certainly newer for CDER than possibly CDRH and CBER. But how do you interact with the FDA on this? And I think John did an excellent job, as he always does, of going through the points that will be difficult. Can you seriously engage the agency and get agreement on a prior distribution prior to a trial? That's a new exercise in CDER. How do you approach this discount factor? What can we do here? I think that going back to Karen, the pediatric example is an excellent one to use for teaching. Not all pediatric diseases are rare, but we want to minimize the number of children we expose to drugs that are still not proven to work in children. So it's the same kind of problem of looking for substituting information. What's nice about the pediatric example is the adult data often are a really good source of prior information. If the disease is similar enough in adults and kids, if the mechanism of action of the drug should theoretically work the same in adults and kids, then this might be a way to go. So you've got a source of data that the FDA has already accepted to approve the drug in adults. Is that a reliable enough source to inform a pediatric trial? If you can think it through in a pediatric trial, then I think it's less of a leap of faith to take it over to another rare disease. And that's why when I was at FDA, we used that as a teaching example. And Karen had an excellent example today. But back to John and his problems, which are no longer my problems.
How much time you got?
So you've got the sticky wicket of the prior and getting FDA's agreement on that. You've also got a fundamentally different way of looking at decision making. And I think John did a good job of talking about they're already cutting me off. Talking about
that's okay.
talking about the way we make decisions, the way FDA makes decisions. All right. So can we have a paradigm shift and have a decision made based on the probability that the drug works, given our prior beliefs, our observed data in this trial, and the computation of the posterior probability, instead of making a decision that, assuming the drug doesn't work, what's the chance of me thinking that it works incorrectly, the type 1 error. So it's a paradigm shift in decision making. It's arguably, I'm looking at Frank Harrell, who's on IPA at FDA, one of the better things I did in my 6 years there. it's arguably a more natural thing to make a decision on. Does the drug work? It's a simple answer to a simple question. It's the answer that we most want. Not do I make a mistake if I say it works when it doesn't, but does the drug actually work? But we don't have a tradition of that. So how high does that probability have to be? 95%, 97.5%, 80%, 60%? Well, maybe it depends on the need and the other drugs that are approved. That also is different, although not without precedent in the FDA having some flexibility on the type 1 error rate in rare diseases. So, it is a paradigm shift. Can the agency do this? I mean, I think that's what we want to talk about today. Lucas is sitting on my left. He's at the agency. I think it's something that we need to discuss, but I think it's worth the discussion. John made a nod to decision theory, decision theoretics, analytical decision making, which fundamentally rest on cost-benefit analyses. You're used to the FDA talking about risk-benefit, which of course is critically important and takes into account the risk of the drug causing bad things to happen as well as the effect of the drug. But maybe there's a cost-benefit analysis. What's the cost of not approving this drug? What's the cost of not using the information we have at our hand when you have a rare disease that has no good treatments to it? And that kind of decision-making is easier to do when you have a quantity that's like the probability the drug works. So I think Bayes opens up the door to a more quantitative decision theoretic approach. Is the regulatory agency like CDER ready to take that on? I think we'll see. I think that's it. I just
Great.
wanted to commend the two speakers and hopefully tee up some questions.
Right, excellent comments. Thank you. Thanks, Lissa. Next is Lucas.
So I'm not here as the mathematician who's going to tell you exactly where you need to set your thresholds, but more as the clinician reactant. And I can sort of give you an idea of like how clinicians have been thinking about this, and I've been pulling people sort of informally around the agency who have to review some of this data as far as the clinical side. And like I like Lissa's example of pediatrics, because about 50% of rare diseases are pediatric diseases, and regulatorily, in order to do a drug trial in a group of children, you have to have a prior. You have to have prior evidence that there is going to be direct benefit for the children. So we already informally do this. It just would be interesting to see it a little bit more mathematically done in general. It'd make writing our reviews a little easier too, I imagine. So the things that I've heard from other clinicians when they look at these proposals are the first thing that people get a little uneasy about is the idea, because at this point frequently you're leaving the realm of the randomized controlled trial, and when sponsors come in and say, well, we're going to use a prior that's based on expert opinion, the purpose of the randomized controlled trial is to remove bias, and it sounds to the clinician like, oh, well, now we're just formally going to place bias into the mathematical equation, so therefore we'll win automatically. And so there's sort of this knee-jerk reaction of like, that doesn't sound like what we really want. Yeah, but for this audience, obviously, we all know that we're doing this mathematically, and it's going to be modeled in a more formal way. But some of the early proposals I've heard at the agency in the rare disease settings is not mathematical and not formally discussed, and so the agency gets this sort of pullback feel to it. And then the other thing that I've heard from clinicians is they really don't understand what the p-values are going to be. So say you do actually win, how do they interpret this p-value, and how are they actually going to put that in labeling, and how are they going to educate the general clinical community about what this drug actually does and what they should actually expect for it to do. So that's pretty much the two major things I've heard from the clinicians within the agency when they sort of talk about these sort of things.
Great. Lucas, thank you very much for the comments. We'll come back to this. I think it goes along with Alyssa's discussion about the paradigm shift and how best to and whether to accomplish that. Next is Gary.
I don't push the button?
Don't need to push it.
All set. Great, thank you. I'm used to pushing buttons. So there are just a couple of points that I wanted to make to add to everything that's been said so far. One point has to do with some of the examples that we've seen, and I want to go beyond that. So the examples that we've seen have looked at borrowing information with respect to outcomes, with respect to the endpoints, and how we might be able to be more efficient, that is, require smaller sample sizes by borrowing that information. There are other ways that we could borrow information that will be perhaps less direct, but I think may be equally useful, if not more so. And that is, as we understand more about the biology, the mechanisms of action of treatments, natural history of the disease, perhaps through registries, we can develop pharmacokinetic models, pharmacodynamic models, or just some kind of systems biologic characterizations of what's going on inside the body. And if we can do that, we might be able to borrow that sort of information and use clinical trial simulation, which I think we'll be talking about later today and certainly tomorrow. Use clinical trial simulation as a way of determining how much bang we get for the buck by actually incorporating some of that information and using that information to support or to buttress the inference that we're going to make from the study. I think in many situations it may be an awful lot that we can gain by doing that, and thereby require fewer patients to participate in randomized studies, and thereby be able to introduce novel therapies for smaller populations of patients. It would also require, I think, a broadening of the idea of what constitutes supportive evidence to justify regulatory approval. I believe there have been instances where the Center for Drug Evaluation, where CDER, has considered clinical pharmacology and considered pharmacometric modeling as support for indications. And I think that can be expanded and perhaps as another way that we can think about borrowing information from one population to another. The other point that I wanted to make was just to echo and fully endorse what John was saying about decision theory. I firmly believe that when we do clinical trials, we're doing it to make a decision. And we might as well make that decision-making process transparent and make it a formal part of our study design and allow for different people who have different sets of utilities. Some people might be more willing to risk things than others for the same benefit. But we should be able to present, or at least develop ways of presenting the information so that people can, in fact, make informed decisions using the information that we get from these studies. You pull an insert, a package insert from some new drug, and you lists all sorts of side effects that have been seen, but it's not clear what the how one should weigh those, what the actual relative frequencies are, and so on. So I think we can do a better job of that. But in the end, it's all done to support decision-making, whether it's by policymakers, by regulators, or by individual patients and their caregivers. And I think we need to focus on that a lot more in the way we design and characterize results of clinical trials. And then another, I think, important lesson that we in oncology have learned from pediatric cancers is the importance of having large numbers of patients participate in clinical trials. We've made tremendous strides in treating childhood leukemias, for example, And part of that is because the vast majority of patients have participated in randomized trials through the different oncology cooperative groups. And I think it's important that the government, but that there be some support to ensure that there are a large number of individuals who do participate in these studies, as opposed to, say, adult cancers where it's 5% or a smaller percentage of patients who participate in trials. So I think that's another thing that we need to focus on. The more patients we have in studies, in fact, the less, to some extent, the less borrowing we need, and the greater the chance of generalizing the results to the larger patient population. So those were my main points.
Great, Roy, thank you. I'm sorry, Gary, thank you very much, and I appreciate the connections as well to topics that we are going to come back to today, including the very important one of engaging patients and making use of that precious resource as effectively as possible. And so next is Roy.
Yeah, I just have three personal reactions to what I heard today, and hopefully will be brief. Was interesting to hear the comments from the physicians about the p-values. I think it's obvious to the statisticians in the room, but perhaps not obvious to other people. If we are going to allow prior information, external information into the inference, the only way to go is the Bayes way, and therefore there are no more p-values. We have to move away from that in this space, and if that brings repercussions in the non-rare disease, well, why aren't we using Bayesian inference there? That's something for the FDA to consider. But I don't think we can continue to go back and forth p-value in the rare disease. I think if we're going to allow other information, it must be done in a rigorous way, and the Bayes way is the only way to go. So that's number one. Number two, I work with the NIH Rare Disease Network with their Rare Disease Consortia. Unfortunately, in my world, the amount of prior information before we start a randomized trial is really small. It might be a handful of case studies, a very small previous uncontrolled trial. We just don't have the luxury of 10 previous trials in adults or anything like that. So I guess my preference in moving toward the Bayes would be to stick with what Karen called the static priors, conservative, so that they don't bring in a lot of weight. I don't think you necessarily have to have the priors where the control and the treatment be the same, but they definitely should be conservative. And finally, my last point, Karen's example was on binary data. We have to realize that binary data is the worst data that we could analyze. It's all or nothing, and we should probably, in the rare disease, try our hardest to get away from that. I realize that there aren't scale data, but clinicians that I've worked with are very supportive of moving to just a small ordinal scale. So something like no or minimal improvement, modest improvement, very great improvement. The benefits to going to that might be on the same order of magnitude as incorporating prior information. So I guess to summarize, we have to go Bayes. I would avoid binary response, moving more toward ordinal, and try to go toward, at least in the starting, to static conservative priors. Those should be easy to work with, and their operating characteristics very well understood before doing the trial.
Thanks very much, Roy. So I appreciate all of the perspectives we've heard, and before we, and last but certainly not least on this panel, I want to hear from Gigi, and Gigi, really appreciate your being here with us today and bringing a patient perspective to this panel.
Thank you for having me. If I wear my bioethicist's hat, I would just like to bring to the forefront of our minds the guiding principles for ethical research, which are non-maleficence, don't do harm, beneficence, do good, autonomy, the subject speaks for themselves, and justice, equitable access to research, and a good balance of risk and benefit to all subjects. So with that as sort of the foundation, I'm going to put on my patient subject hat. And what I noticed in the previous slide was a reference to data sources, and there was expert opinion and individual patient data listed as sources. I know we're talking about quantitative data in this instance, but I'm going to make a push for qualitative data. I hear the voices of more than 1,000 families that I've worked with whose children had brain tumors, and these parents, these parents say to me, "I am the expert on my child's tumor. I'm the expert on my child. I have valuable information, and the doctors are not asking me what I know. The things that I know are not being put to good use. Why? Why is that?" And I would say to them as a mentor, look, you're in this medical emergency right now, and the key concept is survival of your child. That's what everyone's focusing on. But in the world of pediatric brain tumors, there's about a 65% survival rate. That doesn't sound good to you, but we have worked hard to get to that level. So that means that after the medical emergency, there's 65% of these patients who have a long-term life expectancy. And what is the quality of that life? That becomes the question. So I want to give you an example of the kind of information that parents have that they teach each other because the doctors or researchers aren't asking. So there are some children with a certain brain tumor who have surgery immediately and then very shortly have 6 weeks of radiation, and shortly after that have some kind of a year of chemotherapy. That was the case in my son. The doctors who are dealing with a sad, sick child and complicated radiation and chemotherapy combinations and distressed parents say to the parents, do your best to keep your child's weight up, but don't worry about it. We can always give them a nasal gastric tube, an NG tube, if we need to. It's not a big deal, and that's true. But an experienced parent will pull the new parent aside and say, do whatever you can to keep your child's weight up during radiation and in chemotherapy. It is very important. And why is that? Well, the parental experience says, if my child's weight is maintained at a certain level, we don't need the NG tube. And while NG tubes give crucial nutrients, they are uncomfortable, they are stressful, they inhibit the physical activity of the child, they are a daily reminder that the child and the family are under attack. This is life-quenching circumstance on a daily basis, that just this one NG tube that, yes, is providing nutrients, right? That this life, this energy dampening over a long period of time in the beginning of what could be a very long journey is so important, and it impacts outcomes much later on. It's like a lever, right? The little change at the fulcrum affects greatly what happens at the end. This eating example is just one of many topics in the body of knowledge that parents have. There are certain kinds of rehab that, yes, strengthen the child's body, but do nothing to help the child re-enter a social world, right? Maybe a gym is better than the lady who comes to your house with the yarn and stuff. The appropriate educational scenarios are another thing. Early intervention for vision and hearing issues. If these are young children who have these kinds of impairments based on their tumor and treatment, this developing brain needs to have access to all the stimulus so that later on, 10 and 30 years down the road, they will have been able to leverage these skills and to be a more functioning member of society. Okay, so given this is subjectively reported information, it's qualitative research. I understand we're talking about context here. So what kind of conversations can we have with these patient families to discern what are reasonable kinds of information to gather, and how can we gather it, how can we maintain it and make it usable. How do we do that? You know, the FDA has a wonderful patient representative program that look and advise about drug development. I'm thinking that maybe there needs to be a different arm of patient involvement, of subject involvement, that looks at the qualitative measures. Where would we find that? I'm telling you, there are patient groups and disease-specific organizations that have resources, that have manpower, that have expertise, and are motivated to do something that's going to help our researchers make the strides and the progress that they want to make. There's this thing called trauma mastery. It's when you are victimized by something, so then you go do something else to help you gain control over this thing that victimized you in the first place. Thus, I became the leader of a nonprofit organization for families whose children have brain tumors. I mastered my trauma in that way. I did some really good things, but I helped a lot of people. So I would say harness the energy of these patient organizations to look at this, this area in which they are the experts, the qualitative, day-to-day stuff that in the long term makes a big difference. I say, yes, grab as much data as you can, however you can, look to historical measures. Maybe now build in a different kind of data collection so in the future they have something to work with when they look back to us. Thank you.
Thank you. Thanks very much, Gigi. So I'd like to start out by going back to Karen and John, see if you all have any short comments, reactions to what you've heard from the reactants. And then there are a few topics I'd like to follow on before opening up to the group.
Great. Thank you very much. And thank you to the reactants. In particular, thank you, Gigi, for that patient perspective. That's always extremely enlightening and helpful to be reminded of what parents and their children are going through. And I really like this idea of how do we use sort of redefine expert to make sure that we're not necessarily meaning the physicians, but the parents and those who are dealing with this day to day. I think many of the similar things that I had touched on about the role of expert opinion would still be relevant here when thinking about how do we get that information. I don't recall to what extent I talked about it, but there is an extensive literature on this topic and ways to collect information in a more rigorous manner that it can be used in an analysis. So thank you for that. And I appreciate also all of this discussion around rethinking the frame, and I think that's one of the key things that I think almost everyone touched on, that if we're going to do this, we will be Bayesian, and our decision-making will be based on Bayesian probabilities. And as I mentioned, that's another spot where I think bringing the caregiver perspective, the patient perspective, and thinking about, what would we need to see, what level of evidence would we need to see, and how does that translate into a posterior probability to be confident that that this particular treatment is effective. So thank you.
So I found all of those reactions very helpful and stimulating. I in particular, like Karen, found Gigi's comments really motivating and clarifying, and I appreciate them. I only wrote down one specific comment, which says p-value Lucas. Roy beat me to it, but I want to extend an apology on the collective guilt of my profession that we've traumatized you into thinking not only that p-values are the way to measure effectiveness, but even that they might be a way to measure effectiveness. And I think it would be helpful for us to think of ways of presenting evidence and presenting data in a way that, as I think Gary said, helps patients make their own decisions, helps providers guide patients to make decisions, and is interpretable in a human way.
Okay, great. So great discussion. I do want to build on the point that several of you referenced about, and I won't pick on p-values for the moment right now, but about this being a paradigm shift in interpreting evidence. And I'm going to come back to what you all think and push a little bit more on what might be the best way to approach that, if that is the right direction, so we can do a little bit more kicking the tires and all. But before I get to that, I just want to follow up on some of the discussion we had that goes to the question of how much of a difference will this make. So on the one hand, heard about that we cannot do or cannot expect to do sort of normal, well-powered, standalone, frequentist clinical trials here just because the numbers aren't there. On the other hand, because this is a limited size population, there is a concern that there may just be limited data to bring to bear anyway. And I think Roy brought out that point. That doesn't mean we couldn't do better. I think some of Gigi's comments really powerfully went to that issue. But just would like any reactions from you all on how big you think the gain could potentially be, at least in some circumstances, maybe some good early opportunities for making this paradigm shift. I mean, it really does depend on the quality of and the amount of the outside evidence that could be brought to bear.
Well, I'll start off. No, that's an excellent question, Mark, and I think Roy pointed out one of the problems is that there are many areas in rare disease where we don't have a good source for prior information. And if there's no good source for prior information and you can't run a fully powered study, you're left with a single-arm study, so how is that different than what we're doing today? And I know in the public meeting tomorrow that John already made a plug for, I'll make another one. I use a recent rare disease approval as an example where the agency has accepted things that are not standard. Of course, in oncology, Raji's in the audience, I believe, single-arm studies are more likely to occur, but oncology has better endpoints in a way. You can't say better outcomes. It's a terrible disease, but sort of unambiguous endpoints. The very place where you need information and can't get it in rare diseases, you're often using some kind of patient-reported outcome or some measure of patients functioning, and the FDA has requirements for the validity of those outcomes, but you have to test them in the population you're studying to show that they're valid. Well, if you can't get enough patients to run the drug trial, where are you getting the patients to test the validity of the outcome? And I will just say we had a section on endpoints at our statistics think tank meeting, and it was very informative. I think Laura Lee might bring up some of those points later this afternoon in the closing session. But this is a big deal, the choice of endpoints. So I'm rambling a little bit, but I wanted to get that in. Whether there's more ways that we could get things validated from the patient perspective, maybe we're not giving enough credence to the patient voices that are out there, as Gigi said. So I'm not really answering your question, but I'm throwing out more things to think about. But I want to say one more thing. When there isn't, my answer to it is it may not be as big a paradigm shift as we think if there's no data to borrow. Yeah. But when there are data to borrow, then you get to the paradigm shift, and I think we should get ready for that. I think the FDA could get ready for that, and I think the FDA has tried to do that. Source of data, though, Gary mentioned this, the pharmacology data, and I believe Isam is going to be on the final panel. The head of the clinical pharmacology office at FDA, Isam Zane, is going to be on the final panel. The drug-resistant pathogens and antibiotics that Dion Price, who's in charge of the meeting tomorrow or moderating the meeting tomorrow, has been working on, and I worked on when I was at FDA. That's a place where we thought we could do something with other sources. Maybe we don't have early trials in people, but we have in vitro trials showing that the antibiotic kills the bug in the dish, so to speak. We never were successful when I was at FDA trying to figure out a formal way to incorporate that information into a clinical trial. It was a research question we left hanging out there. But if there ever was a chance, that ought to be it, the drug-resistant pathogens, and maybe Gary's working on that now, I'm not sure. I know we're trying to get a relationship with Tom Lewis. The FDA is about working in that area, and Frank... Yeah, and Frank Carroll was looking at that as well. But that could be an example, like the pediatric trial, where you do have a source, is a great teaching example to take the agency through the steps that would be required. The antibiotic space, where you might be able to figure out a way to borrow data from preclinical studies, maybe that could be the teaching example or the place where the research is needed. I don't know.
Great. Other comments on this topic?
Yeah, I'm aware I'm a part of a PCORI grant with Kelly Kidwell and Tom Braun at University of Michigan, where they're actually looking at some of the question, how much benefit is there to using innovative trial design, Bayesian inference, conservative priors, and paper will come out soon. But I was actually surprised at how just statistically, you know, what we call mean square error, the precision was improved by some pretty conservative assumptions that were not even true, were violated, but even in that case, there was a fairly good benefit. I'm not saying you go from sample sizes of 100 down to 2, but it was definitely eye-opening to me. I'll put it that way. It was worth doing, to going down this road.
Okay. Now, let's presume then, at least for the moment, that there is reason to think that this paradigm shift is worthwhile, at least in some early cases where there is either good reason to believe that the additional data and evidence would make a difference, or to Gigi's point, by making this shift, you could potentially generate more attention and effort around additional types of evidence that don't really have a pathway into the decision-making process now. It is a paradigm shift, and I heard 4 or 5 different versions of why this is going to be difficult in terms of needing not only to reframe away from a focus on p-value, but also to consider a framework, a decision theory framework for how to weight this shift away, and maybe a number of considerations in terms of where and how that might be applied. You all have a tremendous amount of experience in this field and FDA regulatory issues. Are there any additional tips or thoughts about how to begin this paradigm shift?
So I was sitting and thinking about Roy's comments and Gigi's, like, sort of right after that. Part of the problem we have in rare diseases is, you know, we have the small number of patients, but then what we've been seeing in a lot of programs, you know, a lot of them are being run by, or at least supported by, the patient organizations that are interested in that group or in that disease. And what they see is a series of, you know, failed trials. They try, something comes up, it's very promising, it fails. You know, 20 people are in that trial. They only have a couple hundred people with this. The next trial takes another 20. Next trial takes another 20. But since a lot of this drug development's all being generated, it's a business, so it's that data is not available to the next trial. We can't borrow that data. And then it goes on, the next trial, those 20 are lost to any future borrowing, next 20. And so I think within the patient community, there has to be a little bit of a thought of how they're going to shift that paradigm of how, if they're going to be partnering with these companies and using their very small, valuable resource of their patients, they need to think about how they can partner in a different way. I think possibly that may be part of the paradigm shift, because like our statistician colleagues say, like, where is this information coming from? Like, it can't come from, if you're gonna do, this isn't antidepressants. We don't have 30 trials from 30 different companies, and each company doing 5 or 6 different studies with hundreds and hundreds of people. So this is just a handful of people, and where is that data coming from, and where is it going? I understand the partnering a lot with natural history studies is where we're getting a lot of the advances in cancer, because at least we have our numbers. We know what the rates are. It's hard endpoints, but in a lot of our other diseases, you know, it's small improvements are very clinically meaningful. You know, if you can walk for a little bit longer, if you can, you know, lift your hand up to be able to feed yourself for a little bit longer, that's, for that individual, that's a clinically meaningful response, not necessarily survival. Just that's where my thoughts were about the innovation, at least.
I have a thought. Scott mentioned the importance of background care, and then you move really quickly on to the next thing. One of our families said, you know, this is like my child is a plant and he has a bug. And so the doctors are coming in with a chemical to kill the bug. And the doctors are really looking at how does this chemical kill that bug. In the meantime, I'm the gardener, and I've got to water the plant every day. And I've got to make sure it gets lots of sun. And if I don't do that, the plant's going to die no matter how good that chemical was, right? So that just reminds me, if I'm such a visual person, but I'm seeing this field of plants, and they all have different bugs, and there's all different kinds of chemicals being used. But what's the one thing that or what are the things that are generalizable to the whole population that you could target? And that would be the background care, right? I think that's a really valuable place to look.
Maybe just an additional comment on making this paradigm shift. Something that I think would be really helpful is to ensure that we do have clear guidance on best practices. So I know in my talk I talked about a few different metrics and types of simulations that one can look at. And I think that would be really key to ensure that when these proposals are coming forward, that we're doing so in a consistent manner and we're looking at things prospectively. And I also would just add to that the comments around thinking about the entire clinical program, so that if we are in situations, I know this is not always the case, but we are in situations where we're going to go into adults, we're thinking about what data might we need to be collecting to ensure that there could be something borrowable, able to be borrowed in the pediatric setting or whatever the situation is, really thinking prospectively across the program.
Just not exactly a counterpoint, because I agree with that, but I think when I think of how we're going to get to a paradigm shift, I think of the need for case studies, just in specific individual examples of decisions being made in a principled way, of data being borrowed in a way that makes sense, and going from those individual case studies to providing the guidance that
That's right.
can be helpful for.
Yeah. Perfect for the pilot program.
Right.
Yes.
Just a couple other things. So the importance of case studies is critical. We did, in the Office of Biostatistics, develop a great case study for borrowing adult data in a pediatric trial that was presented publicly. And I know somebody in the office could mail out the link if anyone's interested. It was presented publicly last year. But in general, you know, the reviewers at FDA can't talk about the protocols they're looking at to review. And so it makes it hard to talk about the things we see that we like or don't like. And until the drug's approved and things are public, or the drug goes to an advisory committee, there's really no use that we have. But we have proposed a pilot study that's going to be showcased tomorrow to address exactly that under the prescription drug user fee number 6. So that's important. The other part of the paradigm shift, two quick things. So the sharing of information. We have a session this afternoon on master protocols, and I think that will be talked about quite a bit, how to better share patient resources. And the patient advocacy groups are so critical to making those things happen. The other paradigm shift is Roy's comment about the endpoint. It is the natural thing inside the agency, I found, for people to talk about responder analysis. They want it. Now, there's issues with responder analysis anyway, because patients responding in one arm, you don't know what they would have done in the other arm. So saying someone responded to the drug is tricky, but they could have hit the dichotomous outcome, which is what we really mean. The loss of power, but then the heterogeneity of rare diseases, making that what clinicians want to use. I think they butt up against each other, and it's a real problem. You've got very heterogeneous disease, people coming in wheelchairs, or able to walk a few steps, or able to walk quite a lot, and now you're using a 6-minute walk or an ambulation outcome. What does it mean to be how do you come up with a continuous outcome that's the right outcome there? People tend to try to define response at the individual level, but that's the outcome that you'll have the least power for in a small trial. So it is a problem, and I think that's as big a paradigm shift almost as what would happen with p-values and probabilities of success.
Yeah, so great points. I do think we're going to come back to this issue of patient perspectives and patient-driven efforts around data sharing and maybe support for platforms, especially in session 3 today, and it sounds like that'll come up a bit tomorrow. I do understand maybe from an individual company perspective concerns about sharing proprietary data, but here with the numbers being so small compared to other disease areas, it seems like the cost-benefit trade-off there might be different, especially if there's an organized patient community around it. But I would like to come back to the challenges in that area later today. For right now, just one more follow-up on the paradigm shifts. So we've discussed a lot of the challenges and a lot of the ways to overcome them. Any quick thoughts about the best way to present what comes out of these new kinds of analyses? Getting back to your question, Lucas, about this is not what clinicians, and maybe to some extent not what patients are used to seeing and thinking about.
I'll take a quick stab. I think a sort of first step of how you would summarize this is moving from p-values, which are measures of the probability of observing data, to summarizing things in terms of the probability for a patient that they will experience a benefit or that they will experience a harm. I think that's a good and almost easy first step in some senses.
Okay. I see nodding on the panel. Okay, good. I would like to open this up now to questions and brief comments, actually brief questions or brief comments from those of you who are here. And remember, we have a mechanism for those of you who are joining online for you to send in questions or comments too. So when you ask your question, please try to be brief and let us know who you are.
Sure, thank you. Is this on?
Yes.
Okay, my name is Famita Gwadri-Sridhar. I'm a clinical epidemiologist and a founder of a global registry company that works in rare disease and cancer. I have two brief questions and/or comments. The question about losing patients to trials where there's trial failure, the idea of potentially using registries that are not tied to a company but rather disease-specific, is there, I mean, I know that there's going to be discussion about that today, but that's certainly a mechanism to start to address that. The second is really around the p-value point that was raised. And some of the work I had done with Roman Jaschke and Gordon Guyatt really focused on the clinically important difference, and the minimally important difference is a different way to start to evaluate the impact of whether or not a therapy has an effect in a patient. And maybe, Lucas, you'd like to comment on that, or Roy, MID, CID, moving away from the p-value, looking at effect size as a potential outcome in the rare disease space.
Great. I want to save your first question for our next two panels, but good points to bring up for them.
So I would just say, I think to address the second question, in the example that I gave in the adult study, borrowing from adult into pediatric, the statement of success was the probability of that effect being greater than something. And that's that clinically meaningful threshold. And I think that is the question that just came, how to set that. And certainly that is where all of the expert discussion, however we define expert, and expanding to caregiver, clinicians, regulators, patients, ensuring that that's brought to bear to establish that threshold. But that's where it would come in, would be that probability of the effect achieving that level, and then the threshold, the probability threshold is how certain do we need to be.
Thank you. Yeah, go ahead.
So just quickly, I think something that we need to do to get along, going along the lines of trying to address this issue of reliance on p-values. Well, something that we need to do within at least biomedical research is to do a better job training or teaching students in medical school what it means. Right now they get very, if any, they get very short shrift sorts of statistical training. I know last week we have an oncology journal club for oncology fellows, and we were discussing a study that was published in The Lancet, and which is certainly a reputable medical journal. And there was a difference in the hazard ratio. One treatment group roughly had it was a 70% hazard ratio, so they did better, but it didn't reach statistical significance. The p-value was greater than 0.05 for a number of reasons, having to do with the study ending prematurely. But the point is that the statement at the end of the paper was that the treatment didn't affect survival. If you look at the hazard ratio, it did. If you look at the curves, it looked like it was going to, but the p-value didn't support that statement. And so the editors of the journals rely on the p-value. The fellows read that, they rely on the p-value, and that's the way they're trained. And so if we're going to affect a change in how clinicians evaluate medical evidence, I think we need to start at schools of medicine.
Yeah, paradigm shift. Okay, we've got about 8 minutes left. I want to get through as many of these questions as possible. Let's go here and then over to...
Tom Lewis, Johns Hopkins. 2 points, neither having to do with p-values. And the first one is, nothing is really radical here. The whole history and future of statistics is combining evidence. When you compute a sample mean, you're combining evidence over what is generally disparate sources. So I'm not trying to make this be a non-issue, but we ought to keep in mind there's nothing terribly radical here. It's more a question of how far to go. And now on a more specific point, we don't talk enough about using Bayesian methods to take advantage of information that isn't the primary endpoint, the best example being measurement uncertainty. And especially in small trials where you may not have the degrees of freedom, especially if you need to support individualized variance estimates. And it's maybe the best entry point. It's not quite as controversial as combining on the endpoint to say we will get personalized variance estimates by empirical Bayes, but we will do it by combining evidence over the ensemble of variance estimates we have. And so I'll just leave that.
Great. Thanks very much for the comment.
Yes, I'm Neil Bellson. I'm an attorney with Potomac Law Group in Washington, D.C., and increasingly I'm focusing on precision medicine. And my question is specifically for Gigi, and I'm wondering if there is any institutionalized or proactive mechanism to collect that sort of family data that you've described, because as you point out, it is expert information. You're living with this day and night, but is there any sort of institutional way to collect it, the family experience, to put it into some sort of aggregated way so it's available to physicians in a quote-unquote scientific or formalized manner, so it is seen by them as scientific or useful guides for their, as for practitioners, and even conceivably, and I may be way off here, I'm wondering if that data, if organized institutionally, could even be useful in designing clinical trials, i.e. this drug with this background care. I don't think you'd ever want to give bad background care in a trial, but I'm just wondering if it even could be useful for designing clinical trials, but certainly for physician care.
And we will come back to this topic later today too, but please go ahead. Yeah.
I just want to then put there's an academic journal published by Johns Hopkins University Press called Narrative Inquiry in Bioethics, and I am affiliated with that journal. But it does collect personal stories of patients and then has experts comment on the themes that are woven through the collections, and it's highly credible and in a scholarly format, so I can talk to you more about that. Yes. But otherwise, it's just mostly the disease-specific groups. They have their own internal data that they share with each other through peer mentorship. There isn't any widespread collection of this data that I know of.
But this isn't something that normally physicians would look to in treating a patient as a first line.
Yeah, the patients and families access this among themselves, and usually the doctors are not accessing that.
And we will come back to how we can get better registry data and platforms for using additional data later today. So please go.
Chris Holland, Amgen. Hopefully this is a question that we're not going to talk about later, or maybe it would be a good thing if it is. But we've heard a lot today about the use of Bayesian methods, borrowing for the setting of rare diseases, but there's also plenty of examples of an entirely different approach being used for the same situation of rare diseases. And that's one of using causal inference methods, such as propensity score methods, to synthesize both observational historical data with clinical trial data. So I would appreciate some thoughts on the pros and cons between these two approaches and maybe examples of where one approach might be better than the other. Thanks.
Just very quickly, they're not mutually exclusive. There are plenty of people, myself included, who will use propensity scores or some other things, but within the context of a Bayesian inference. So they're not at all mutually exclusive.
Thank you.
Frank Harold, FDA and Vanderbilt University. I just want to suggest that we need a paradigm shift whether you borrow any information or not. And the paradigm shift is making better decisions and using the right metrics for making the decision. So an analogy would be, and this is just trying to show that optimum decisions come from playing the odds and knowing the gains, and you're trying to optimize your gains. So a poker player is not looking at her hand saying, of all the times I lost a round, how often did I have a hand as good or better than the one that I have? But she is saying to herself, what's the probability I'm going to win this game with the hand that I have now? And I think that's the simplest explanation I have of the paradigm shift that's really needed towards Bayes. And it has a lot to do with decision making, not just the formal way that Thelma Irony can talk about, but informal decision making, because if you have no available alternative therapy and you're trying to decide whether to let a drug out on the market, if the p-value is 0.1, there have been trials that FDA has seen where the company said this is a failure. But if the probability that the treatment benefits patients is 0.8, if you're playing the odds, you have a very clear decision to make. It is because you're playing the odds on the scale that allows you to play the odds.
Great. Thank you very much for the comment.
Hi, I'm Thelma Irony from the FDA CBER, and I have two things to comment. One thing is I'd like people to consider when they think about using prior information from experts or from patients, being a bias to the trial or to the data. And you can think the other way. If you don't use information that you have, you're also biasing the data. So, it goes both ways. And would you like to bias with information from an expert and from a patient, or, just any bias? That has to be considered as well. Second thing that I wanted to comment on is related to that Dr. Harrell's mentioned, is about not having data to borrow from. So we'll have to learn how to accept more uncertainty. When we do a benefit-risk consideration at the FDA, we'll have to consider the risk of not being treated, particularly if you're talking about a life-threatening or a permanently debilitating disease. If you don't accept more uncertainty, you might not be able to approve a treatment. So that's another way to say the same thing about the odds. Thank you.
Thank you very much.
Claudia Morris from Emory University. I'm a pediatric emergency medicine physician, and I've been running clinical trials on sickle cell disease for over 20 years. And I think this is really exciting. How do I have one positive phase 2 trial, and I'm in the middle of developing my phase 3 trial for arginine and sickle cell? How do we translate these innovative strategies to acceptance at the FDA level, like right now, as we're developing these trials? And how long do we have to wait?
Well, I think we've been talking.
I think we're out of time. No, it's an excellent question. I would, not to be a broken record, but I would refer you to the discussions we're having tomorrow about the pilot program we're starting, which is a way of trying to provide more interactive and intense feedback to people who are proposing complex and innovative trial designs. And I think this kind of thing would definitely fall into the general category. So it's a way to get FDA buy-in on a strategy before committing to the trial.
But it's also a moment to plug Julie Bites in her Office of Inborn Errors, because that Laura Lee Johnson, who you heard from today, supports, because they often do interact with sponsors in pretty severely rare disease cases where studies are just so hard to run. The pilot's going to formalize that interaction, but a lot of that happens thanks to Julie right now.
Yeah, well, it sounds like definitely contact FDA on this. Last comment?
Thank you. I'm Mike Proshan from NIAID. I absolutely agree that I think this is an area where Bayesian statistics is very attractive, but I absolutely disagree with the sentiment that p-values have no use and ought to be banned, which is proposed by some people. So I think you can go too far. I think p-values do have a place, especially in a regulatory setting, but I agree that in this setting, I think Bayesian methods are attractive.
Okay, great. I don't know if those people want to make any comments about p-values.
I think it came from Roy.
All right.
Just kidding.
Seeing some agreement. I want to thank all of our panelists and all of you for a great discussion during this first session. Thank you very much. All right. We're going to take about a 10-minute break, start up right about 11 o'clock again. So please stretch your legs. So I'm going to go ahead and introduce our next session while you all are headed back into the room. This is a session on utilizing patient registry and natural history study data to advance therapeutic development for rare diseases. As you all heard during the first session, there is a lot of interest in using these sources of data, not just in the context of potential shifts towards more use of Bayesian methods in rare disease drug development, but in their own right. And we're going to talk about that in this session, as well as connect to the discussions we had earlier this morning. We're going to start this session off with a presentation from Nicole Meyer-Hamblett, who is going to talk about the use of patient registry and natural history data in rare disease settings. Nicole is an associate professor of pediatrics and biostatistics at the University of Washington and the co-executive director of the Cystic Fibrosis Therapeutics Development Network Coordinating Center at Seattle Children's Hospitals. Next, we're going to hear from our lead reactant, Petrula Simpoku, and panelists. Petrula, I'm going to go ahead and introduce everyone now. Petrula is the lead medical officer in the Division of Gastroenterology and Inborn Errors Products at the Center for Drug Evaluation and Research at the FDA. We're then going to hear from Randall Bateman, professor of neurology, director of the Dominantly Inherited Alzheimer's Network, and the director of the Dominantly Inherited Alzheimer's Network Trials Unit at Washington University in St. Louis, and then Yefeng Chen, who's statistical team leader in the Division of Gastroenterology and Inborn Errors Products at CDER at FDA, Petra Kaufman, Vice President R&D Translational Medicine at Avexis, and PK Tandon, Senior Vice President of Biometrics and Development Strategy at Ultragenyx Pharmaceuticals. So you can see why we've got a lot of ground to cover in this session, and we needed to get started. But right now, to kick us off, we're going to hear a presentation from Nicole Meyer-Hamblett. Thank you, Nicole.
Thank you. Well, we'll definitely be switching gears here, but definitely building upon many of the themes we discussed earlier today. And I'd just like to start by centering ourselves on our common mission of delivering safe and effective therapies to our patients. And in the rare disease setting, it's absolutely critical that we maximize the use of our patient registries and natural history studies to support our drug development efforts. We have to understand the disease to enable efficient drug development. And this really begins, we need to start by ensuring we have sufficient data resources to first and foremost identify our patient population and then track their disease course. Now, there are some subtle differences between registries and natural history studies. Patient registries can be rather broad in scope and objectives. For instance, they can be used for patient recruitment efforts and communication. They're often established to fulfill post-marketing commitments. But within them, they can also include an embedded natural history study. And these are really those studies that are meant to describe the disease irrespective of any investigational product. And they enable tracking disease course on a much more comprehensive and granular level. The Cystic Fibrosis National Registry is probably one of the largest and longest rare disease registries. It was established in the 1960s. And as time has gone by and technology has grown, so has its data collection capabilities. But despite these changes in data collection over the decades, one thing has remained very clear and focused of this registry, and that has been capturing a representative patient population within the United States. And in fact, this registry captures about 84% of individuals in the US with CF. Now, I know that 29,000 patients relative to many rare diseases seems like a lot of patients. I will point out that there are over 1,700 different CFTR mutations, which less than 50 CF adults carry. And this has been a considerable challenge for us as we are now developing mutation-specific therapies. So it's our job now to identify who and where these patients are. And so for our drug development efforts, we have now linked with our registry and real-time reporting to our clinical centers to generate reports of potential eligible patients to fill each of these trials in our pipeline. And this is really an example of where just a few key clinical parameters on select eligibility criteria within our registry, but collected on a large patient population, can have quite a powerful impact on drug development. You don't need a lot of data to make some big impact when it comes to drug development. There has been significant investments in many rare disease registries and natural history studies over the recent years. And in particular, I'll mention that NORD, in partnership with FDA and NIH, have now built a standardized platform, an affordable platform for other rare disease organizations to build their own registries without having to reinvent the wheel. But as we look towards the future, towards building these data resources, we need to partner with industry, and we need to change this paradigm to build disease rather than product-based registries. Of course, this avoids duplicative infrastructure, but it also removes the limitations of product-specific registries. These product-specific registries may limit eligibility, for instance, following only patients who are on label with a particular drug, or they may remove patients who have stopped taking a drug. They will have limited enrollment size in comparison to the entire disease population, and they may lack a comparator or a control arm that's so critical to doing comparative effectiveness research. And there's also some low-hanging fruit. I think this was mentioned earlier this morning. Complementary natural history data is available through completed clinical trials. We have actually had great success with placebo data donation from industry sponsors to a central data archive. So while there may be concerns about donating data from industry-sponsored trials, that placebo data can be quite rich. And we've had success partnering with industries through data use agreements for donation of that data. And the beauty about this data is that it mitigates issues related to missing data and endpoint consistency inherent with registry and observational natural history data. And now that we've built these data resources, but then we can do what I like to call therapeutics-driven epidemiologic research, which is really focused on informing our study design, endpoint selection, and also helping us to interpret our clinical trial results. When designing trials, it's very tempting to start with sample size, even though there are so many other study parameters to worry about. Here is sample size curves for a typical event-based endpoint, and you'll see that it really depends on the expected event rate in the control group. And if we are powering a study to observe a 40% reduction in an active arm as compared to a placebo or control arm, there's about 100 patient difference for just a 10% difference in that control group proportion. That's huge, and that's unbearable for a rare disease setting. So really, we need to step back, and this is where we put our registry and our natural history data to work. Not only do we have to understand what are the event rates of this endpoint, which we can kind of glean from our registries, but we need to go further. We need to understand, are there patient subgroups with higher rates of this endpoint, such that we can maybe enrich our study population and reduce overall recruitment burden. So, we work with our sponsors, and we have in an industry consulting program that we have the infrastructure to do time-sensitive queries of our registry to support this type of epidemiologic research for this purpose. We can look at key eligibility criteria and identify subgroups of patients that may have higher rates of this endpoint, and work with the sponsor to determine if we can enrich our trials and hence reduce our sample sizes without compromising generalizability of the study. But I will point out the limitations to consider with using this data. We always have to be mindful, do these event rates actually reflect the most current standard of care? Sometimes our natural history or registry data may be a little outdated as compared to our exact point in time that we're planning a trial. And is the endpoint consistent between the registry and how we will collect it for clinical trials? This is sometimes a big, major issue. Another example of this epidemiologic research to support drug development is providing context to clinical trial results. So I just want to provide you an example with respect to safety data. In the rare disease setting, and actually in any chronic disease setting, it can be very difficult to determine if adverse events are attributable to the new treatment or to the disease itself, even when you have a placebo group. In this example, hemoptysis is a clinically significant event for CF patients, and you'll see here that 6 patients in the active arm had an event and none in the control arm. You know, we don't do statistical testing on safety data, and if we did, it would be post hoc. You know, did we just get unlucky here? The 2% of the patients in the active arm experienced the event. Is this what we could have just expected, you know, with the chronic disease itself? And so we can go to our natural history data, and for this in particular, we went to data from completed clinical trials. We pulled together data from 8 completed randomized trials, either patients on placebo or standard of care therapy. And again, the beauty of this data is that we had standardized adverse event coding across these trials. And we can look at different patient subgroups and get an idea of the expected event rates of hemoptysis, and then toggle back to our clinical trial results and sort of put those results into context. And we can really go beyond just doing epidemiologic research with our registries if we're able to build infrastructure to transform them into a platform for regulatory studies. And this could either be as a source for historical or concurrent controls, or for the fulfillment of post-marketing studies. It is not uncommon in the rare disease setting that we either don't have enough patients, or it is unethical to do a traditional randomized placebo-controlled trial. And in this example, it was reviewed by actually one of our panelists later today, P.K. Tandon. It was in the setting of Pompe disease, and they were studying an enzyme replacement therapy with recombinant human GAA. And it was felt to be that there was a placebo-controlled trial was unethical. So they used historical controls to support the regulatory approval of this therapy. And here's the primary endpoint was survival-free from invasive ventilation. And here's the Kaplan-Meier curve corresponding to that. What you'll see here is that there was a very robust treatment effect. And as the authors point out, that this example really represents sort of an ideal setting for the use of historical controls. We have a very objective and well-defined endpoint of survival. We also have a very large, undeniable treatment effect. And so we need to keep in mind these attributes, these ideal attributes, when we look towards the appropriateness of using historical controls with our registries to support regulatory approval. And we also need to lay some groundwork. We need to, again, be very mindful of the consistency between trial and registry endpoints. Not just the definition of the endpoint. You know, when we're using something like survival, that's very clear, but that's not the case for other endpoints. But also the timing of data collection, and of course, missing data. We have to understand historical trends and clinical trial participation bias and the impact that may have. And ideally, our registry has built, our natural history studies have built an infrastructure for quantifying and ensuring data quality. The stakes are much higher when using the registry for these purposes as opposed to the epidemiologic research that I mentioned before. But if we're able to do this, it can be quite powerful. Not too long ago, enzyme companies were issued a post-marketing commitment to conduct a 10-year prospective observational study to assess the risk of a rather rare event, fibrosing colonopathy in CF. And because this is a rare event, the incidence can really be more accurately estimated by leveraging the entire registry. So our solution was to design a registry-embedded master protocol that was negotiated with 4 different industry sponsors to meet our post-marketing to meet the post-marketing requirements for each of the sponsors. And this really then saved the building of 4 different shadow registries to complete these requirements. And I think we are seeing every week practically new guidance coming out for rare diseases that allow newer and flexible approval pathways. And often these require post-marketing long-term effectiveness studies. And this is both true for the FDA and the EMA, and in particular for the transition to pediatric approvals as well. So another low-hanging fruit is we have an optional consent form to collect registry IDs in many of our therapeutic trials. And this enables long-term linkage and follow-up through the registry. Many of our CF trials, the randomized controlled portion of the trial is only 6 months before patients roll over onto an open label study. In this example of a therapy called ivacaftor, that's exactly what happened, and you'll see that there was a very robust improvement in lung function in the patients on this therapy. And we, as part of our post-marketing efforts, we wanted to determine whether or not we were also slowing the rate of decline in lung function over time, which required a much longer follow-up period. So we used the registry for two purposes. We not only followed the trial participants long-term, but we also constructed a control group to compare the trial participants to. And for this, we used propensity score matching based on risk factors for increased rate of FEV decline. And doing this, we did find that there is an association between ivacaftor use and slowing the rate of FEV decline over a longer 3-year follow-up period. But I will caveat this and say, the use of the registry and match controls is fairly tricky, and we need to be mindful of all the different sources of bias. Confounding by indication cannot be avoided. We've got some tools in our toolbox, including propensity scores and instrumental variables, but they're not foolproof. There is selection bias, that clinical trial participation bias that we need to be mindful of, and of course, missing data. And I will point you to, there's a very nice handbook by AHRQ that really summarizes all of these sources of bias and the different methods to address them. What we need to also be aware of is how these methods perform within the rare disease setting. So as we round out all of these pillars for how registries and natural history studies support our drug development pipeline, I will just end by saying that our greatest success is by partnering with our patients. We recently asked our community what questions do they want asked from the registry. This is their resource. We got over 1,000 different questions, and we had to prioritize and vote on them, and now we have patients on project teams to actually address those questions. And so this is really just our continued commitment towards investment in such an important resource for our drug development efforts and just for understanding the disease. So I just want to acknowledge my colleagues at the CF Foundation and the Therapeutics Development Network. Thank you.
Great. Thank you very much, Nicole. And could I ask our other panelists to come on up for the discussion? And as I said, we're going to hear first from Petrola and then from the rest of the panelists. But you all can come on up at this point. Thank you very much. All right, well,
okay.
Hi,
good morning. You can hear me okay? So thank you very much for this opportunity and for the very nice presentation. I think it really highlights a lot of the points and challenges when thinking about natural history studies and patient registries. So from our perspective, in our work in inborn errors of metabolism, and those are rare genetic diseases that really encompass a spectrum of enzyme deficiencies. As already noted, the knowledge of the natural history, of course, is fundamental, and that makes intuitive sense. But many times we are kind of taken by surprise when in a clinical development program, through interactions with sponsors or other stakeholders, there's really not a lot of information of what happens actually during the course of the disease. So I think I cannot stress enough the importance of information on the disease natural progression in terms of what body systems are affected, meaning what symptoms do you see, at what time points, how fast or how slow those can show up, at what ages, is it mostly pediatric, is it mostly adult, with what severity those may show up, which groups or even subgroups of patients within the patient population may be affected earlier versus later or more severely. Are there markers for diagnosis which can be used when enrolling patients in clinical trials? Are there useful biomarkers that can be prognostic of a particular disease course or rate of progression? And are there some biomarkers that may be even predictive of some clinical outcomes that can be leveraged during clinical development program or even for some approval pathways? And then are there biomarkers that can be useful for patient selection, potentially for optimal dose selection in a trial? And also, how long does it take for a disease symptom to appear or to get worse? Are there inflection points during the disease trajectory that we need to make sure that we are aware of, especially when comparing to some treatment effects? And so all those are very fundamental questions that always come up during discussions at the agency and with sponsors or outside stakeholders. But these questions need to be addressed to the degree possible before even starting a clinical development program, which means a lot of preparation needs to happen. So particularly in rare diseases, and in our area of work of inborn errors of metabolism, every patient in a clinical trial counts, and as we all recognize that, and that's why we're here. And we often emphasize that randomization really truly starts with the very first patient which enrolls in a clinical trial. So not only are there very few patients to study, there's a pressing need, as it was previously mentioned. Those are all serious diseases, and which are often progressive, and especially in children are actually more severe, usually, than in the adult population. So with all that in mind, preparation prior to initiation of any clinical trial is really paramount. And this is where information from registries, natural history studies play truly an essential role. And so I would emphasize is that really the knowledge of the disease natural history, natural progression, trajectory over time is a prerequisite really to designing informative clinical trials, especially in the rare diseases. But in order for this knowledge to be really maximally useful and to be leveraged within a regulatory context, studies that are designed to serve a regulatory purpose need to be very well designed. They need to follow a specific protocol. For example, natural history studies that may be used later on to either inform drug development or be used as historical controls need to follow a particular protocol, use standardized methods to collect data, document, code all data, and need to have a pre-specified statistical analysis plan, much so that we see in our clinical trial development. For instance, a natural history study that's designed to inform clinical development programs of therapeutics may follow certain standards, and it was previously mentioned, one of those may be a CDISC format for data, may use certain specific coding dictionaries to avoid any confusing terms or maintain some consistency with coding terms. And this is really all to aim to maintain consistency and standardization within those. Another example may be using guides that are used potentially for publications for natural history studies or observational studies. One may be the Strobe statement, which can be actually leveraged as a useful guide when thinking about designing good natural history studies. One other point I wanted to make, and it was previously mentioned, is this international or cross-group collaboration, cooperation in this area. What we often see and what we hear actually from patient groups is in a particular disease, a rare disease, for example, where there's multiple different products developed. It seems like a sponsor starts their own natural history study, and then another sponsor comes in for another drug for the same disease. There's another natural history study yet started, and so is that efficient? Is that really useful? And so where the collaboration, cooperation really becomes essential in terms of what was highlighted previously is sharing of the data. And so we see that a lot in our interactions, both with patient groups and with industry, in designing those trials, are really trying to move trials forward. In rare diseases, in inborn errors of metabolism, this is actually an important issue that needs to be really talked about openly with patient advocacy groups and sponsors. We do know patient advocacy groups are very willing to share their data, their registries or natural history studies, but there does need to be a lot of openness and cooperation to that effect. So another potential use of the natural history studies that was highlighted previously is to use as non-concurrent or even concurrent control groups to which the treated group in a trial may be potentially compared to. Of course, as we hear, we use statistics. It really comes down to comparisons. And so we do see a lot of open label trials or single arm trials in rare diseases where the thinking is that placebo may potentially not be ethical to use, and that can be a different topic of discussion. But really at the end of the day is we need to be able to compare it to something. If we don't have a concurrent active treatment or placebo group, then we need to have something to leverage. And so that's where natural history studies may potentially come in. But this particular use needs to be especially carefully planned. And so in this context, I know we heard the example about Pompe disease, which is actually a great example of using natural history information. But the caveat is what was previously discussed, that this is a uniformly fatal disease within a few years. So you have a very objective measure, which is survival, which is really a binary measure, as previously discussed. And there was a very large treatment effect. In the rare diseases that we work on, in the inborn errors of metabolism, we often don't really see large treatment effects, and that is for multiple different reasons, one being the disease heterogeneity. And so in that sense, it becomes really challenging to use natural history data or historical controls. That's not to say it's not possible, but I think it needs to be very carefully planned ahead of time, and it has to be disease specific. And I just wanted to finish by saying that from our perspective, we really encourage very early communication with the agency and with our division. There's multiple ways to do that, and it depends on the types of questions or inquiries that are raised, and also the type of disease. There are very many resources that the agency is in the process of developing and streamlining. And one example that people have used before is with a PACE group, where external stakeholders can now actually request meetings online on FDA.gov website. It makes it very easy, where you click a button, you submit your request for a meeting, and the staff of the PACE group actually coordinates with the external stakeholders, identifies the divisional office which needs to be contacted and has the expertise, and everything gets scheduled actually in a very streamlined way that way. And we've had actually a few meetings like that have been very useful in the past. And the other is the CPIM, the Critical Path Innovation meetings, and so I'm sure most of you know of those. We've had many of those before, which are not product specific, but they are disease specific or disease group specific, in which the agency is engaged with outside stakeholders, for example, patient advocacy groups, and the agency is able to provide this non-binding scientific advice in order to move things forward in drug development in a specific disease. And I think there's continued efforts to streamline a lot of those avenues for communication. So what I wanted to really emphasize is that in terms of natural history studies, registries, if there's a regulatory intent and there has to be a goal in mind, and there's usually a goal in mind when such studies are initiated, we always encourage early and continuous communication, and we're very open in engaging those types of conversations. Thank you.
Great, thank you very much, Petrula. Next is Randall from WashU at St. Louis and the Dominantly Inherited Alzheimer's Network.
So in our case of autosomal dominant Alzheimer's disease, it's a very rare disease. It only affects maybe 10 to 50,000 people worldwide, but it represents a much larger population of people who are at risk for Alzheimer's disease. Starting in the early 1990s, mutations were discovered which caused the disease, and individual centers began to study these very rare families for a few decades until there was a movement in the field to bring together the various centers which specialized in having 10, 20, 30 people at their center to form a worldwide network in order to enable the ability to understand the disease and to set up actually for future trials. And it turned out this was instrumental in allowing the field to move forward in thinking about how to offer specific treatment trials to these family members that were largely excluded from Alzheimer's trials, but had informed the field so much into the biological causes of the disease. And in 2008, that was established by the National Institutes of Health with about 14 centers worldwide. And that grew to now include more than 500 participants of these rare families who are across this entire network. And that really served then as the foundation to be able to build a trials platform to test multiple drugs in this rare population. And I really greatly appreciate the discussion on this morning's panel about what's needed and how can one implement these kinds of trials in a very rare population. One of the things that's needed is to have this kind of observational data, especially if you want to borrow from it. And so you can leverage existing scientific or observational studies and registries and try to bring those groups together to make a more comprehensive network that both increases the power and enables a disease area to really negotiate things like data access. So all trial data, for example, from the DIAN-TU trials will have public data access to researchers and will be useful for all future trials to come. Scott Berry will cover more of the trials this afternoon, but I just wanted to highlight what I think are some of the key factors of using a registry, of using ongoing observational research and what's needed. In addition to being able to establish something like a trial platform and use Bayesian design, multiple drugs, common placebos, you can begin to think about how to borrow the observational data from your prior and concurrently ongoing observational studies. And in this case, there was a natural progression from the observational DIAN study to the DIAN-TU trials. But now we're circling back around. We had established in 2012 an expanded registry where we've enrolled more than 3,600 total people that we know of around the world. And these studies are now reconverging into a common protocol to do exactly what this panel and what the prior speakers have discussed, which is set it up in a way that your data is directly translatable back to your trials. A very important part of that was the engagement of the patient population. And so by establishing the expanded registry and having that observational study, these family members, this rare disease and autosomal dominant Alzheimer's disease, they really weighed in heavily on the design of the trial. And that's critical for good trial design, and it's critical for engagement of people who, I think as Gigi put it very well, people who are expert at the disease and who know what it is that they're looking for in a treatment so they can help guide that trial design. A third thing that I think is going to be very important, I was delighted to hear discussion about it this morning, is to be able to use the biological understanding of the disease and deciding whether the drug is actually doing what we hope or it's what it's designed to do. And so in our observational studies, as in our trials, we collect an enormous amount of biological biomarker data, and that is growing, and it's going to be massive, so that if these drugs do change the biological properties and the progression of Alzheimer's, we'll be able to track that in a very empiric kind of way and hopefully use that in understanding what effects these have on what is the gold standard, the clinical and the cognitive outcome. I mentioned the use for the potential for external data. This goes beyond the observational study, and you can reach in, as the speakers here talked about, into the expanded registry, even into medical records, into daily use. We now have smartphone apps where we're testing people's cognition, and there are other ways to integrate with daily life to try to capture more information so that can be used, and that's much easier implemented across things like these large registries. And it can be done well outside of your clinical trials that you're running. The fifth point is that engaging stakeholders beyond the families, beyond the patients with the disease, is a great benefit of having these observational studies. So very early on, we found fantastic support from multiple stakeholders, including the FDA. And I want to call out Lissa and Billy and others that really led the way for us to think about how could we design the most powerful and meaningful trial designs. And I see that, as the FDA has led these efforts, it is likely to cause a change, hopefully not just for orphan diseases, but for all diseases, and that there are more powerful ways we can answer questions, whether drugs are useful or beneficial or not. And finally, the data that you collect in the observational studies and registries can actually inform your statistical plan, not just borrowing the data, but actually tell you what should be your statistical plan. What should you be looking at? What is important? How does it change over time, and how can you implement it? And Scott Berry will cover this in much more detail in the next session, but a disease progression model was built to be able to look at Alzheimer's over the course of 20 years pre-symptomatic disease through 10 years of symptoms, and to be able to use that model to greatly increase the power of the ability to find drugs that change the course of the disease. And that observational data is really essential, can't be done without it. And so I think starting there is a good place to start. Those are all my points.
Great. Thank you very much, Randall. Very impressive work that the network has accomplished here. Next is Yifeng.
I agree with all have been said. I want to add 4 points. The first point I want to add is sometimes I am hoping the natural history can help the already completed clinical trial to provide trial information. For example, into some, like a drug has been approved in the market. For the second drug, maybe we are going to use a non-inferiority trial. We need to determine the margin. So it's just based on the one previous trial, that will be not sufficient for us to determine the margin. So maybe natural history data can help us on that. And the second point I'd like to make is maybe I'm hoping to have a complete like a database for the natural history, because sometimes we will use that database to identify some kind of the right population for the study, for the biomarker endpoints, even though the sponsor may conduct a new trial, but because the patient population may be different, sometimes the measurement can be a little bit different as well. So if we can have some kind of more flexible and complete database, and that will help us to, collect the data for the right patient population, the right endpoint. Then that, most likely the biomarker endpoint can be used for as a surrogate for the accelerated approval. So that can be helpful if we have a very complete and flexible database. The third point I would like to make is regional effect can be very important. To U.S., we can have maybe we will have a different endpoint as a Europe. However, we will consider maybe there's a regional effect, that can into the play. The fourth point I want to make is, even though when we review the trials or FDA, when we give a recommendation, we can be flexible, but I want to emphasize the importance of following those basic statistical principles. Sometimes the sponsor just argue, saying, "Oh, we see the signal," but everything is a post hoc. That can be hypothesis generating. So we still need to follow the basic statistical principle. I think that's very important too. Yes.
Great. Thank you very much, Yifeng. And next is Patrick Helfman from Avexis.
Thank you. So I apologize. I have a cold, so I hope my voice will hold. I just want to add maybe or underscore maybe a couple of points here. One is the importance for rare diseases to understand that every data point has to count. We have now therapy development for the more common rare diseases, but there is thousands of rare diseases that equally deserve a treatment and where there is great need for better therapies. In order to get to these very rare diseases, we need to make sure that we have more shared platforms, that we use more standards. So it just cannot be that when we have only a few individuals affected or a few families to provide information, that we have clinicians in one university collect data, in another university, in another country, then patients collect data, maybe mobile devices collect data, and all of these different sources of data points are not connected. So it's upon all of us to think how we can make sure that these are connected. That's challenging, but it's so important. And I think platforms and standards, as Patul alluded to, are clearly an important basis in order to facilitate that downstream connection of different data sets so that we don't have all these islands of good information, but every time we need the information, we discover that it doesn't speak to each other. And also, as Petula said, I think patients can drive that very much because they are the ultimate source and, if you wish, the owners of these data. So I left the NIH recently, but while at the NIH I had the great opportunity to work on a couple of things in that direction. One is common data elements. In my case, I worked on common data elements for neurological diseases, but there's a whole portal. And, there's also, of course, the CDISC standards that you mentioned. But I think one thing is to encourage people to look at existing data standards rather than reinventing the wheel, because that sets the stage. Doesn't make it all easy, but at least makes it potentially easier to connect data sets. And the other thing is that I know I used to be, until 3 weeks ago, at the Office of Rare Diseases Research at the NIH, and my colleagues, Eric Sitt and Alice Chenna over there, and they're working on a platform. They're hoping to help patient organizations get better information on what it might take to get to these data centers. Of course, registries can be very costly. It's like buying a car. So, some people, of course, everybody would like to have a Rolls-Royce, a registry that does it all and has the perfect data quality and standards. But many disease areas, many organizations can't afford that. So what does the minimum sort of standard, practical, economy car look like that would still drive me to where I need to go? So that's just one point I wanted to make. So think about platforms and standards and don't reinvent the wheel. And then the other point I wanted to make is, from my recent experience, I now work for a gene therapy company, and I'm very gratified to see that some of the data that are used in these phase 1 studies came from work I had done when I was in academia in a natural history study. So they were actually able to use the natural history study that I had done with colleagues when I was at Columbia, at Harvard, and UPenn. And the reason I think why they were able to use the data is that we, at the time, really struggled a lot. So we started by saying, okay, we are academics and we want to gain knowledge on the disease. And we had these discussions on what would be great to collect and what would be interesting. And then we were funded by a patient organization, and they said to us, it's not about what's interesting and what you think would be great. It's about what will really ultimately downstream be useful for evaluating therapies and what would be feasible. And we had a lot of debates, but we ended up designing a study that had two characteristics. One was that it was not just all the things that were great to collect, but we really try to hone in on a few things that would be really useful in potentially a treatment evaluation context. And then the other thing was that we treated it like a clinical trial. So we had a protocol, we monitored the data, and we very much worked very hard on data completeness. So it's not the quantity of variables, but it's really the quality of what you collect and the completeness, I think, that makes a data set really good. So I guess that's a signal to stop. So I'll stop here, but I think that, in terms of, natural history, we can look at case studies. So what data were actually used in a regulatory context? And I think it's the data that are of the kind of quality that you need and completeness.
Great. Thank you very much, Petra. And next is PK Tandon from Ultragenyx.
Yeah, thank you very much for inviting me. It's a pleasure to be part of this panel. So I think I agree with a lot of the comments made, but I've been very fortunate to be part of a number of registries in rare diseases and natural histories. So I just want to share some experience and some lessons learned in my career. And I think one of the lessons was somebody asked, when do you want to start natural history registry? Answer is as soon as possible, So I think the company sponsor should be thinking about maybe pre-IND, IND stages, rather than not wait until phase 1 is starting. Oh my God, we don't have a control group here. So I think should be thinking about early on. And another thing is, for registries, I think what I have found is that, as Petra mentioned, they are very expensive. So one way to do it is do what I call a survey, just a cross-sectional observation study on all rare disease patients, and basically collect as much data as possible and look at the data and see what is important for the patient and for the caregivers and so on. And based on that, design your registry or long-term follow. You can really save a lot of money doing that, and you can learn it quite a bit from having the survey cross-sectional study early on. And another thing is, third thing, I'm a little bit surprised that we as an industry don't use technology as much. And I think especially for registry and for the natural histories, I think online data collection could be extremely helpful, because remember, most of the registry have been involved. They're voluntary, so patients coming to the site, and you may be missing very important information, especially patient daily living and so on. So nowadays, you have an iPhone or iPad, and those kind of things could be easily be used in designing very simple way of collecting the data in a very robust, reasonably robust way in terms of using that data. So I found very useful technology. I think we should be thinking more about using technology, collecting the data. And these are data coming directly from the patient to or caregiver to directly a computer. So you don't have to worry about a lot of cleaning and so on. So I think you can gain a lot of experience with that thing. Another thing is, I think I already mentioned, I found registries or natural history, I think we use sometimes interchangeably, is a endpoint selection. And really, I can give you a very good example, is a Niemann-Pick B, which I was involved in. We found that spleen volume in that patient population, very sensitive endpoint in terms of looking at changing. And very useful, you can select the patient population based on spleen volume, and that could be a very good biomarker, surrogate marker in terms of showing a clinical benefit with your therapy. That's another example. And identifying, patient population as mentioned, I think is really key. So in a nutshell, I think if these natural histories or registry designed appropriately can be very, very helpful, and especially in rare diseases. I think I'm going to stop here. Thank you.
Great. PK, thank you very much for the comments. Thank all of you for an excellent presentation and discussion of what I took away is mainly many of the strong advantages of having a registry, or at least a well-curated natural history data collection process you all mentioned, and PK was just talking about how this can help save a lot of money in understanding whether or not treatments in rare diseases are effective, and designing trials, as well as providing useful information to patients. So maybe I'd like to start by asking, why don't we see more of these, or are we getting there? I think several of you alluded to the fact, I think Petra and others, that this can be costly to do, especially to do well, and people should at least aim for the economy car version. But what do you see as barriers to why there aren't resources like this in more disease areas? And if Nicole, if you or Randall want to talk about, I expect you all get asked by other groups, how could we do something differently? And would appreciate any of your thoughts about what the barriers are and what might be done to address them.
Yeah, I think that the biggest barrier is cost, and I think this is where forming those industry partnerships can really be a huge advantage towards building a common infrastructure. And especially, I think, based on today's session, if there's motivation that this could really be of help in the regulatory space, I think that will drive momentum, even perhaps before there's a product. But if an industry is actually in an indication, they will be willing to invest early on. And so we need to strategize in how we can build those partnerships, early on, to invest in a common infrastructure that they would have access to do their trial planning, and then use them for creative designs to approve their therapies.
So my experience has mostly been with neurological diseases, including the less common neurological diseases and other areas where people have tried to pull together global registries or even national registries. And I think the challenges are several-fold. So one is that it's just not been the way that rare diseases have typically been studied over the past 50 to 100 years. Most of the time it's an individual center that's following their own people, and there really hasn't, until recently, the past 10, 20 years, been a lot of networking done across centers. And that's partly an academic thing, where you're at a medical center, and to really get a national or global network is a more recent approach. I think with that, the cost and the funding is a major issue. In our case, the National Institutes of Health was transformational in that, because they saw the wisdom of putting out a very specific RFA to create this network. And that really prompted academic centers that normally competed against each other to come together and form a network and work together. And I think once the academics saw the value in that, then it was clear where we could go with the trials. And then the pharma partners came, which was hugely helpful in getting the structure and the design set up to prepare for clinical trials. And as part of that, of course, the other stakeholder is the public stakeholder, the government regulatory bodies, which in our case is global. It's more than the FDA. And having those stakeholders take an interest in the disease, and things like the pilot program that I'm hearing about, and our prior experience in this meeting, I think these things will also be transformational into stimulating various groups to come together and say, this is a path forward. We can approach a rare disease this way by getting an observational study.
What I wanted to add to that is actually, I find that education becomes important, and the lack thereof becomes a big barrier. And what I mean by that is not only industry education about how the FDA works or how we think, but the patient advocacy groups and patient and caregiver education of how do we think at FDA, or how do we make our decisions, and what kind of data are we looking for, and for what reason. And we do try to do that, and I think there's large efforts to do that, but I think that becomes a great barrier. And, example that I have heard in terms of a patient advocacy group really actually not wanting to participate in a natural history study, saying, we know how the disease progresses, why do we need study to show us that? But I think there's some legitimacy to this, and I think that goes back into we need to listen to the patients and the caregivers and really use them and leverage this information to get the data in a more standard or objective way as much as possible. But also it has to do with education of the patient groups and the patients and caregivers themselves, or, what is really involved in drug development, how do we think about it from a practical perspective, and what does it take for us to be convinced that a drug works, for example, and a benefit outweighs its risks for regulatory approval. And so this piece, the educational piece, I think becomes very, very important in that realm.
Thank you. Other comments? PK, you look like you were going to...
Yeah, I think we know they're expensive, but in long term, to me, they're not, to be honest with you. And I think one way to solve this problem could be investigator grants given to the investigators. And I know, for example, Pompe disease in Amsterdam Medical Center, they have their one of the best database available in natural history, and they got a lot of grants from European Union, and we were able to leverage with that database. So I think one way to solve is giving the grants to investigators, and they're collecting the data, and that could be part of consortium or something like that. As a industry, I think I'm right now in industry. So for me, it's a competitive advantage, to be honest with you, to doing this kind of, this is the initial investment needed. You have to invest if you really want to develop a drug and really understand what is important for the patient and selection of the endpoint and subgroups and so on. And I think it really helps in interacting with FDA, especially the example Chauvin Myozyme. It was a very cooperative, interaction with FDA, showing the natural history, how we're going to use a control group. So in the long term, it pays off.
You all have talked about a number of barriers and some very positive and constructive comments on how to overcome them. I just want to follow up on a couple that have been mentioned, or maybe one that's been mentioned and one that seems related. And that gets back to some of the proprietary concerns. So historically, talked about separate studies driven maybe in part by individual academic centers wanting their own publications and wanting their own grants. And PK, your point about maybe having a consortium approach to grants could help address that. But I'm wondering if there are any other thoughts that you all have about overcoming the barriers from not getting recognition that academic groups might have by participating and really getting behind these efforts. And then similarly on the company side, I take your point, PK, about competitive advantage for, say, your company to be involved with a natural history study, but does that same spirit apply to, say, other companies that might want to benefit from some of the same data that might otherwise be regarded as proprietary? So any comments about, further comments about alignment of both academic participants and industry with getting behind these common approaches?
Well, I can just say what I said. It's patients have to be the ones driving that. I think they have to, talk to academics and industry, and I think it's in everybody's interest. We have now really unprecedented opportunities to translate progress that we've made in medicine and biology and research into treatments. So if we consider things like information on how a disease progresses as competitive information, I think we will, as a society or as mankind, not be able to ensure that all people in need will get to these potentially transformative treatments within reasonable timeframes. So I think it's upon all of us, and I think patients can really make this point best, to see that we can have the competitive part of this more around the intervention or the product, if you wish, but the non-competitive part being sharing of data that lay the foundation and set the stage for being able to evaluate different interventions.
Randall and PK.
Okay, so I just add to that the same thing that makes orphan diseases really difficult to study can, in fact, make it quite powerful for creating shared networks and shared data, and that's the rarity of the disease. And so the population being rare does not afford the opportunity for any single group, company, drug program, the real ability to go in and do all this. Nobody's going to have a 10 to 20-year plan of how to create a global network of a rare disease, study it, and for a drug program to get through a clinical trial, that's not going to happen. But the rarity of the patient population, with the researchers, the academic folks that work on it, if those people, and as I said, in our case, that was stimulated by the NIH having an RFA to form a network, if those people come together and create a network, now you have a natural foundation and basis for sticking together, right? Otherwise, if everybody goes it alone, it's really difficult, and the odds of success are really small. But once you have that foundation built and you're generating the data and you're showing that people come in by the hundreds year after year and are willing to do the spinal taps and the PET scans and the MRIs and the cognitive test, now people who are in the business of developing drugs to try to change the course of that disease have an easier entry point, and then they can come together. We form, for example, there was a Diane-2 Pharma Consortium. The pharmaceutical companies agreed to work together pre-competitively before their individual drugs are entered in the trial. And they added resources because they saw the value. So similar to Petra, I'd say it does start with the patients, but it really needs to have the help and the stimulation of patient advocacy groups. And the academic researchers need to come together. The NIH has a key role to play, and I think the FDA and others can stimulate this by saying this is a path forward.
PK?
Yeah, so I think Cystic Fibrosis Foundation has done an excellent job, and I think they have more numbers on their side, about 30,000, 40,000 patients. But I think the challenge is for diseases when you have less than 1,000 patients in the United States. Who's going to do that? I think the onus is on the sponsor then to take the lead working with the KOLs in terms of building the database, because I think it costs money. And I think that is, that's why you're investing it. And you really don't know when you're starting that, your drug development, you don't know if it's going to work or not. So you want to take risk, and I think that's taking risk for early, very rare diseases. In that sense, I think maybe. I think one thing I've found is a lot of KOLs, and they publish papers. A lot of, say, for example, Niemann-Pick has a very good registry done by Genzyme. A lot of papers have been published on that thing, so you can get some really good idea what's going on there in that registry, or in terms of endpoints and so on. But I think we need to think about very ultra-rare diseases, how we're going to handle it. I think a sponsor has to take a lead working with patient groups and the KOLs.
Right, thank you. We do have a little bit of time for comments and questions. So once again, in the room, if you could go to the microphone, and if you're joining us online, it's margolis.rare@duke.edu. And we'll try to get through as many of these as you can. Once again, if you could introduce yourself briefly when you make your brief question or comment.
Sure. Monur Saleh, statistician, Audentis Therapeutics, San Francisco. So we are so much not mainstream statisticians here discussing rare diseases, and amongst you, there are even more rare statisticians like myself dealing with ultra-rare diseases, where some of the discussions about subgroups or regional differences may not apply so readily. Like, for example, if you have an international study that you have one patient in UK, one patient in Israel, one patient in Italy, it's very hard for me to establish criteria to demonstrate regional differences. So those are one of the real issues with really ultra-rare diseases. In ultra-rare disease, I mean, some of these conversations, of course, I'm very happy to see, but you are more advanced because you are in majority of us perhaps are thinking about the kind of rare diseases that there are a lot of patients still available for them. Whereas some of the ultra-rare diseases are challenged with even characterizing the disease itself. What does the disease constitute? What is a clinically relevant endpoint and things of that nature? So I'm hoping that some of the untouched areas of clinical need and unmet need is in that area. And by the way, the treatment is extremely expensive. Some methodology like AAV technology and things like that, $500,000 a pop.
So do any of you want to comment on this? PK, you were headed in that direction, I think, with your comment, but this is even smaller.
I can start. So I think this problem you describe is not uncommon, and I think the way to do these kind of things is you have to collect within patient data, a lot of within patient data. You have to follow these patients, whether they're coming from UK, from Israel, from anywhere. And it takes time. And maybe you have to go to case by case. You have to go really retrospective chart reviews for these patients to really understand what's going on there. And for endpoint, I think that's a really difficult issue. But I think the way to do this kind of thing is a really understanding within patient kind of things. And there's methods that are available where you can look at it, crossover trials where you can have first patient is treated with therapy, then off, then treated again. You can look at within patient N of 1 trials are possible doing those kind of rare disease trials. So I think methods are there, but I think it requires efforts in the sense collecting the data over time.
Thank you.
Hi, Cyrus Mehta from Cytel Corporation. We've been accustomed to the gold standard, which is randomized clinical trials, and now we're moving away from that gold standard into determining therapeutic efficacy through the use of registries. I wonder if the panel could tell us something about the pitfalls that one might face when using registry data as opposed to randomized clinical trials to evaluate therapeutic efficacy. What are the main difficulties, particularly statistical difficulties, that one might encounter?
Thank you for this comment, and it's actually a good, maybe a clarification would be needed. So I don't think we're making a point that we're shying away from randomized clinical trials. There's still a standard for that you need to prove substantial evidence of efficacy. The standard is no different in rare diseases. And so in that sense, randomized clinical trials to some degree are needed to demonstrate that. I think it depends on the disease specifically, and it is a lot of case-by-case basis. We do interact with sponsors where they try to leverage retrospective data or natural history data and generate, quote, a clinical trial post hoc in a sense. And we oftentimes this is very challenging because of the issues that have been raised in terms of bias in collecting or others. And so I think the clarification I wanted to make, and Yifan can probably speak to that too, is that the bar for showing the substantial evidence is really no different. We still need to see data and evidence that a drug actually works and that its benefits outweigh the risks. Now, what has been already talked about in terms of natural history data and registries can inform those trials, and in some instances maybe serve as a control group in very specific situations only.
Yeah, I want to emphasize, to some cases, the randomized control trial is not possible. We use a historical control. However, in that case, we need to make sure patients are indeed matched, like similarly with the comparability, including those, endpoints and the patient baseline characteristic and so on. And they can do some kind of not one-to-one randomization, maybe like a 2-to-1. We have a 2 in the study, two patients in the study and one patient from the natural history or historical control. We try our best to ensure the comparability. Yeah, to ensure we have a substantial evidence.
Just one brief point about that. You don't have to select one or the other. So you can do random. You can do just as Yifan was suggesting, differential random. So we have a 3-to-1 randomization and a true double-blind randomized placebo-controlled trial. But you can also use data from other data sets potentially, if it's appropriately controlled.
And yeah. Yeah.
Yeah. I think it speaks to a lot of, I think I had that one slide. I tried to cover it in one slide, given 15 minutes to cover a lot of topics today, but
did a nice job.
There are many sources of bias. I think confounding by indication is probably one of the biggest sources of bias. And as pointed out, there are methods such as propensity scores or instrumental variables that you can use to account for that. But they may not work perfectly, and so you have to be mindful of that.
And I think we will keep coming back to these issues. They're tough ones. So we're almost out of time. I'm going to try to get in one or two more. Do you want to go ahead?
Okay. I'm Estelle Saccone, and I'm hiding in my third center at FDA. So one of the things I think natural history studies can be used for are things like maybe repeatability of the endpoints. How do you match people if you don't have natural history to say who is going to have a like prognosis? And I think natural history databases could inform a better design and a better basis for matching. So that's one. Two, my other comment from today is I don't think pediatrics and adults are quite the same. And we tend to think about an endpoint in children. Children are heterogeneous just on the basis of their age. And natural history studies could inform how the disease progresses and understanding the heterogeneity with respect to age. I'm reminded of a paper that I read in muscular dystrophy where they looked at a 6-minute walk. The kids walk more and then walk less because as they're growing physiologically, they're getting stronger, but then the disease catches up with them. How you pick an endpoint in that space becomes really, really important. And so I think natural history studies, to me, if you don't have them, you're in a sense shooting in the dark in many cases, and I think that needs to be done. Finally, the only other thing I'll say is in ALS, they've done a nice job of taking all their failed trials and put them in a public database. Then they've done a lot of neat analyses, not only to show that the placebo arm can be really quite different across studies, which says you really have to understand the heterogeneity in your patients. They've also done things like retention and recruitment rates for each of these studies, which are critical in planning studies, because if the patients are gone, what are you going to assess? So that's my comment.
Thank you. Great comments. And I think, Alyssa, you may get the last word here. We are going to have some time this afternoon for public comment, too. But please go ahead.
Now, well, this was on a different topic, although I do. I'm happy Randall brought up that it may not be either or, you can do both, and we'll hear about that this afternoon. But this was a different question. So drawing on Nicole and Randall and PK and others that were talking about the benefits of collaboration and getting multiple sponsors to work together, plus the NIH and so forth. In a rare disease setting, in a sense, there's competition for the patients, I think. And if one company is working on a drug and they've got sort of a hold on patients in their trial, that might impair the recruitment of another company which may have a drug that has more promise. And so I wonder if you could speak to what the collaboration might do to help resolve the competition a little bit.
And hopefully do it in a way that won't discourage companies from supporting the effort. Yeah.
I can take a first stab at that. So, I mean, we're experiencing this a lot in cystic fibrosis, and I think, we run a consulting program such that we work with our industry sponsors, and I think the first and foremost, we set the tone that we are there to help them be successful, and that means that there's a little bit of trust there. So we work with them to, not only look at their study design, but their eligibility criteria. We toggle and use the registry to see, how can we enrich your patient population? And because we are the ones that are looking across the entire landscape of therapies being developed, and we've got to find a slot for all of these studies being performed. And So there is a lot of trust there, but we've had great success and with them accepting our advice to sort of optimize our sample size and focus on need to know versus nice to know, I think is the common theme for rare diseases that has been our motto for success.
Great.
So I'd just add to what Nicole just said, which is that it's also important that the registry, that the study really be an open study, that it be supported with public funds or academic, and that the rules are clear and transparent, set up front, that the goal here is to come up with the best treatments for this disease. It's not necessarily for your individual drug program. And so we want your individual drug program to be successful. We will do everything we can for it to be successful. But we keep the goals in mind, and everybody agrees to that coming in. And that's what I was trying to indicate, that that's the advantage of having a public and an academic registry, a cohort, to where you set the ground rules up front. Data will be shared. We will be using the pooled placebo data. We will be, doing these different things. And I think if that's clear from the get-go, it puts at bay sometimes what are individual competitive company issues and even academic issues, competitive academic issues, on what the real mission is. You keep the mission on the patient's disease and trying to treat that, and that's been very helpful for us.
I'd like to thank the panel for a great discussion, a very important topic. Thank you all very much. We are now going to break for lunch, so we're going to reconvene here at 1:15. Lunch is on your own. There are lots of restaurants close by. If you need a list, you can get one outside. And you can also feel free to bring food back in here for the 1:15 session. But we are going to try to get started promptly, given how much we have to cover. Thank you all very much.
And I'll go ahead and ask our speaker and panelists to go ahead and join me up on the stage for this next session. Great, thank you. So welcome back, everyone. I'm Greg Daniel. I'm Deputy Director in the Duke Margolis Center for Health Policy. In this next third session, we're going to explore how master protocols, which has been brought up earlier this morning, how those can be leveraged for trials with small patient populations. Our presentation for this session will be from Scott Berry, who's President and Senior Statistical Scientist at Berry Consultants. Following Scott's remarks, we'll hear from our lead reactant, Michael Prochen, and other panelists. Michael is a mathematical statistician, biostatistics research branch at the National Institute of Allergy and Infectious Disease. Following Michael, our reactant, Billy Dunn, who's director of the Office of Drug Evaluation 1 in the Division of Neurology Products at Center for Drug Evaluation and Research at FDA. And then finally, Raji Sridhara is Division Director and Division of Biometrics 5, Office of Biostatistics at CDER at the FDA. And with that, I'll turn things over to Scott.
All right, thank you. I have to move the microphone down a little bit. You have to move it back up for Michael in this. All right, so I'm going to talk about master protocols, and I'm hoping that these are seen as a solution to many of the things that were discussed earlier. My goal in this is that you not look at this and say, oh, we've been doing this, cooperative groups, for years. This is something really quite different than that. So I'm going to try to get that message across. Master protocols. Part of the problem is the name master protocol has been used many times. Platform trials have been used many times. This is something that's really quite different but doesn't have a name that differentiates. I hope master protocol becomes that. We're used to traditional trials where we ask the single question, does this treatment work in this disease? We treat the disease as homogeneous and we ask the one question. And once that's over, the data's gone. Maybe meta-analyses come out, but it's a single question on a single population. Basket trials are now a personalized medicine. We recognize that diseases are heterogeneous. And we're investigating a particular drug in a heterogeneous set of the disease. And it could be phenotypical or genotypical characteristics of the disease, but we recognize it might have differential benefit in the groups. So we run basket trials. Sometimes this is referred to as a master protocol. I'll refer to it as a basket trial. So what do I mean by a master protocol or a platform trial? Let me first talk about a platform trial as one where let's think about a homogeneous disease, and we're not going to investigate a single drug. We're going to investigate multiple drugs in that disease within the same protocol. And this could be perpetual. We could start off with 3 drugs. We could make a 4th, add a 5th, add a 6th. There's no natural end to this. As long as there's a need to find things that help, the protocol can run and investigate that. You can, of course, cross this and investigate multiple drugs in a platform setting within diseases that are very, very heterogeneous. And so you could have a platform basket trial within the setting. So what do I mean by this being different than the way we've done things before? First of all, there's a single protocol. And what I want to differentiate this from is where there is a cooperative group that runs trials in a disease, and a pharmaceutical company may give them a protocol, say, can you enroll my protocol? And they enroll that protocol, and somebody might come in and, okay, you go into protocol A or protocol B. There's a single protocol here. The master protocol assigns the patient. They come in, they're eligible for the master protocol, the protocol assigns them. It might be randomizing across arms, it might be response adaptive randomization. They don't say, I'd like treatment 2. I want to go into that protocol. The master protocol assigns them, which allows you now to create a great deal of statistical synergy in comparisons, and everything is randomized, assuming the master protocol randomizes patients. One of the beautiful things about this, and Dr. Bateman talked about this, is it doesn't say the goal of this protocol is decide whether treatment A is safe and effective. It's how to best treat the disease. The protocol exists to find the best way to treat the disease. They're disease-focused protocols. We bring in multiple therapies trying to figure out what works and in whom, but it's disease-focused. What's the term master protocol? The master protocol may exist and say what we do to patients, what are the inclusion, exclusion criteria, but there's not a drug name in the protocol. The drug names come in as modular appendices within that, and they can come and go during the course of the trial. So when a treatment leaves, the protocol doesn't actually change. The appendices change. And it's modular and it keeps going, where what you do to patients and who gets in and how we treat them, visit schedules, endpoints remain the same during the course of this. So each one of these are modular, and again, it can evolve. So the trial can start off with 3 treatments, and after a while, treatment 1 drops for futility. Or if it's a phase 2 platform, maybe it graduates to phase 3, and it leaves this particular master protocol. And then after a while, treatment 4 comes in, and we add a modular appendix. And by the way, you can have first patient into a particular appendix in a few months, as opposed to years of what it takes to develop a protocol now. Now, this was a paper that came out last July. Wonderful paper talking about master protocols. It talks about basket trials as well. And I want to bring a quote from this. And this is what I hope to differentiate from what you may think about collaborative groups, where there are 2 really huge innovations to these master protocols. One is the use of a trial network that has the infrastructure in place to streamline the trial, improve data quality, facilitate data collection and sharing. So that's typical of what we've seen of collaborative groups, and they're wonderful, and that does bring it out. The second part to this, though, is the use of a common protocol that incorporates innovative statistical approaches to study design and data analysis, enabling a broader set of objectives to be met more effectively than would be possible in independent trials. Statistical synergy is created by everybody playing in the same protocol. It comes out quite simply in power, strength of the trial. There are many other aspects of this that come out of it as well. So let me introduce you to a couple of these. One exists and has enrolled patients. The second one is being developed. Dr. Bateman talked about this. This is the DIAN-2 trial. This is in a rare disease, genetically positive subjects, and these are people who have a gene that means they're going to get Alzheimer's before the age of 60. Very rare. It's autosomal dominant, so a 50% chance it's passed on to the offspring. So somebody can be mutation positive, and they know with certainty they will get dementia, they will get Alzheimer's at an age that's incredibly predictable for their mutation status and their parents' mutation status. So somebody at 30 years old could be told you have a gene positive and you will get Alzheimer's at approximately 41 years old in this setting. So now they're enrolled into the trial, and this is set up as a master protocol. The subjects in this trial, there are multiple drugs in the exact same protocol. So what are some characteristics of the trial design? The trial it's a master protocol. It's a common platform. There are 2 drugs that have enrolled in the trial, and there may be 3rd and 4th, I won't say much more than that, that can be added in the trial. It could perpetually go on as long as we have a need within the setting. Every patient that comes in is assigned to therapies by the master protocol, randomized to all the arms that are currently enrolling. 3-to-1 active to placebo. All of the placebos are now combined together to form a common control group. So if you were to individually run these 2 separate trials, you have inferences that can't be done across those 2 trials. But in the same setting, you have randomized active arms and you have randomized controls taking place at the same time. I'll say one comment is Randy talked about the natural history study here, which was incredibly valuable in creating the analysis of this trial, which, by the way, is fully Bayesian. fully posterior probability that a drug is beneficial, and the endpoints and the model is based on the natural history study. So here's the particular setting that we enroll 3-to-1 in 2 different drugs. A third drug is added and it enrolls 3-to-1. It contributes placebos, common controls. A fourth could be added. The really neat thing about this is when a third drug is added and it starts to enroll controls, it makes things stronger. We have more power for the third drug than we had the first 2. In the normal setting where we run siloed trials, the third drug that investigates a particular disease is the same as before. We haven't learned anything. We haven't changed anything. We haven't borrowed that information in ways, and this is now the exact same protocol. Inclusion, exclusion are the same. These are all randomized patients in this learning system. This is a natural history study that's randomized and perpetual with the same things going on. It brings together those concepts. I'll share one aspect of this. This shows you the statistical power. If you were to enroll 80 patients 3-to-1 and do an MMRM analysis, which we typically do in Alzheimer's at 4 years, and there's a 30% effect, you have 16% power if in fact the drug slows the disease by 30%. With the model that came out of it, which, as Dr. Lewis talked about, we have 2 random effects per patient that came out of the model. That Bayesian analysis bumps that to 63% with the same patients. By borrowing controls from the other arm, that jumps to 90%. The natural history study and the platform trial combined take 16% of what we would have done 10 years ago and makes it 90%. And then the third or fourth drug that comes in will be even higher because of the data that's already built up. It's more than increase in power, too. There's a disease understanding that comes out of this. I-SPY 2 has had 14 different drugs go through it. It's a master protocol, randomized trial. And now the question comes up about surrogate markers. And there are 14 drugs of differing benefit on a surrogate marker and then different outcomes on the event-free survival. This is something you can't do across other trials. They're trying to do this, and there's this incredible engine to understand disease progression within breast cancer that can't be done if you ran 14 separate trials. And now it's the same quality, the same standards, and 14 drugs of differing benefits on each endpoint to investigate this. So the disease understanding that comes from these is much larger, so it's not just power. Abby Bronson, I think she's coming up in the next section, is leading the Parent Project Muscular Dystrophy in the creation of a master protocol for Duchenne. And this is a beautiful application of this in rare disease where it's creating a master protocol, a phase 2-3 platform trial design. We don't know what drugs are going to be in it, but we're building this master protocol to have sponsors bring their drugs into the exact same experimental conditions. Work together, enroll common controls that everybody can compare to in a randomized trial. We'll be able to enroll a much wider disease severity. Rather than taking these 4- and 5-year-old boys only, we can take the entirety severity of the disease. A boy could actually come into this trial, be randomized, be re-randomized afterwards if that drug didn't work. If they were on control, they can be re-randomized. They can spend their life in this platform trial with different experimental therapies. So we're in the building stage of this master protocol. But I think this is something that could be done in a very wide range of rare diseases, every rare disease. So I was asked to talk about the challenges. This is a new kind of trial. It has different challenges than building a usual trial. It takes more work to build a master protocol than a single drug in a single disease. We can bang those out and get them rolling reasonably quickly. Now you have to do the upfront work. Who pays for that? Who's a part of building this? And it takes longer. You're not building it for the drug that's sitting there right now. We're building it for drugs in 6 to 12 months, which we know there's going to be a need in both of these situations. We want to build an integrated inferential system. So every drug that comes in adds to the statistical strength, adds to the information in the disease. We need to do that recognizing that this has to be something that sponsors are going to want to bring a drug into. If we build this amazingly inferential thing, but I love response adaptive randomization, but a sponsor may say, okay, you might accelerate somebody else's drug faster than mine. So we might not do that in the master protocol. Every drug gets its particular shot. It has to be desirable that sponsors are going to want to come in. And in most of these cases, we're going to do it cheaper, faster, and stronger within the master protocol. If everybody works together, it's cheaper, faster, stronger. But you have to build that and make it desirable for the patient organization building it, the inferential aspects as well as the desirability for somebody to bring their drug into it. Doesn't work if nobody brings a drug into it. Writing a master protocol that doesn't have a drug name in it is a different beast with modular aspects to the drug part of it. Writing a master SAP and appendices for an SAP. This is a new sort of thing to be able to build. I'm writing a protocol for a drug that's going to enter in 3 years, and I don't even know what it is yet. Who's going to pay for that upfront part? Does a sponsor pay to come in? Do they pay per patient? Who funds this? These are all things that, now there are many of these that are being developed, some in rare disease, some is not. So this is being tackled differently. Andrew Lowe at MIT is working on different economic structures for how these work in the settings. And maybe even the master protocol has some sense of outcome if a drug's effective out of it. How do you select arms to come in? How do you recruit arms to come in? Who makes that decision? What is that process? It's a new process. Typically, you have a drug and you build a trial. Now you have a trial. What do you allow in as a drug? Do you diversify your therapies as a disease-centered trial and not let 4 in that have the same mechanism of action, for example? Complex simulation. These trials are more complicated. The characteristics of the synergy and the borrowing across trials, almost all built by simulation. Do we do response adaptive randomization? Do we have subgroup effects? Do we have enrichment that goes into it? How do we count the first set of controls that existed before my drug came in? Time in these trials is a really important part. It's almost like side effects. We now have potential time effects. How do we model that? Typically, we don't do that in a usual trial, but they're really, really important here. Do we have global rules for futility that a drug needs to leave because it's not demonstrating effect? And will sponsors agree to that in a particular setting? Blinding is very different in these trials. For example, in the Diane trial, you're not blinded to whether you got A or B, but you're blinded as whether you got A or A's placebo. Everybody's blinded to active or treatment, but you know you're either drug A or drug B. So you're not triple blinded in the setting. So how do we do blinding? How do we do consent? Do we give investigator brochures for every drug in there? Do we do staged randomization and staged consent? iSpy has a 2-stage consent because of this. So this is different. Arm-specific exclusion. Who knows what win in a perpetual trial? The first patient in may contribute to the 4th drug's efficacy analysis. How do we publish that data? How do we share it when that patient is part of the ever-growing set of data? So who knows what win is always quite different. Creating a consort diagram for how patients were assigned to the trial is a little bit different than your usual trial. So we're creating problems there as well. So these are certainly challenges. Now, the potential is a much better understanding of the disease, better inferences per arm, certainly. More shots on goal. We can cycle more arms, more therapies through such a trial than can be done in independent trials. They're cheaper and faster. You're sharing infrastructure. Just the fact that you're sharing sites and you're sharing a database brings out large synergies. Now you're sharing controls in a randomized setting. So there are enormous advantages. And imagine in the setting of rare diseases now where we have very few patients, to have that patient continue to contribute, the more arms that come in and the more understanding of the disease. And in both of these cases, the Alzheimer's and Duchenne, a huge question is going to be, if we find effective therapies, will early surrogate markers suffice? And now we've got a single place to look at incredible data to answer those questions that we can't do elsewhere. So disease understanding is a huge part of this as well. So I will stop there.
Great, thanks, Scott. We'll turn to Michael for some reaction.
All right, thank you very much. Let's see, is mine up
Just
there?
keep.
Okay, great. So I love the hockey analogy, shots on goal. But excellent presentation by Scott, as always. So I wanted to just start with what we usually like to see if the disease is not rare. We like to have large sample sizes. That's clearly out the window. We like to have replication of trials. That's out the window as well. We like to have results that are convincing, even when you do simple kinds of analyses that don't adjust for different covariates. That probably can't hold either. We like robust results where the answer doesn't change if you do different sensitivity analyses and you look at secondary endpoints and so forth. We're not in that situation. And we usually like to, protect against multiple comparisons. We like to protect against all the statements we make in a clinical trial. So we often do a multiple comparison adjustment that may not be feasible in this setting. And then we like to have substantial data on safety. I don't think we can have substantial data on safety. So all these things are out the window. And so you need to make the most of the data that you have. And the master protocol approach, I think, does that through sharing the control arm for multiple treatment arms, eliminating poorly performing arms, and then possibly re-randomizing those patients to some remaining arms. Using a model-based approach where you make a few more assumptions, but you get more out of it. You get higher power. Conserving resources by using the same master protocol for different treatments and different diseases. And so I think these things are all good. And avoiding an overly conservative type of analysis, and the primary culprit here is Fisher's exact test in the binary outcome setting. We know that with small sample sizes, that's a very conservative analysis, and because you restrict the number of tables severely, that 2x2 tables. So all of these things, I think you have to, are beneficial things. I do have some concerns and some suggested safeguards. I do think it's important to protect against temporal trends. So I personally would want to compare arms to concurrent controls. If you have started out with 2 treatments and then a significant amount of time has passed and you're bringing in another treatment, I would want to use only the controls that are contemporaneous. And I personally would avoid response adaptive randomization. Because that can result in long strings of the same treatment being assigned, which, if there are temporal trends, creates a problem. And usually, you have to have a substantial burn-in period where you use standard randomization until the response adaptive randomization. So I would just burn in for the whole trial. I would use standard randomization for the entire trial and not use response adaptive. However, this might be a reasonable thing to do, covariate adaptive randomization, and then adjust for the covariates that you use in the randomization to give you more power. That could be the cat's meow. That was, incidentally, I do a big cat imitation too. I'll share that if you want. So now, if you are going to use unequal allocation, there were some problems of unequal allocation using covariate adaptive randomization that were identified in this trial in Pompe disease, the LOTS trial. But fortunately, there's a beautiful fix by Kuznetsova and Timofeyev. This is not Yevgeny Kuznetsov of the Washington Capitals. This is someone else. But they have a beautiful solution that avoids the problems that occurred in that trial, where the probability of being assigned to the different arms stays the same over time, so you don't run into this problem of the temporal trend problems. Okay, so Bayesian methods and prior distributions, I think Bayesian methods are the natural approach in adaptive settings. I remember once in a non-Bayesian trial, someone said, hey, it looks like, looking at the blinded data, it looks like the variance is smaller than we thought. I think we can reduce the sample size. I was very skeptical. We looked at the blinded data. The variance was about the same. We didn't change a thing. And then someone said, oh, but now we need to take into account the fact that we could have changed something, which is the problem with standard statistical methods. And so I do think it's natural in an adaptive setting. However, I think there is a need to use a skeptical prior, at least somewhat skeptical, that is going to be overwhelmed by data pretty quickly. I would want to see an explanation for how the prior was selected. Often it seems to be sort of a black box. Okay, I'm the statistician. Here are the results. Believe me, just don't, believe me. These are the results. I don't like that. So I'd like to see more explanation for where the prior distribution came from. Now, sharing control arm is a good thing, but it does raise some questions, I think. First of all, how many arms are too many? If you have a promising treatment, one treatment seems better than the other, should you just have that treatment versus control? Or should you add some more treatments where you're going to take patients away from what you think is the best arm? How do you decide how many arms you're going to include? I think there should be more research in this area. Is there a need to adjust for multiple comparisons if you have many arms? Again, we like to protect against, we like to have all the things we say in a clinical trial be true. Now, the argument has been raised, not in this meeting, but in other meetings and in papers. Well, you wouldn't adjust for multiple comparisons in separate trials. But I'm not sure that's a fair comparison, because in this case, in separate trials, you have separate control groups, so you have independent comparisons. Here you have a single control group, and so if you, for some reason, get a bad control group, that's going to affect all the comparisons. And so if you find out later that one of your comparisons turned out to be a type 1 error, then how does that make you feel about the others? Okay, then you start to think, oh, gee, well, maybe that was a bad control group. That's going to affect all the comparisons. So I like to protect, against making any errors. So I have sort of mixed feelings about multiple comparison adjustments, but I do feel very uncomfortable about having a trial that has many comparisons and just not doing anything about that. So the bottom line, I think that compromise is needed. The traditional type of analysis with a large sample size and a robust analysis is not really feasible, I think. I think the master protocol approach offers more advantages than disadvantages. However, I would, again, compare with concurrent controls. I would also avoid response adaptive randomization. I also like what Berry Consultants and other groups have done, where if you're going to use a Bayesian approach, you also evaluate, for For frequentists, you evaluate type 1 error rate and power and those kind of things as well, which they do. That's where the extensive simulations come in. All right, and then the last thing is Florida State 75, Xavier 70, go Noles.
Great, thanks, Michael. Unfortunately, Billy Dunn had to leave the panel. He's not feeling well at all, so we're gonna go right over to Raji for some reaction as well.
Thank you. The presentations, of course, throughout the day, right from the morning have been excellent. And as Michael pointed out, master protocols certainly have an advantage. And we at FDA have, recommended to many sponsors that in certain settings, it may be more advantageous to use a master protocol. But having said that, I think going into something like this, the objective of the trial should be very clear. Is it a screening trial? Is it an exploratory, or is it a confirmatory trial that you are looking into? And also some idea on the pipeline of treatments. There is initial investment is very, very high in something like this, but of course, if you are running it for a long time, it evens out. So without having a pipeline of treatments to open and keep it open for a long time can be costly. The other things are, you have to decide, are you going to use a common control or not? And the knowledge about the disease itself is very, very important. And knowing the mechanism of drugs, if they are very different, and if it's a targeted or precision-guided type of study, then the patient population, to keep it the same is going to be hard. If you set out an eligibility criteria, you may have to amend that as more treatments come out and if they are more a targeted treatment. There is also a challenge in how do you allocate if it is a targeted kind of or a biomarker-driven type of study. You need to think about how do you allocate patients to one arm versus the other if the patients have more than one molecular biomarker or the disease they have has more than one biological or molecular biomarker. The question of long-term sustainability of these master protocols also have to be thought out. How long do you keep it open? When does it become too much to keep it open and you need to consider closing it? In other words, how many patients that you're willing to take into this? And once there are products available, even for a very rare disease, then the whole dynamics changes. Now when you don't have any treatments at all, you can examine, you can get more data, everything. But once a product is approved, what happens to this master protocol? Or how do you then account for it? What changes need to be done? These have to be kept in mind. The whole idea is that you will find treatments. And so if you find one, then, do you still have to keep randomizing or keep continuing with this, or do you have to think about other designs? In other words, these have to be very flexible designs, adaptive, and new diseases keep coming with more and more genetic biomarkers that are being developed and targeted therapies. So we need to look into it. I'd seen that there were 3 specific sort of questions. And so for the, what is the big learning from some of these? You have to be adaptive. Initial investment is high, and objective needs to be clear. And how can something like this be started? Using existing cooperative groups or strong involvement of patient advocacy group is certainly needed in any of these. And a strong leadership. multiple stakeholders have to come to the table, and the operational aspects of it, and particularly if it is international, unless you have somebody who you have a strong leadership, such trials are going to be very difficult. I'll stop there. Thanks.
Great, thanks. Thanks for all your thanks for your presentation and your reactant comments. We are going to open it up to the group for questions, but I do have a few questions back on the panel. All of you sort of seem to agree that the benefits of doing these kinds of studies outweigh the limitations, but still brings us back to the first question. And Scott, I think you emphasized that one of the biggest barriers is just getting the right investment, because the upfront investment is significant. And so circling back on that, what is needed to, in terms of taking the first step? I get that you need investment and you need a strong patient advocacy group can help a lot, but what is that first step? Like, who takes the next step? And it seems like we do need to do more of these, and what would get us over the hump to be able to do that?
So I agree with that completely, and most of them have been patient organizations. Parent Project Muscular Dystrophy represents patients. And so they see the long-term aspect of this disease, and we need to do something. And it's not just this one treatment right now, but it's a longer-term vision of it. They have the ability to raise capital, bring in different parties. We have groups talking about endpoints, industry representatives coming in, patient groups. Those are the ones that have been very successful. Randy's in a situation where the NIH has helped to fund that. We're in a sporadic Alzheimer's trial that was 53 million euro funding of the IMI in Europe. The government is funding it. We'll stay away from government comparisons right now. But that can be a huge aspect of it, in that the one we've seen that hasn't gotten off the ground, despite the fact that it's a tremendous design, is in antibiotics. It's absolutely needed. It's a tremendous place to happen. There are no patient organizations. Who represents people who had infections? Nobody. Should the government fund this? Should BARDA fund this? There are no patient organizations, and so that has sort of floundered as a counterexample of a group that doesn't continue at it and beat down barriers. There are going to be 20 people who say this is why you can't do it, and you need somebody who just moves by and keeps going. Well, we have to. And patient organizations have been wonderful at that.
So considering the variability in the sort of types of companies that are developing therapies for rare diseases, and getting back to the underlying premise of what master protocols are, is being more disease focused. It's not about an individual drug. What kinds of companies are sort of more suited for these kinds of things? So, or I guess maybe to turn it the other way around, what's the real business case for companies that might be really small companies with one or two products in development, or just one, versus companies that have many products in development within a given therapeutic area? How does the business case for these companies change?
Yeah, so this becomes a very interesting interaction with sponsors. You have an existing platform that it's enrolling patients, and you can talk about the number of patients coming in. You can share with them the protocol. There's a cost to come in, time to first patient, and most of the time this becomes a very positive business case. They tend to worry about lack of control. I'm giving my drug to this trial, and they're running it. Have I lost control of my drug? Boy, I'd actually like if you do the battery of tests in the opposite order. I want you to do A before. And we've got a master protocol doing it that way. Are they willing to come in and accept the rules of the master protocol? And then it becomes a question about how quickly can we enroll? What do the resources look like? What is the likelihood for success? And it depends whether it's a phase 2 trial, phase 3 trial. But these tend to be the interactions. It's harder before it's built in somebody thinking about it. But once it's built and up and running, these become very straightforward interactions with them about it. And actually, the idea of being in the same trial, most of them are quite receptive to it. It's a very natural thing for a small biotech company who maybe they can't go out and create their own centers and do this, their interactions with the agency. It's really nice if the master protocol has been to the agency and gotten agreement that these endpoints represent this, that the trial represents success, that there's a path to approval, and that's already been done through the master protocol in a representation at a level that isn't just a single company?
I guess, so another sort of question is, the case is pretty solid for doing these, and it clearly seems to make sense for the rare disease setting. So would they work for all rare diseases? I mean, are there particular disease characteristics that lend themselves better to this type of approach versus not?
I suspect it's probably very similar to a lot of other situations. If you've got a Proteus syndrome where everybody has such a completely different response to the disease, and you run N of 1 trials, these are really hard, but they're more needed in that situations. Places where we have a pretty good understanding of the progression of the disease, there are natural history studies that have demonstrated that. We understand the endpoints in the battery are easier. I don't want to say we should be doing these in almost all rare diseases. I don't see a reason or a case where you wouldn't in those situations. There are some that are problematic. Michael and I both worked on Ebola master protocols. And an infectious disease brings about different scenarios and different problems than other diseases do. Ones that are particularly, as Raji brought up, that most of the therapies are targeted to 5% of this incredibly rare disease, it's harder to have synergies because everybody's in a different 5%. You just have less synergy in those particular situations. The infrastructure would still be wonderful in that setting.
So, Mike, sure, go ahead.
Yeah, I wanted to ask Scott, these companies, you say they are willing to sign on. Do they ever bring up, the issue of, well, but we don't think you should have, 8 different treatments all being evaluated at the same time?
So when they come in, their question is how long till we're done? Enough resources. Yeah. We have to promise that we're not going to sell more airline seats than we can enroll and ask people to leave the trial, is a bad analogy. So we're to promise those resources. So we never bring arms in that we can't support in that setting. So we may have a wait list because of that part of it. So they're going to want to know that you can support the resources for sure. So there are, in situations, too many. That you can't enroll the patients in that particular setting, and then you have to sort of wait list them, and that's a big part of the interactions. They don't want to give you your drug and you don't enroll for 2 years, and then you enroll 1 patient a year. I mean, they would never sign up for that situation.
So related to the number of arms that you're bringing in, and I think, Michael, you brought up this question too, which is, how do you know when to stop adding? And Scott, you brought up sort of the question of, how much variability in terms of the class and mechanism of action would you want across your arms? What are some of your thoughts on, can you get to that assuming you can still enroll patients, but is there an upper limit to the number of arms you'd want to include here?
Yeah, when we do that, it's mostly a function of number of patients you can enroll as to what's the accrual rate to support the number of arms. And we do simulations to say if we add an arm, what happens to each of them in the particular setting. In the Diane setting, there's a limited number of patients who are genetically positive for Alzheimer's. We couldn't enroll 10 drugs, but so we set it up in that setting. How many could we enroll? We do simulations to support that in each of the settings. Just a bit of a back to a question you asked before is, individual pharma are doing these trials now with their own compounds. They have if you're a big enough pharma and you have multiple drugs in a particular disease, they're now doing master protocols of only theirs. But these, I'm generally talking about settings where you're a parent organization in a rare disease and you're supporting that. And suppose there are 15 different drugs that everybody's interested in, you're going to have to make decisions around that, and then you might want to diversify the therapies you bring in so you don't address 4 that have exactly the same mechanism of action, and you haven't done the disease the best service that you could.
Raji?
So, regarding how many arms, particularly if we have a common control, and let's say, we are doing it at once, all the treatments are going in at the same time, it's known that the power advantage you lose after a while, having a common control. So it's not you can have as many and you still gain on control. The increase in power doesn't really affect as much after a while. The other thing is, for the companies to come and work within this kind of a framework, as Scott said, if it's already up and running, it's a different issue. But at the very beginning, it's the question of timing. You see, the pharma companies, of course, there are many pharma people who can speak about that. Timing is everything. So if it is going to take 2 years to get this protocol up and running, they will not be waiting for that. They would rather go by themselves and do the study. And yes, typically, smaller companies who don't have the resources probably come to these more often than the bigger companies. But at the same time, the bigger companies, if it's already an approved product and it's a very rare group that you're studying, then they don't mind investing in something like that. So you do see sometimes the bigger companies also participating. But more than anything, it's the competition and who gets to the market first is very important. And that's where it is. And some of these master protocols, they do worry about comparative efficacy, even though, we say we are not going to compare the treatments within the protocol. They always worry about when you are in the same trial, you are going to compare, and there can be differences. Although, from FDA perspective, the drugs have to be better than placebo, and that's it. And we don't have a regulation to say you have to be better than what is available other than in the accelerated approval pathway. So, these are some of the issues. We have tried in oncology. It's the same class of products, and everybody is doing, this PD-L1 products that are coming up, PD-1 and PD-L1. There are more than 800 trials that are ongoing. And we bring them all together and say, okay, why don't you do a combined master protocol and all of you test it out, have a common control, saves patient resources. Each one is at a different stage of development, and the ones who are already have received approval in many diseases, they don't want to wait for the rest of them to come on board. So there's always, that issue as well.
Great. So we do have time for questions from the audience. So if you do have questions, go ahead and line up at the mics. Also, if you are watching through the webcast, margolis.rare@duke.edu. I just have one question as folks are lining up to the group. Michael, you brought up a lot of questions in terms of operational statistics. One of the things that I think both you and Raji touched on was the sort of multiple comparison, the common control. Any other sort of, you recommended sort of more of concurrent controls. Any other sort of comments from you, Raji, on that, or Scott?
I mean, I always struggle, with the area of multiple comparison adjustment, and I constantly think, wait, am I being consistent here? So I don't really have, a magic, here's when you should adjust and here's how you should do it. No matter how many times I rethink the issue, I always wonder if I'm being consistent. So, unfortunately.
So this would be a great discussion. I'd like to argue against your multiplicity adjustments, that when Amgen runs 3 trials, they don't adjust because they're running a 3rd trial. But this will look like statisticians arguing at that point. But one thing that is amazing about these trials is if I have 4 or 5 experimental therapies in a control, and I'm in a situation where many drugs don't work, you don't have the variability of the control in a single trial, the type 1 error that happens because the placebo did poorly, when I've got all my other drugs that also aren't effective. So I've got this incredible ability to compare to other drugs, and I lose some of those type 1 errors.
Well, this is kind of a cynical comment, but fortunately, it's going to be difficult to be in the situation where you realize you made a type 1 error rate, because you're probably not going to discover that in this, small sample setting anyway. But that's unfortunate.
Yeah. So, multiple comparisons, we have not insisted on adjusting for multiple comparisons in master protocols. But it's really how the design of the trial is and whether actually you need that at all or not. If you think of it as they're all independent, independently these treatments are being entered or being taken out from the protocol, and particularly if we are comparing contemporaneously to the control arm, the contemporaneous control patients, then we have not insisted on multiple comparisons.
Could I just one thing? Dean Follman and I did read a paper that's kind of amusing. And I did write a paper in American Statistician. I don't know the exact reference, but the title is "Multiple Comparisons with Control in a Single Trial versus Separate Trials. Why do we feel differently?" It's kind of amusing.
So if we're in the mood for a comedy,
yeah.
I didn't get that. Question?
Sure. Hi, my name is Deborah Miller. I'm the CEO and founder of Cure Duchenne, and I also have a 21-year-old son with Duchenne muscular dystrophy. And I'd like to thank everybody here, all the panelists and presenters today, the FDA, to be thinking innovative and thinking outside the box and thinking forward. We've talked a lot about, the opinions expressed here, and thank you for considering pharma and some of their considerations, but we haven't really talked about the patient voice here, and I think that that's really important. There's in the Duchenne community, and I assume that this is the same with most rare diseases, the families, the parents are extremely well informed, and they pretty much know every single drug that's coming to trial way before it even comes to trial. And they have very strong opinions about what they would want their child to participate in. And there is such a variance in the severity of this disease in terms of the manifestation of loss of ambulation and such. And so, and there's also a very diverse way of thinking. There's multiple advocacy organizations within the Duchenne community. And I just am curious, or I would like to ensure that the parent and patient voice is part of this discussion, like now. In fact, I think it should have been part of the discussion prior to this, and that information has not been presented to the families. And I think these are not insignificant changes to the way that things have been done before, and we're always open for new ideas and new ways of doing things better. But, the families, again, have very strong opinions about this, and I think that it's doing the patient community a disservice not to be actively involving them at the very inception of this discussion.
So I agree completely, and I wanted to make this point that part of this trial is it's created by patients. The patient organization, Parent Project Muscular Dystrophy, and they're very much involved in the creation of it. And the needs and the desires of the patient is absolutely at the center of it. And they're really disease-focused trials from that setting. And then it's combining together with the needs of the science and the general community. So they're absolutely at the center of the construction of that trial, as they are in the Diane trial. They're a huge part of the construction of the Diane-2 trial, which is nice in a master protocol that can be done before we even know the therapies coming in.
And I appreciate that,
Yeah.
and that's fantastic. However, there are other Duchenne advocacy
Oh,
organizations.
yeah, no, absolutely.
And,
yeah.
and I think that, I've not yet talked to one parent myself who wants somebody else making a decision for them on what drug trial their kid's going to go into. Cure Duchenne is counseled all the time about, our opinions on the different trials, and we will never tell them which one to go into, but we always are able to offer, a lot of scientific and clinical evidence or information for all of them. But it's a big community, and I think that we need to be inclusive at this point and really include all voices at this point.
Okay. I'll go with Alyssa and then over here.
So great panel, thank you. And I wanted to, it's no surprise I'm a fan of master protocols. I wanted to just say a couple of things about the multiplicity. There is one example where it's not controversial, and that's the lung map study that Raji and her staff have worked very hard on. I was looking around for the reviewer, but I don't think he's here. Because that master protocol was set up, it's an example of one at the very end stage where drugs come in really firewalled from each other. And it's basically running parallel arms or parallel trials within a master protocol. So it would probably make you the most comfortable, Michael. But it's something that you might want to look at. And I think Raji was making this point when she said, is it a screening trial? Is it an exploratory trial? Are you trying to find the best treatment for the disease? Or are you trying to get evidence to get 4 different drugs approved under a single protocol? The objective tells you, can really guide you. The point I wanted to make quickly, and Billy's not there, so I'll pose a question to Billy and then I'll answer it as if I were Billy. Is that okay? I'm just kidding.
Please. Yeah.
A big advantage, and Raji mentioned this, but I want to mention it again because I think Billy would have mentioned it, is the FDA interaction on these master protocols. The advantage, and Scott also alluded to this, the advantage of the master protocol being able to come into the agency, talk to the statisticians, engage with them on the design, especially if it's adaptive. This happened in Diane 2, multiple meetings with Randall Bateman and his group for the really without even the sponsors or sometimes including all of the sponsors that might be in that trial or that master protocol, getting FDA buy-in on the general approach, the statistical approach in particular, because it can be complicated, before you get into individual conversations about the particular risk of each drug that might be in the protocol. I think that's a pretty big advantage to the patients, and the spirit being you want to find the drugs and get them to the patients if they're good as soon as you can. And with the antibiotic example that Scott alluded to, you had no less than Janet Woodcock herself come to a public meeting about the antibiotic crisis and basically encourage sponsors to enter into this master protocol so that we get better solutions in a pretty dire public health crisis like drug-resistant pathogens. So you've got there's advantages for patients, and patient advocacy groups may be pushing this, like Friends of Cancer for the lung map study. You've got the FDA that sees all of these trials being run independently and sees economies of scale and, encouraging them. So you've got both of those forces encouraging them. Yes, it's a big leap for sponsors, but, I think in the rare disease setting, it's maybe easier to see that the advantages are really there. So I think Billy would have said something about that, meetings with the FDA.
Great, thanks. Thank you, Melissa. Go ahead.
Hi, Martin Jones from AstraZeneca. In the FDA paper on master protocols, they use the term master protocol for basket studies and also umbrella studies. And you referred to basket studies, but then really talked about umbrella studies. I was wondering whether the panel had any experience of running pure basket studies, and what are the complexities or the differences from the umbrella studies, particularly outside of oncology, if there are any examples?
Sure. So I'll answer the question I want to answer. I think it's what you're asking. So by basket trials, you're taking typically a single compound in, and you're investigating within a disease that you label the disease as heterogeneous. It could be, in some trials, in stroke trials, we investigate time from onset of stroke to treatment. Do we have differential benefit? It might be genetic types of it. In many of the ICU settings, it's time from onset of injury to treatment. There can be many things that define what could make it heterogeneous. And so a company or a trial gets started, and we investigate many of them. And there are a number of these trials happening, and you might do adaptations where you stop enrolling particular subgroups where the drug is not beneficial. And you might continue on into a phase 3 trial within a subset of these, adaptive enrichment. The important thing is all the analyses you're doing recognize the potential differential effectiveness of the drug in the different groups. And then how you model that, do you model it as a Bayesian hierarchical model? Are they ordered? time from onset of injury, they're ordered groups. So you have a model that looks at the benefit of that as a function of time to make your adaptive decisions or your end of trial decisions, because the multiplicity there can be huge in the different baskets.
Okay, thank you.
Let me also say that, in general, those basket studies where basket designs where they have been used are where you have something common among that heterogeneous group, some way of defining either the disease or what have you, and the drug is supposed to work on in all of these heterogeneous groups. If you don't have that reason to do it, then you shouldn't be doing a basket study. You have a good reason to say, yes, it can work in all these different heterogeneous groups.
Thank you.
Yep, great. Go ahead.
Hi, Katie Donahue from the FDA. I'm one of the team leads in inborn errors. And I have a question for the panelists about what we can do to minimize the risk of temporal bias, because I'm sensitive to why you flagged that as an issue. But by only using concurrent controls, it would seem to eliminate a lot of the benefits of one of these platform trials. And I think it's an issue that cuts across all of the panels we've heard today, which is that an evolving standard of care, we want the standard of care to evolve, but then it creates these huge challenges for registries and historical controls. And it's an issue that Gigi McMillan framed, I think, really nicely in our first panel, that we have parents who know all about this and they care about it, and this is their everyday life of maintaining the standard of care, and what can we do to capture it more accurately, both to help patients and the science.
Thanks for bringing that. It certainly came up a couple of times.
Yeah, one of the beautiful things about a trial that's continually running and you have overlap of arms and you bring in other ones and you have overlap. It's a little bit different if there's a time when there's no overlap of arms, but maybe you have a control that continues on, is you get to observe time in your trial. So we model time as a factor. For example, in I-SPY, this is neoadjuvant breast cancer where they're differentiated by hormone receptor status and HER2 status. There's been almost no variation over 6 years. In the Ebola trial, we worried that Ebola was going to be different next month than it was now. But if you have arms continuing on the trial, you actually observe and can model the effect of time, which allows you to use data at different times if you model that effect. But this is a whole new thing in these trials to understand the time effect. So we do concurrent controls. We absolutely do. Your randomized concurrent controls as a subset, all controls, and then a model adjusted for time. And these are factors that are randomized comparisons to learn about in a master protocol, where if you run separate trials, you don't have the connectedness to be able to understand those differences.
So then why wouldn't that be good enough for Michael?
Well, it's not good enough for me because to me that's not a randomized trial anymore. If you take controls now, I agree that it makes a big difference about whether there's expected substantial change over time. But I mean, I've seen a lot of trials that have a pretty substantial change over time, and so. So to me, if you're going to do that, it takes it out of the realm of a randomized trial, and to me, it becomes more of a, more of an observational trial. But as I said, I wouldn't have a problem with, as you say, doing it more than one way. It's just that I personally would make the concurrent control the primary analysis.
Okay, question?
Waldorf and AbbVie. That question was, and the answers was a perfect segue to my question, which is time is one factor. So Scott, I was happy to hear you say that you do model it. But there's another important factor, and let me ask a question, clarifying question, then I'll make a comment. Clarifying question is, just suppose you have a master protocol, you've got 2 drugs in that master protocol at a given time, new patient presents, I assume there are baseline characteristics and biomarkers measured, and then based on that, they go either into drug A versus placebo 3-to-1 or drug B 3-to-1. Is that correct? So they're different based on the mechanism.
Some of them can be you randomized to either.
Okay.
You're eligible for both.
3-3-1-1. Okay.
Right.
Okay. So I guess it's for the case where there are biomarkers. Like I think you mentioned you'd like to see drugs with different mechanisms. So if you want to do what's best for the patient, you would actually earmark them to.
Neoadjuvant breast cancer, a drug may come in only HER2 positive cancers.
Yeah, okay. In that case, the bigger concern, and this is where I'll agree completely with Michael, I don't think the non-concurrent placebo patients adds much because it's a different patient population. And the other thing I would add is that the schematic you had on there where you had 2 drugs initially in there and then they would either graduate or flunk out, you didn't mention that, but I assume there's a way that they can finally graduate and you no longer study them or they flunk out. And then you have patients later. That's really the same thing as doing, I think, you can push back on this, it's the same thing as doing naive pooling of a historical data. Like I've got a phase 2 trial, placebo and drug, and I've got a phase 3. I'll just take those placebos naively and just throw them in there. And I say it that way because one of your slides said 60-40 instead of 60-20, that you showed 90% power by using the 20. But my point is you may not be able to use those 20, or at least not a full 20.
Yeah. So on both those points, the biomarker positive, if you come in and you're investigating biomarker positive, in Duchenne you're investigating ambulatory boys, you're not going to borrow a control that didn't have that characteristic, but we've been modeling that throughout. So you borrow biomarker positive controls. Yeah, absolutely. That part of it's balanced. In the second setting of what's so different about this than taking a different trial with a different protocol, what creates the biggest variation in controls is the protocol itself, inclusion, exclusion criteria, and what you do to the patient. That's identical in this. So we might have, for a while, we're randomizing patients in this period of time, and then a new one comes along. They were randomized in the exact same protocol with the exact same endpoint, the exact same procedures, same inclusion, exclusion criteria. Everything's identical of it other than time.
Yeah.
And so the question, that's a very, very different thing. And we've got interesting names for these. It's not historical data, it's non-concurrent randomized controls, other names to that. It's a very different beast. And when you've got arms that then overlap this, you can understand, are they different or not?
Yeah.
And that can go into your analysis. So it's a very different thing than historical data.
I agree with everything you said. That's in the case where you don't have a biomarker that would target a patient to drug A versus placebo. But
the important thing, the 60-20, 60-40. In the Diane trial, there were 2 arms in that enrolled 60-20. It's not exactly 60-20, but 60-20, 60-20, concurrently randomized against each other.
Yeah, I understand.
So the additional 20 were randomized at the exact same time that arm came in. The only difference is they get a green pill rather than a blue pill.
Yeah.
In the trial. And then we talk about, does that matter? And that's an interesting discussion in and of itself.
Yeah. And so in that example, they are concurrent. Makes sense.
Yes.
Yeah. Thank you.
Okay, great. Thank you. Time for one more question.
Yeah, quick question. Maybe a long answer, but I want to go back to the multiplicity issue, and I want, especially to Michael, and ask if you have any idea how much inflation of type 1 error you have and how much that will be relevant when we consider that these master protocols are used in rare diseases in which you won't have enough sample size anyway.
Right, but yeah, you won't have enough sample size, but I mean, you will have a multiple comparison issue. It's a question of whether, whether you care about that or not, but I mean, certainly if the global null hypothesis is true, irrespective of the sample sizes, you will have an inflated probability of at least one false declaration. So it is there. But like I said, this is one of those things that I feel really uncomfortable about because I certainly don't feel like I have the answer, you should always do it this way, or anything like that. And I always struggle with, are they really needed here? Are the multiple comparison adjustments needed in this setting? And I could see why in this rare disease setting, obviously that requires a larger sample size if you insist on that. But I certainly still feel uncomfortable with, 10-arm trial and just saying, well, here's what we found. I certainly don't feel comfortable that all the statements that were made are correct. Now, whether that should bother you or not, I don't know.
Well, do you feel comfortable when you say the statement is correct with 1 or 2 trials with a p-value of 0.05? There's still uncertainty there, depending on the adverse events, for instance, of the treatments. You should not feel comfortable even if you are pretty certain about.
Right, but in 2 separate trials, though, they're 2 separate control arms. And so you have independent comparisons. And to me, that feels different. As I say, because when you do them in the same trial, you have that same control group. And if you did find out you made a mistake, then the conditional probability, if the global null is true, conditional probability of the other one being a mistake is much higher.
But if you have the 2 other arms, the probability that you make one mistake is also smaller because you have another comparator that's not the control arm. So you have to take into account both ways to account correctly for multiplicity.
Okay, so this next session, we're going to have lots of opportunity for all of you, the public, to ask your questions, but maybe one quick comment.
Yeah, just a quick comment about the multiplicity control. Consider arm A, arm B versus control. The type 1 error rate is controlled. The only difference is that given the control, what is the probability A and B simultaneously get proved? That is different. If you have 2 different control trials, that probability is much smaller, 0.00625. But given the same control, the only problem is if the control is bad, then it's holy water, approve all the drugs. If it's too good or very good, then no drug gets approved. So marginally, type 1 error rate is controlled. Only difference is the probability of simultaneously approve a drug that is different. So we have to think what we are going to control. Is that altogether less than 0.00625 or just one drug, 0.05? That's why I'm thinking 2 different drugs from 2 different companies is different than one drug plus different backbone drug. The former one, I think there's no need for multiplicity control. But one drug from the same company, different multiple backbone drug, we need multiplicity.
No, I absolutely agree that part of the decision making about multiplicity should be, and I ran into a lot of trouble for saying this, but I absolutely believe that part of the consideration of whether a multiple comparison adjustment should be made is whether a single entity stands to gain from the multiplicity. And, like if you take one drug and you break it up into 5 different doses and you declare victory if any of those doses beat the placebo, that doesn't feel right. If it's by 5 different companies, that feels different. So I absolutely agree that there are other considerations like that go into whether a multiple comparison adjustment is needed.
Okay, thanks. I'm sorry, that's all we have time for this session. So let me thank our presenters and reactors. It was a wonderful discussion. I'll invite Mark back up here to introduce the next session, which is public comment. So all of you get to ask questions and continue the discussion as well. Thank you.
All right, we are going right into this next session. There will be a break after this session. And I'd like to ask several people to come join me up here as we listen to these comments and perspectives. And that includes Laura Lee Johnson, the Acting Director of the Division of Biometrics 3, who you heard from in our opening this morning, Alyssa Lavange, who is now Professor and Assistant Chair at the Department of Biostatistics at UNC Chapel Hill, who obviously you all have heard from as well today, and Aloka Chakravarti, who's the Acting Director of the Office of Biostatistics at CEDAR. So some of the important perspectives from today's discussions represented here. Now, we've had a lot of time for comments from many of you along with questions during the course of the day. What we'd like to make sure we do during this time is get any additional ideas or issues that you all would like to see considered as part of these efforts around utilizing innovative statistical methods and trial designs in rare disease settings as this work continues at FDA and with collaborators throughout the healthcare environment. So if you have a question or a comment, please go to the microphones again. For those of you who are joining us online, again, it's margolis.rare at duke.edu for a chance to get your comment in online. We'll definitely take time to read those publicly as well. I suppose we could keep talking about multiplicity comparisons if you all would like to, but please come on up, and I'd like to hear from all of you. And again, let us know who you are when you make your comment.
Hi, RJ Worth from Vector Psychometric Group, and I just want to thank everyone who has presented or was involved in this today. It's been a wonderful, wonderful meeting. Given my interest in sort of, where I spend most of my time is in endpoint development, I would just like to put out there that it'd be great to have another meeting like this that focuses much more on endpoint evaluation, selection, and development, because we face a lot of the same problems when trying to develop a new scale or a new measure or trying to select which one is best for a particular study. But there's sort of unique challenges in that in and of itself. Just trying to understand how an item functions in a population when you only have 10 people is really hard to do. So I would love to see this general discussion continued at another meeting in the future.
And if you all have any comments that you'd like to make on the comments, please feel free.
And so thinking about that just a little bit, and always happy to have another measurement meeting. But one element that comes up is many times we see folks that are groups that are trying to create de novo, so a brand new scale, a brand new tool, where realistically there might be, if you're trying to measure physical function, there might be a tool that's already out there that's applicable for the population. But again, thinking about some of those examples that have been mentioned today, you need to choose something that's going to work for that population that's going to be in the trial for that duration that you expect them to be under study. But there are instances, I think, in MS and a few other places where they've turned to tools that were already developed and basically did some qualitative work to say, yes, these tools are applicable, the changes, the scoring makes sense, now we're going to use them in our patient populations. And so that's a way to take something that's already developed and make sure it'll work instead of trying to create brand new every time. I remember my NIH study sections, they may like the brand new, but realistically, it's not always your best way to go.
Thank you. Please.
Gary Rosner, Johns Hopkins University. And this comment question really stems from the previous session about registries in natural history. And I was struck by the fact that something that I've learned through my work at Johns Hopkins is just how the implementation of electronic health records has really not lived up to its promise. And in particular, they don't easily speak to each other across all the different institutions, and there's a particular company that has a huge percentage of the market, and still each institution seems to be its own little instance, its own little island. So what can we do, or what can anyone do to try to use that as a way of generating natural history information at the very least? I know diagnostic codes are going to be different because those systems are really billing systems and they're not clinical research systems, but still natural history labs, things like that, could well be shared across institutions, and I think that would really help a lot.
So one of the things I think we can do is to learn from some of the large-scale databases that we have experience on. And for example, if you think about the Sentinel experience, what that needed to do was to create a common data model so that you're capturing the same units of information and in the same granularity, because at the end of the day, you wanted to put it together and to be able to interpret the combined results in a meaningful way. So you're right that electronic health records do have their own things, and if people are developing them in their own silos, it becomes very hard to put together. And in many situations, the missing information is probably the most crucial information that you are looking for in a particular domain. So that is very important.
And I think I saw Jason Gershon here earlier today from PCORI, but I think this is something that was really addressed was they were looking at PCORNet, and especially PedsNet. How do we do unified research across all of these? But I'll also say the sellers of those EHRs will comply with what they're asked to do. And so perhaps we didn't always know what to ask for if you were trying to buy those, but time has taught us some things. And so that's something to also consider, which is I always remind people as they're going to contracts, really read through the contract and make sure it's helping you get what you want.
So I'll take a moderator prerogative and add a little bit more onto this on this last point about really getting to the intended meaningful use of electronic health records around interoperability. HHS made some significant announcements last week based on new authorities that were in the 21st Century Cures Act for not only further steps on standards for interoperability, including the use of open APIs for at least most or many elements of EMRs that aren't free text at this point, but also, and even some that are, but also penalties for, quote unquote, data blocking. Now, they still have some work to do to define what exactly that means in practice, but there is a significant amount of financial penalty authority that HHS has said that they're going to use. Beyond that, I wanted to pick up on another major theme for today, which is the importance of patient groups in making these kinds of activities, especially involving large curated natural history data sets or registries, work well in the rare disease space. There are some new opportunities that did not exist before for patients to get control of their own data. I'm not sure if Hopkins is part of it yet, but for example, Apple HealthKit announced earlier this year that they had partnered with a number of major health systems, including some major academic centers that use some of those proprietary EMR systems to make it easy for patients to download their electronic record data. And CMS, as part of those HHS announcements last week, announced a version 2.0 of their Blue Button system that is open API based for having immediate access to your Medicare claims data. And a number of the health insurers have said they're going to participate in the same program. So when you're thinking about these systems going forward, I think having a more active component around using patients working through apps, getting access to their own data could really help improve participation and data access in these programs. It does seem like it's been a challenge, but it does seem like that the future is going to be different than the past, hopefully quickly. Next.
Hi. Excuse me. Rendell Marshall from Artora Therapeutics. There was a comment earlier about the importance of considering cost-benefit analysis in addition to risk-benefit. And I think that could be an important theme for us, because to me, cost means time to review and approval for these families and patients. We're all happy that we've moved away from totally unrealistic expectations for the level of certainty required. But what I am sensing in the field as a scientist myself is that desire for certainty is creeping into our other kind of innovative design discussions. So we've seen a number of examples from of gold standard examples where there are large, well-established, well-funded patient advocacy groups. There's years of research. There are several drugs out there. Everybody agrees on the endpoints. That's not my read of the rare disease space. To me, that is the exception, not the rule. And I've worked in multiple spaces where there are maybe a couple hundred patients at most. There are no approved drugs. There's a terrible progression to death or disability. There are a handful of advocacy families who are loosely organized. They have no money. They're not scientists. They have a few doctors who've taken a special interest in the disease. And then there's some clinicians out there who are also following these patients until they die or have to be institutionalized. Then when we move into that space, of course, the new vogue is we need a natural history study, which to me is functioning largely as a placebo group substitute. And the easy way to do that, and the fast way to do that, is to get in touch with all these doctors and cull what information you can from their medical records. That information is inevitably messy and medically driven and practical. And some researcher, and because of the heterogeneity of the disease, some patients you follow time to respirator. Other patients might be loss of ambulation. Another patient might be difficulty communicating. And another doctor's interested in the labs, so he's following labs. So you pull it all together, and it's very difficult to quantify. And then which is intrinsically unsatisfying to most of us in the room. So then what you really want is a prospective study, which could take 2, 3, 4, 5 years. And by the way, you need to develop some kind of an endpoint that everybody agrees on, and that can take a year or two and lots of money. And I can tell you from personal experience that these families have no patience for that, no patience. And so my thought would be that in a space where there is no approved treatment and no disease-modifying treatment, maybe the cost benefit could influence the bar on the risk benefit. And then as we learn more about a disease, or maybe for the second or third or fourth treatment, the bar could get higher. Or we could move approval earlier and then continue to collect data. But what families have told me over and all is they don't want something that they know is safe and that they know works. They want something that's reasonably safe and that might work, or something that's reasonably safe and has a decent database suggesting that it could work for some patients. That's all they care about. And if we're, to me, as both a scientist and a physician, I'm torn between the two, right? Because I'd like to let the clinical reality, which is very stark, influence my thinking as a scientist and how we can continue to be more and more flexible in response to this desperate need. So maybe that was a soapbox, but anyway, I'd be interested in the panel.
I really appreciate the comment. Thank you. And if any of you all want to.
Yeah, that was a beautiful comment, and so much nicer on the ear than arguing about multiplicity. So when I was at the FDA, the two things that I've always thought about in rare diseases, I want the best evidence I can get. I'd love for it to be something a cut above anecdotal evidence. No, it can't probably go to fully powered randomized controlled trials, both with a p-value less than 0.05. But the best evidence I can get given the situation, and then I want to make the best decision I can make given the limited evidence that I probably have. And how we go about doing that is, that's what we're here to talk about today. And there's no easy answers, but that's what we want. And we want, maybe you add a lot of things to that. You want between the evidence and the decision, you also want to engage the community, particularly the patients and the families, and understand the impact of your decisions on all of the players that are here or that are affected. But that's the devil's in the details.
Yeah, some very special challenges for the ultra rare populations that starkly highlight the issues that we've been discussing today. So we have a couple of comments that have come in during the course of the day that I'm going to paraphrase from our online audience, and thank you all for sending these in. One relates to the natural history data sources. I think the questioner or the commenter suggests that most of these are likely coming from academic centers, and does that have implications for data standards, collection methods, and documentation? Is it different than, say, the traditional studies that are done through sponsors collecting data according to standards that they presumably have worked out or have some expectations will work out with the FDA? I don't know if any of you all want to comment on that topic. And going to our earlier comments, maybe future natural history data won't be all or mainly driven by academic centers alone.
So there is a wide variety of natural history data. I'll say that politely. And I do think, though, that ultimately data quality is an issue. So there can be data that's there, but it may not be usable for a particular study, or it may be usable to get an estimate in order to actually develop your next study, but may not be usable as an external control group. So for different settings, again, somebody described earlier the different types of variants, and that's really what it comes down to, is the amount of trust, the amount of data that's there. And so, there's a lot of variety. You might have a natural history data source, for example, from an academic group that goes back 40 plus years across multiple different countries, because those couple of doctors that the previous person talked about have, at least we have that information from those medical files. And sometimes there's a mixture of retrospective and prospective data that's put together, but maybe you don't have the time points you need, but you can kind of put together what the trajectories might have been for the time points you care about. Maybe they didn't quite measure it the way that you're interested in, but you're still able to cobble together an endpoint in some way. But you may have sponsors that are running trials that are far shorter in duration, so you don't have that long-term information. So a lot of times, I think we have it in our mind, and maybe it's because it's the basic way we're taught about clinical trials. I have a control group and a therapeutic arm. But a lot of times we're pulling together information from a lot of different sources, some supportive, some actually used in other ways. But a lot of times there may be multiple control groups, and we have to kind of build a pen around. So I grew up in Texas, so I think about things like this, where I'm like trying to add together all my fences and gates together to say, okay, have I surrounded what I think the truth is? Because realistically, we have to make a regulatory decision. And so how confident are we that we're going to put a drug on the market that should be on the market, or that we're keeping a drug off the market that shouldn't be on the market? And so that's kind of where I see a lot of this. But there's a lot of varying quality for a lot of different reasons. But I would say, and we've talked to our academic partners about this before, do data audits. There's no reason that in an academic setting you can't have extremely high quality data that's good for regulatory use. And we've seen some beautiful data come in too. I know you all want to comment more.
I wanted to touch on something important that you said is the natural history can augment the information that is in the follow-up data, because many times it's not feasible to do a study of that duration, but a natural history study or registries can provide that additional information that is needed for long-term treatment and care of the patients. That's important.
Okay. And one other specific comment question relating to the Bayesian methods that were discussed earlier. This is a question about, I think Dr. Price used the term dynamic, and the question relates to whether as data are accumulated, does the similarity or difference with historical data influence how much strength is borrowed from these other data sources, or is it better to pre-specify how much borrowing there will be, which seems more like, according to the questioner, more like a static model. I don't know if, Alyssa, you want to take that one?
So Karen should probably answer this, but I know that there has been there was interest when I was at FDA in some methodology that was proposed. I think it's published in the 2014 special issue of Statistics and of the journal sorry, no, it was Pharmaceutical Statistics, the Wiley journal from the UK. There was a special issue on Bayesian methods in drug development. Karen and I were the guest editors for the issue. There was a paper by, I think, Kurt Veal is the first author. I think Scott was a co-author about having an imbalanced randomization to begin with, planning to leverage both concurrent controls and historical controls, stopping a couple of times at interim, comparing the similarity of the two sources of controls, depending on how similar they were, adjusting the randomization ratio. So you could imagine in the extreme, if they were completely similar, you could stop randomizing to concurrent controls altogether, or maybe go up to a 5-to-1 or something. If they were disparate, you forgot the external controls, went back to a 1-to-1 randomization, and tried to make up the difference. And I think the trick here, people, you know, the transparency and the beauty of the Bayesian approach is putting your beliefs up front, and people get nervous when you change those beliefs based on the data. So you would need to do this in a prescriptive fashion, and now I've enticed Frank to get to the microphone and give you the right answer, which is my goal all along.
Yeah, dynamic but transparent and pre-specified. Yes, go ahead.
And
Go ahead,
somebody can correct me if my understanding about this isn't correct, but if you're talking about dynamic borrowing through a mixture of two prior distributions, you might have a 50-50 mixture where one prior is very skeptical and another one is trusting historical data. And so you start off, with a 50-50 mixture, you start off with prior odds of 1 in terms of the borrowing. And when you're done with the study, you might solve for the posterior odds, which might be 3-to-1. It's say, well, from what we've learned with the new data, we're actually, the posterior is constructed after the fact with a different mixture. But I would say the word dynamic isn't necessarily a very good term for what's going on, because you set the amount of borrowing when you start, and you don't really change that. It's 50-50 no matter where you are in the study. But you can solve after the fact for the updated estimate of where information was coming from. Anybody that has a different understanding, I'd like to hear it.
I also have I've seen some work on the power prior, which is something that also Karen mentioned this morning. In that approach, what I've seen is people do pre-specify before seeing the data the actual function that's going to be used for discounting. But the amount that you will discount from that function will be determined after you see the data. So the methodology of actually discounting through a function is pre-specified.
You just said after you see the data.
Yes.
Before the data?
No. The amount of, so there's a function, and so this power prior has a likelihood raised to some power, which so what's the value of that power? I mean, the ideal would be to pre-specify what that is, but in this alternate approach, which I think is used in the Center for Devices, you specify what's the nature of that function, and then you can read off the power from that function after you see the data.
I see that we have inspired some further discussion of Bayesian methods. We'll let this go on for a few more minutes. Right?
I think Frank may be right that dynamic is not a great term for these methods, but there are a variety of methods that people have called dynamic, all the way up to classical Bayesian hierarchical modeling. And the dynamic aspect is that the amount of shrinkage of the data depends on the compatibility between the prior and the data. and people call that dynamic. It may not be, but you are pre-specifying the approach before the study.
And I can give a hint about my experience with the Center of Devices. When we pre-specified and we did borrowing, we used an adaptive design in addition to that. So where was the dynamic was that, depending on interim results, we could increase the sample size of new patients. In that case, even if you're borrowing the same thing, you will increase the strength of the current trial. And as John said, updating the parameters of the hierarchical model. So it will be adaptive in these two ways. We'll have a prior distribution and we'll have a adaptive design that will increase or decrease the sample in the current patients. And I think that's fundamental when you use prior distributions. My experience before, if you pre-specify the amount of prior and pre-specify the sample size, you're probably going to fail because you don't know how much the new data will be similar to the old data. So what I would advise is every time you do that, you should have an adaptive design in addition to the borrowing from a prior.
Thanks so much. Didn't realize there was such passion about these two types of approaches. And so I thought it was an interesting comment. I know Roy earlier voted for static. I personally prefer the dynamic, which may, again, be not the right word. We can come up with a different word. But in that paper that Lissa's referring to, there's discussion about the a priori ability to show how under various settings that will happen, how much it's borrowing from the different pieces. And I think the mixture priors or hierarchical methods, part of the reason I like the mixture prior approach is because you can bring in the skepticism, you can bring in expert opinion, you can bring in different data sources and a priori specify, as Frank mentioned, how much you're going to give from the get-go to those. And then as the data comes in, the posterior will update appropriately. And if it's looking similar, then it's going to borrow from those pieces. And you'll see, it was hard to tell, I know, on those plots, but one of the nice things about the dynamic is as it deviates away, then the probability of showing an effect when you shouldn't, so it's beginning to get at this sort of type 1 error discussion, will either plateau or then begin to decline, whereas in a fixed approach, it would continue to escalate. So it does give a way to sort of dampen the amount that you could have a type 1 error, if we want to talk about it in that setting, or an error of some sort. So that's one of the reasons why I personally prefer the dynamic approaches. I think the question may have been coming out a little bit, what would the FDA prefer? And so if there are comments on that, I think that would be welcome, certainly as someone from industry, but just to clarify a couple points on that.
Thank you. Any further comments on what the FDA might prefer or other things?
Pre-specify a good plan,
yeah. It seems to be
and then we
recurrent
will
theme.
discuss.
Pre-specified. I have one more from the plan pre-specified. I understand. One more comment online from Michael Freese at CSL Behring. This is on the topic of population shifts over time in platform studies. So he notes that there are two events that can change populations quite quickly, regardless of consistent or constant enrollment criteria. One is when a new treatment becomes available, when the first drug becomes available, the population available for a master trial protocol is going to shift. We've talked about that to some extent. But he also highlights when a new diagnostic tool becomes available, which he says often happens after a treatment becomes available, as now there's a real motivation to develop the diagnostic. And what he's seen in the past, especially in prostate cancer, is population shifting to earlier stages of diseases. And also, even without a new diagnostic, this phenomenon can occur through patient education, outreach efforts. If a treatment becomes available, this can shift the stage of disease that's available to a study, lead to a widening in effect in enrollment criteria, also can cause changes in original power calculations, et cetera. And this is something asking is this something that can be handled in a master trial protocol?
So I just have a couple of comments, and there are things I've said before. First of all of these things can happen in the course of a single trial, and you have to deal with that. You may have a single-arm study, you're relying on a registry or a natural history study, and then things change with new approvals or new diagnostics. The one advantage I see in the master protocol is you have multiple sub-trials or multiple sponsors engaged, and you can work together to figure out how you're going to solve the problem. And I use LungMap as an example of this, where there were 4 or 5 sub-studies going on, as well as a no-match arm, no biomarker match arm. Nivolumab hits the market. That docetaxel had been the standard of care. Out goes the standard of care. What are you going to do? And there was criticism leveraged against the master protocol. Oh, look, there's 5 or 6 different trials that are all affected by this. But to me, being glass half full, I thought it was great because you had all of these sponsors working on this problem together in a collaborative fashion rather than having 5 insulated conversations all about the same problem. So I actually thought the master protocol helped, even though it caused a pause and a little bit of a redesign. So yes, all of those things are inevitable, they change, but you can maybe leverage your collaborative setting in the master protocol to come up with a really good solution.
Thanks very much, Lissa. Please.
Hi, this is Ivan Chen from AbbVie. A couple comments. The first comment refers to the platform trial where we might have multiple arms or multiple groups of randomization going on, but they may be sequential. And one of the issues that we talk about is potential bias, and can you combine the controls, for example, from all the randomization? I think it sort of depends, and I appreciate what Scott had talked about, that they're being run under very identical, similar conditions. However, if we are waiting long and to add new arms, then they may become farther and farther apart. So one needs to be careful whether you can really completely combine the control in 100% sense that lump it all together. So we might have to use a little bit of the historical control borrowing technique to address that. And the other question is around this historical borrowing in common is that inherence, there might be potential bias one way or the other, whether you want to talk about in terms of type 1 error or whether in terms of effect size estimate is not going to be the same as the concurrent controls. And I'm interested in hearing maybe from the FDA's views about how flexible is in terms of looking at some methods that might be, whether it's inflation or type 1 error or potentially is get into a bias depending on the comparability of the historical controls. Just to get a sense of that would be great.
Thank you.
So I'm not going to directly answer your question, but I am going back to kind of that beginning part, like how much we're going to talk about multiplicity or type 1 error, et cetera. And I think, John Scott, maybe you had it on your slides or somebody did, which is, I don't want us to get too caught up on we need strong type 1 error control. That said, I don't want us to say, oh, I found one thing out of 100 and now it's awesome. So I think most people in the room are good at that balance. So, I have to state it. But I think we many times do a disservice. We have regulation that tells us for each of our different centers what is approvable. Kind of that sets those standards of evidence there. But it doesn't say there must be a p-value, it must be 0.05. So I do think we need to think about this in a more holistic sense, and that's going to help us move forward in better decision making.
You also mentioned the bias and lack of comparability, and I think we've talked this morning about attempts with either covariate adjustments in a model-based analysis or propensity scores in a matched analysis, whatever is the most appropriate for that setting. I think everybody goes into use of historical controls with that in mind. Everybody knows what the pitfalls are. And it then becomes a playoff, right? So if I have no chance of a randomized controlled trial, I can only get 25 patients enrolled, and I'm stuck with a single-arm study, then am I going to just you know, if how bad does a potential bias have to be before I just throw away whatever natural history or registry data I have and deal with a single-arm trial, but then how am I better off? I mean, I think it all comes back to how can I best use the information that I have. It may not be perfect, I know it's not, but I'm in this pickle, and I've got to get the best information I can. So you apply the statistical tools that you can, but, you know, I just would keep thinking about the patients and say, don't give up too quickly. I guess that's my overriding emotion.
Yeah, I once sat in a room with 100 FDAers. I think we had more people at FDA working on this than there were patients in the United States. And we were trying to figure this out, say, okay, like what is the path forward? And we found a path forward. But that's part of it is saying kind of, okay, what can we do? But it depends on the situation you're in. And sometimes there's not a great answer, except there's a way to say, okay, this is what we need. But we can be creative, but it is a big collaborative process along the way. But a well-done trial, even a 25-person study that's really well done, and you have folks that have been collecting that natural history information, all this, like, that speaks. That's important information. But I think Petrula mentioned this earlier, like sometimes, like, you know, it's easy. Like if everyone's supposed to be dead and they're all now alive, that's pretty easy to handle. It's that we're usually nowhere near that situation.
Thank you.
Waldorf and Avi, I have a different kind of question. I think we would all agree that in rare diseases, you do studies, they have to be small. The chance that a product gets approved by the FDA that later we learn wasn't effective is higher than in the non-rare setting where you have hundreds or even thousands of patients studied and 2 studies have to be significant and so on. So my question is, do you think there's a way that the FDA could grant conditional approval, but maybe even to Roy Tamura's comment regarding endpoints, moving from binary, which it is today. It's hard to say this drug is approved for this indication when you may not be sure. It'd be nice if there were different stages of approval, and so a drug for a rare disease may get on the market with something, you know, that says this has been shown by studies of 40 patients or something like that. It would make kind of clear what it's based on, and then as more data comes in, perhaps from a registry, it can be updated to stronger and stronger approval until it finally gets full approval.
So I want to start this and pass it to Aloka. Because we've talked about this internally when I was at FDA. So we have accelerated approval that will put a drug on the market and on an intermediate outcome while you're accumulating data on a more rigorous clinical endpoint. And that's been very effective in oncology. A lot of these rare diseases don't have those early outcomes, at least not as good as we'd like. My understanding of our regulatory authority, and Mark, you also know about this, we cannot approve a drug and require a post-market trial for efficacy. We can require a post-market trial for safety. So accelerated and this actually was what I meant to say to the gentleman sitting right here and when you asked this question too. And I mean, I think there had been discussions maybe around the 21st Century Cures Act of that authority and whether it could be expanded, but that kind of conditional approval isn't in the FDA's purview right now. But accelerated approval is. And to that end, the discovery and validation of early endpoints, which comes under biomarker validation, is critical. And a place where I think FDA can really work with patient groups, with academics, with lots of scientists to come up with these. Aloka heads the biomarker group at FDA and working with NIH as well. And anyway, I just wondered if you had anything to add about that aspect.
Yeah, so one of the issues that is particular in this case is there are very few early biomarkers or qualified biomarkers. So that adds to the challenge of going the accelerated approval path. So one of the things that we have tried or to look at is to see if there are biomarkers for safety, because many times, they are very interrelated, and kind of put the two arms of the risk-benefit discussion together and see if that's the path we can go forward.
I think this discussion could go on for a while longer. I'll just say that we've had a lot of discussion today about, life isn't quite binary, and FDA standards are safe and effective treatments subject to biomarkers for accelerated approval and the like. But we've heard a lot today, and there's a lot else that FDA is dealing with around patient-focused drug development initiatives. What should the endpoints be? Where I think these issues are to some extent live, and certainly are getting to be more opportunities to develop better evidence on safety and effectiveness after approval with all that's going on with real-world evidence and platform studies and the like. So stay tuned, I think, for
yes, and one thing that I wanted to, I forgot to mention is many times, when there's residual questions about the efficacy, we will put that as part of the REMS or, things that has to be answered after when there are more information available.
And certainly a lot of interest from payers, purchasers, and patients in getting that better evidence. So we are about out of time. So this has been good discussion. I want to get in your two comments quickly, plus one more, last one from online. Sarah O'Meara from Regeneron Ireland comments. Given the subject of rare diseases and the need for it to be considered in a global global development context, have steps been taken to engage with other regulatory authorities, ICH, EMA, PMDA, et cetera, to aim for global alignment on the regulatory requirements for some of the innovative trial design and related issues discussed today? Is there a quick answer to that? A quick answer hopefully is yes.
Yes. Actually, I'll probably talk about this a little later, but in the ICH E17, we particularly call out that there are special challenges to rare diseases and the need for regulatory collaboration and regulatory alignment early on in the planning.
Great. Maybe we can come back to that. And so last couple of comments quickly. Yeah, please go ahead.
Hi, I'm Angela Johnson with CTI Consulting and also with Texas Tech University. I wanted to ask a question that's probably more broad here. Having done some work with Device and CDRH over the years, I've noticed there's some really robust sampling methods for working with human factors, usability, that are very small sample size. You could probably extend that even farther to say if you go into ethnographic methods or social science, there are some really broad approaches that allow us to take user perspectives and come up from that into research questions that can be statistically analyzed and adapted. I've noticed that hasn't really permeated this discussion, but has there been any thought, and particularly from the agency, on how you feel about these interdisciplinary methods or bringing in those methods for working with small sample size that have been robust in other areas?
I would say bring it in.
So yes. Okay, last one.
Hi, Dacheng Liu from Boehringer Ingelheim Statistics. So we've been talking about historical control, right? So in the platform trial setting, you can continue the enrollment of your control group, right? So let's say I'm done with my comparison, but then a year later I accumulate more data. Let's say at the first time of my comparison, the treatment effect turned out to be marginal. But I'm getting more data a year down the road. Do I have a second shot?
You look like you want to answer there, John. And on that note, we can.
All right, it takes John to get up.
The only suggestion I have is if that were part of a pre-specified adaptive design plan, that might be
that's still possible.
Yeah.
Thank you.
Yeah, pre-specified does seem to be a recurrent theme here, but I think the reality is that you find out things later, and And as I think you all have talked about, I think it's worth at least some discussion with FDA about the specific circumstances too. But pre-specification is clearly helpful. Okay, I want to thank the people up here on stage with me, but especially all of you here and online for making this a very interactive session and bringing up some extremely important and relevant points that have added a lot of richness to discussion. So thank you all very much. And we are now going to take a short break till 3:30. Going to start promptly then. We have actually our powerhouse finale session coming up that is going to bring in some perspectives on key themes and takeaways. So I hope you'll all join us for that. Thank you all very much.
sooner the better.
And I'll invite our panelists on the last session to join me up here on the stage. Okay. That was great. You just lean into it. Okay, welcome back. In this final session, we're going to discuss some of the forward-looking highlights from stakeholders from FDA, NIH, industry, and patient advocacy, who will discuss some priority areas to consider for advancing treatment development in rare disease settings. Panelists should feel free to pick among the ideas discussed earlier in the day or pose new thoughts, whatever's on your mind, for us to consider. Following brief remarks from each of the speakers, we'll conclude with a moderated discussion between the panelists. And I want to thank you all for sticking with us throughout the day in this what seems to be an increasingly warm room. So thanks to all of you for sticking it out. I'll go ahead and introduce our speakers in the order that I'll ask for your comments. First, Julie Bites is Director of the Office of Drug Evaluation 3 at CDER at FDA. Abby Bronson, Senior Vice President of Research Strategy, Parent Project Muscular Dystrophy. Aloka Chakravarthy, Acting Director of the Office of Biostatistics at CDER and FDA. I'm not going in the right order, actually. Sorry for that. So I'll go to, third would be Assam Zaneh, Director, Office of Clinical Pharmacology, FDA. Jerry Schindler, Adjunct Professor, Biostatistics, Harvard T.H. Chan School of Public Health at Harvard University. Telba Irony, Deputy Director of the Office of Biostatistics and Epidemiology at CBER at FDA. Then Aloka Chakravarthy, Acting Director of the Office of Biostatistics, CDER, FDA. Ellen Werner, Senior Science Officer in Data Science for Blood Therapeutics Branch, Division of Blood Diseases and Blood Resources at NHLBI at the NIH. Lisa Lavange, as you've heard from before, Professor and Associate Chair, Director of Collaborative Studies, Coordinating Center and Department of Biostatistics at UNC. Cartier Esham, Executive Vice President for Emerging Companies, Biotechnology Innovation Organization. And then wrapping us up, Rich Mashicki, Chief Medical Officer and Executive Vice President at Pharma. So now I'll go ahead and start with Julie. And we have so many, I don't get to sit down, but that's okay.
Good afternoon. I'm something of a history buff, so I'm going to start off with a quote from General George Patton. And the general once said, "Never tell people how to do things. Tell them what to do, and they will surprise you with their ingenuity." So today, the what for us is getting new drugs on the market. So what needs to be done to get new drugs on the market? First, I would say we need to develop ways to better capture the patient experience. Patient input can inform the selection of clinically meaningful efficacy and safety outcomes, and that we can use in our clinical trials. Patient input can inform trial designs by tailoring schedules for clinic visits, numbers and types of procedures to be performed. These are trial design features that are particularly important to ensure participation and retention of patients in the trials we design. Patient input can also inform our understanding of the level of benefit patients would require in order to accept a certain level of risk associated with a new treatment. The second thing we need to do, I would say, is to facilitate the advancement and use of novel, adaptive, Bayesian, and other clinical trial designs, such as master protocols. We need to develop case studies where these designs are used that can be shared and vetted both here and globally. Today, we heard a great deal about the use of prior information on treatment effects and use of data from external control patients from registries, from natural history studies, or from earlier phase trials. We need to define when it is acceptable to use these methods and how they will inform decisions on drug approvability. And lastly, I would say that the third thing we would need to do is to write informative drug labels that accurately communicate the benefits of a drug treatment for healthcare providers who are very busy at times and who are trying to decide whether a drug will work for their patients and how well it will work for them. When the trial objective is straightforward, such as showing a survival benefit, and the results are unequivocal, it is fairly easy to write a label to convey this information. However, it's often not that easy. So when the trial objective is to show a delay in disease progression based on patient function, not only is it more challenging to design the trial and compare the results relative to what would have happened without treatment, it is also more challenging to explain the results in product labeling. So I'm going to stop there and let others contribute.
Okay, thanks. Abby?
Good afternoon, everybody. First of all, I want to thank the organizers for this meeting. It was really informative and exciting, and we just had our patient-focused drug development meeting last week in DC, and we heard a lot of patients came together and talked about things that matter to them. We heard about how overall things just don't move quickly enough. There's unequal access to trials in terms of age and ambulatory status. We hear they know that there's going to be a combination approach needed, but where's the method? Where's the way of studying combinations? We hear that they look around and they say one patient responds to one drug one way, but another boy responds another way. How are we actually trying to study that? So there's a lot of things that we covered today that I think are really going to meet the needs of our patient population. So I think I was asked here today because you heard Scott say that we are trying to facilitate and start a master protocol in Duchenne. We can hear all these statisticians talk about the statistics, and that is great, but then you get this protocol and you have to execute it in the community. So a couple things. First of all, we think that as we look, and I've been working with what we call our design team, which is a group of sponsors who volunteered to help us we first called it the exploratory team, to sort of look at it from the sponsor perspective and say, what are the challenges here? Why would we want to do this? What are the barriers? So that was sort of the first step. It was a very nascent opportunity, and it was very lonely out there. There are sometimes I was on a call with Scott and maybe one other person. So to see, now, Deborah, thanks for your comment and your interest. We're really glad that this has turned into something. So in our world, it's pretty congested out there. You'll talk to a PI, and he'll say, I have 11 trials running. I cannot take on any more trials. We hear research coordinators say, I can't keep track of the order that the assessments are done in a protocol. We need some standardization. So we think that this master protocol will help lend some order to what's going on at the site level. So we also hope that with this clear regulatory endpoint, and if we can come and as a group decide on the endpoints that are approvable, that will also make Duchenne a very attractive target for perhaps new entrants and maybe some sponsors who might have a product that will work, but they're worried about the cost of entry, so they don't come into the space. So that's another benefit that we're really looking to capitalize on. So the other, and I think, Deborah, you brought up this point about one of the biggest hurdles that we heard at the beginning was that, well, in our space, parents are very educated and very motivated, outgoing, and they study the trials. There's multiple clinical trial finder tools out there, and they choose the trial that they think is best for their child, and they go to their clinician, they say, this is what I want. And sometimes we've heard stories of where they save their patient, their child, for this one particular trial. And so there are those patients out there and those parents out there, but I think for every one of those, a patient audience is a big group of people, and there's others that are overwhelmed with the burden of caring for their child. They research as much as they can. We had a burdens of clinical trial participation meeting last year, and we heard that it was just overwhelming sometimes for parents to make the decision. And then once they make the decision and their child is in a trial, living with that decision, did I make the right decision? What happens if something else is coming along? Was that the right decision? I think that you just don't know. You just don't know. And I think Malcolm Gladwell said once that, do more choices always lead to more happiness? And maybe that's not the case here. So we've done, but we did want to allay the sponsors' concerns about that. So we did do a couple informal focus groups just to sort of get the lay of the land. And when you work with patients and you start talking to them and you explain the concept of this master protocol, and then you sit back and you let them talk amongst themselves, they realize that they are giving up choice. But then they also realize that they might, for themselves and for everybody, for the whole community, get to the answers faster. So I've seen, we've done a couple of these, and I've seen them be very open and receptive. And it is going to take some education. In fact, our design group, our sort of core group working on this, we've just put a team together to look at communication plan for going out to all the different stakeholders that will be, have to bring under the tent, so theoretically, to make this happen. So our success going forward, I think it rests on two things. And the first is, and I think it's been talked a lot about today, is access to data. That you need that data to be able to design these trials. Those trials, this master protocol will generate new data, which will be very helpful. But to get us going, we need access to data. And unfortunately, our community is siloed. And we are working very hard to sort of make everyone realize that they're part of this, and we need to share the data. So that's one thing. And we need the data in a timely manner. And the second thing, and I think I mentioned it, is stakeholder engagement. We're basically asking every stakeholder to do something differently. They have to change behavior, and that's hard to do. And that's a paradigm shift. So we're really working on looking at what the value proposition is for each of those stakeholders and helping them understand what they are going to get for what they have to give up. So it's not a one-shot process. It's going to be multiple conversations. And as I said, we have this group looking at that. So I think that's going to be, those are the two key things for us going forward to make this a success. And so thanks again to everybody.
Great, thank you. Sam?
I wanted to echo my thanks for organizing this and inviting me to think about some of these issues. I was mentioning to Gary yesterday earlier today, a quote that Lou Scheiner used to say, which is, it's at the intersection of statistics and mechanism that you get true insight onto, for example, how a drug works. So I'm going to try to marry the two a little bit in my comments. One thing that I've been thinking about quite a bit since the invitation is the concept of borrowing. And then it became very apparent that we borrow information all the time to make drug development and regulatory decisions in terms of taking information from one context to another. Establishing maximum recommended starting doses for first-in-human trials. We mentioned extrapolation from the adult to the pediatric context in certain situations, extrapolation of efficacy from one stage of disease to another. Lissa mentioned anti-infective drug development. I'd also add development of medical countermeasures under the animal rule, development of generic small molecules and biosimilars, derivation of alternate doses and regimens, oftentimes untested from the empirical extant clinical trial database. And the newest kid on the block, I think that's of relevance here, is evaluation of targeted therapies in low-frequency subsets of disease. And we have a new guidance on that. And so some of these exercises are algorithmic, some are structured, many of them are kind of informal, and there's a gestalt to it. And I think we can think a little bit about how to bring more formality to some of these exercises. In terms of paradigms for rare disease drug development, one paradigm that's worked fairly well has been demonstrating safety and efficacy in a fairly homogeneous population, for example, with a single genetic mutation that's the driver of the disease, using parallel group study designs, typical study designs, but then extending that with follow-on studies in a mutationally diverse population that has a functional similarity or molecular similarity with the original population. This has essentially been the cystic fibrosis paradigm, and it's worked well in that area. Of course, our thinking is evolving in this area. As I mentioned at the tail end of last year, there's been a guidance on developing targeted therapies for rare subsets of disease, and it tackles a variety of complicated problems like cohorting patients for enrollment into clinical trials, data analysis and generalizability of the findings, labeling, which is exceptionally challenging, and then the use of real-world evidence and other data sources to refine our understanding of risk-benefit after the initial approval. That guidance is light on statistical principles, but there is an accompanying paper in CPT that came out this month, Clinical Pharmacology and Therapeutics, that recognizes the impracticality of estimating a treatment effect in a subtype from results in that subtype alone. It encourages consideration of methods that involve borrowing information from other subtypes. It calls out empirical Bayes methods, and it leaves open the use of real-world evidence for refining post-approval. We're also, someone mentioned N of 1 studies. We are seeing those. So for drugs with, let's say, amenable features and diseases with amenable features, good safety profile, short half-life, quick on-off, we have seen these N of 1 studies with Bayesian hierarchical modeling. There's an extensive literature on randomized comparative approaches that could be useful in the rare disease context. There was a nice review of these in 2013 by Catherine Cornu, this is a modeling group out of Europe, and they categorize or describe the trade-offs in these designs and developed a choice algorithm based on what you know about the disease and the outcomes. Now, we haven't applied those formally at the FDA, although we've used many of these, whether they be N of 1, randomized withdrawal, a variety of others. And one thing I think would be very interesting is to essentially catalog our uses of all of these alternative designs and see if we could come up with a choice framework for trial designs in rare diseases as a starting point. I just want to end on what I want to highlight as sort of needs or critical features of success for some of these programs that we've seen in the past. The first is deep mechanistic understanding. So whether it be molecular, genetic, disease biology, pathophysiology, natural history, this makes it certainly much easier to have discussions about assumptions, leveraging translational models, even maybe quantitative drug disease trial frameworks. Also strong understanding of the mechanism of action of the drug or mechanism of benefit of the drug. In cases where we've had extensive knowledge about the compound, this has been, again, easier to have conversations about what those trials might look like. The third ingredient is extensive discussions between drug developers and regulatory health agencies throughout the drug development program. I don't think this could be emphasized too much. Fourthly, teams of very strong people, very smart people working on complicated scientific and regulatory issues, and some of that requires some education, and I think we heard that in the first panel today. Ultimately, a formal synthesis framework would be nice. I think going back to the first speaker, this is all about a framework for synthesis, and now we're trying to formalize that a bit, and I think these are the kinds of discussions that would catalyze that.
Great, thanks, Issam. Jerry?
Sure. I'd also like to thank everyone for organizing this meeting today. I think we covered a lot of really interesting topics and really important topics, and we took them as sort of discrete pieces. And what I'd like to do is maybe break them apart a little bit and view them from my perspective as a drug developer and try to understand what does it mean to me and how do we look at this in terms of drug development. And one issue that came across in multiple sessions that I think is important, and that is if you look at what we heard about natural history studies, that was often done by the patient groups and controlled by the patient groups with some support, financial support maybe, from some of the other groups from industry that produces the products. And so I think that focusing on the patient groups as sort of the center of this drug development process, I think would be very important for the development of products for rare diseases. And if you look at the use of registries and natural history studies before you have a treatment, that's very similar to observational studies post-marketing. To me, they're almost the same thing. They're different versions of kind of the same thing. And the obvious place for the patient groups is that they could participate in both and essentially own the data for both. And if they own the data pre-product approval and post-marketing approval, why not own the middle piece as well? So because the data comes from the patients and the industry sources, who have the products test their products in the patients, but the data are really derived from the patients. So it seems to me that there's an opportunity for the patients to own the data of the whole process, and they shouldn't have to go to industry to ask for access to the data, but it should be the other way around. Industry should ask for access to the patients' data from the patients. So if they own that process, then you really have the patient groups that are driving the process. And then some of the pieces that we've heard today really fall into place really nicely. So the first piece, and maybe I'll go out of order a little bit, but the first piece is this whole trial network, where the trial network and the registries are kind of the same thing. So why not make that all the same thing, where you have a trial network that is responsible for putting together the registries, and a trial network that's responsible for putting together post-marketing studies, and a trial network that's responsible for putting together the phase 3, phase 2, and phase 3 studies of the product. And when you're doing that, you also ensure that you have harmonization. Because one of the critical pieces of the master protocol, it seems to me, even though it was wasn't specifically called out in the discussion, I think is the harmonization of patient population definitions, of endpoints, of the time that the endpoint is measured. So you have all of the data collected in the same way. That's obviously part of it. If it's all the same protocol, it's all going to be the same. But that's the critical piece, that you have the harmonization there. And if the patient groups are driving that, then you don't have different companies that are using different definitions along the way. So using the same definitions for the endpoints, and the same definitions for the differential patient populations, and the same time points to do the measurements, I think then allows you for the patient and the physicians who are treating these patients to compare products post-marketing. So assuming multiple products get approved in the same patient population, the obvious question is, which one is the right one for me? And if they're all measured on different endpoints and different time points, it's hard to compare. But if they're all the same, which is what the master protocol gives you, then you have that benefit. So to me, that's one of the biggest benefits of the master protocol, is the harmonization of the process, which allows you to do some comparison across the products. So to me, that's an important thing, and the patients should be driving that rather than the pharma companies, it seems to me. The earlier topic that we discussed about borrowing makes a lot of sense, but the one area that wasn't really called out is when you're doing a study, sometimes you have no choice because, and it was mentioned earlier, that sometimes it's impractical to collect enough data to use either a frequentist approach or a Bayesian approach with a lot of data. So you want to borrow from other sources that might be similar enough to borrow from. So if you have no choice, that makes a lot of sense. But sometimes you do have a choice. And then the question is, do you have a study that might accrue patients over the next year or two, or do you try to accelerate the process? And I think there, it's not quite so clear-cut. And the question that was brought up earlier about the inflation of the type 1 error, I think becomes a more serious question in that setting, where you could have done a larger study, because it was possible, but you chose not to. You might want to consider not doing it. And there, I'm not really sure what the answer is. But if it's in the situation where you have no choice because the patients aren't there, they're not available, then it totally makes a lot of sense. The other place where it makes a lot of sense, it was casually mentioned a little bit just now, is the synthesis of the data across the entire program. So what you want to do, I think, and the Bayesian approach makes a lot of sense to me, is to look across the entire program, to look at the very beginning of the data, once you start collecting data in a particular product, and look at it all the way through phase 2, if there's a dose ranging portion of the trial, look at the confirmatory data, and potentially even look at post-marketing. And you can do that very nicely with the Bayesian approach. And it seems to me that the synthesis of the data across all stages of drug development makes a lot of sense. Another area that wasn't mentioned a lot today was the concept of missing data. So if you're doing a very small study, where every patient is valuable, you don't want to have a lot of missing data. You really don't want any missing data. So there should be a lot of effort to make sure that missing data is minimized or reduced as much as possible. There's also this issue about differential missingness, where you have more missing data in the control group, more missing data in the treated group than the control group. So you might not have the same, so you might really have missing at random. And if it's a small study, it seems to me that that's a big issue. It wasn't specifically called out because this isn't totally talking about some of the statistical issues, but I think trying to make sure that you have as little missing data or no missing data over the course of the trial would be important. So I think I'll stop there.
Okay, great. Thank you. Telba?
Okay, I will try to summarize what was discussed here, bringing first the issue of uncertainty. So uncertainty is a big thing in rare diseases, and I think we need to learn how to live with it, because in many cases we won't get more certainty or less variability just because we don't have the large sample sizes. As Laura Lee said in the beginning, first thing you have to think about is reuse data, use different designs, and leverage information. But, using the Bayesian approach in which we actually are not weakening the information or the evidence when you use a prior, but actually we are strengthening it in many cases. I said before, if you have some information and you are using, you have to consider you are not using it. You have to consider that it may be biasing against, effectiveness of some treatment. Another thing that we have to consider here is how do we balance benefits and risks. And we, as I said before, we have to learn how to live with less certainty about the benefits. And when we balance benefits and risks, and if we consider the risk of not being treated, we will actually tolerate more uncertainty on the benefits, because we know if we are not being treated, particularly if we're talking about a disease that's life-threatening or permanently debilitating a patient, is a very serious risk. So we might want to consider more uncertainty on the benefit. And a way to do that is through the Bayesian approach and Bayesian posterior probabilities, in which we will set thresholds for these posterior probabilities that are not necessarily very high thresholds. If you have a life-threatening disease, you might consider a good treatment, something that will give you 80% probability of survival, sometimes even 60% if you know you're going to die otherwise. So these things have to be considered. The risk of not being treated is big in the rare disease for which there is an unmet medical need, and sometimes we fail to consider that. Another piece of information that's very important in this benefit-risk consideration is patient input. Sometimes patients will tolerate more risk or less evidence on benefit in exchange of a probable benefit. So patients have to be consulted for that, and being in the Center for Devices, before we were used to consult patients. Of course, patients have to give scientific information, and there is a way to get scientific information of preferences. There is a guidance that's signed by both CBER and CDRH on how to get patient information on preferences of benefits and risks in a scientific way. Another thing that I wanted to talk about is the master protocols and the other way to conduct, the way that an advantage of a master protocols that I think is fundamental is that during that clinical trial, you were treating the patients in the trial in the best possible way. So I think that's a very big advantage of these protocols. And I think the patient advocacy groups have a big role to play to incentivize sponsors to participate in these master protocols. Perhaps the FDA could also think about ways to provide incentives to sponsors to participate in these master protocols. I think this is something that needs to be addressed and it needs to be talked about. That's my comments. Thank you.
Okay, thank you. Aloka?
So as you know that a lot of very seminal work and thought had gone into this space when Lisa started putting together the group to work on this. So as I came into it, my biggest charge in this area was to keep the momentum up and to encourage any possibility of collaboration, of being flexible, and to keep the different thoughts on this at the highest scientific level. So one of the things that I have been listening very carefully and getting very impressed by is the collaboration, and collaboration on various fronts. First of all, collaboration of statisticians, clinicians, patient groups, and the patients themselves. That's very important because unless we all put our thoughts together, this area will not get the optimum amount of information. The second thing is collaboration of data. And many times, the failed trials in these spaces do not get reported or do not get shared. And many times that can be a gold mine of getting the information on the patient characteristic, on the endpoint, on the follow-up, on the mechanism of action, and all of that. So we need to put our heads together to see how to bring that up in a very clear, collaborative, non-competitive area. The second thing on collaboration is the data standards. Data is sparse in this area, and we need to recognize that and be very open and be very comfortable with the amount of data that we put forth, that it is of the highest quality, so that this small, sparse data can form the basis of our regulatory decision. And there has to be a certain amount of flexibility also to look at this data in a tiered fashion. What are the absolute important things that we need to know pre-market? And what are the things that we can learn in a tiered fashion in post-market? So those kind of things, we need to look at. The consortium or the collaborative pathways are going to be the way to go because, on your own, you may not be able to have all the information that is needed for fully quantifying the risk and the benefit. So, this consortium has to be also international, because what is rare in US may not be as rare ex-US, and you have to go where the incidence is. This brings to front, some of the things that we talk about in multi-regional clinical trials. Just like you're not able to quantify what the level of efficacy would be in a traditional pattern, you'll not be able to look at the regional consistencies or you're not going to be able to have the standard traditional thoughts on allocation to regions. So these things really need to be flexible to bring them together. And the last thing that I would mention is the use of real-world evidence. We have heard about the platform trials, we have heard about natural history. Many of these may not come in fashion of RCTs, and we should be able to garner as much information as we can from the observational studies of various flavors, as long as we have the idea that these are of highest quality, that they can be used in a meaningful way. So with that, I'll stop.
Great, thanks. Great comments. Ellen?
Okay, hi. In the interest of full disclosure, I do hold an IND in pediatric sickle cell anemia that is in response to an FDA written request, but I will not be discussing that today at all. So at NIH, we fund research. We do not participate in the regulatory decision-making at all. We depend on our partners at FDA to do that. And with that in mind, I'd like to focus on our charge today to come up with some solutions. And I really identified 3 themes today, and I apologize to Jerry and Aloka. I'm going to be repeating some of what you said, but it's from the NIH perspective. The first theme that I saw was optimal planning. What are going to be your strategies for drug development in rare diseases? The requirement to really build partnerships and infrastructure is the second theme. And third, assuring data integrity, which in the NIH lingo includes reliability, validity, completeness, and the emerging technologies for collecting, standardizing, and harmonizing data. And I'll speak a little bit more about those methods. Now, optimal planning really includes both short and long-term and everything in between. With NIH funding, please do not wait for requests for applications, because what we do mostly is fund investigator-initiated research where you all, in collaboration with your partners, identify those gaps in knowledge that are needed to move the field forward. So in your partnerships, you identify the clinical chemistry, the biomarkers, the early phase studies on through that spectrum of research to post-marketing, although we don't call it post-marketing, we might call it cohort studies. You identify those areas that need further research and at the same time allow for back translation. So in your observational studies, if you discover that you need to get back to the lab or your partners need to get back to the lab, that's the kind of research that we at NIH do like to fund. So we do look for proof of principles and incremental research. And I have permission from Claudia Morris to cite what she has done. Claudia asked a question at the first panel this morning. She's an ED doc working in pediatric sickle cell anemia on arginine therapy. And in fact, she started this 20 years ago. The first time I met her, I was her program director in her K Award. That's a Career Development Award. It's a small salary-only research grant. But she built the level of evidence where now she is completing a phase 2 trial, and what the future holds will be determined. But she's got a body of evidence now working not only individually on her K23 with a small group of collaborators and mentors, now working with PECARN, the Pediatric Emergency Care Applied Research Network, with a national presence. So this is the kind of planning that I'm going to encourage you to do, where it's not just that 1 5-year grant. At NIH recently, we have changed how we conduct programs in clinical trials. We now want single IRBs, and we allow for longer-term grants. We allow for pilots, feasibility studies, and up to 7 years of funding in cooperative agreements that include a planning phase. But that planning phase should not say, oh, we're going to talk to the FDA, or we're going to develop a protocol. The planning phase is already built on your foundation, where again you're incrementally building on what you've done before, where you're going to be finalizing protocols during that planning phase. You're going to be possibly validating patient-reported outcomes, which I'll discuss in a minute, for your disease-specific population. You might be finalizing some biomarker validation studies. And then the remaining 5 years enables you to conduct that full phase 3 trial. That also I should include in that planning phase, you need to be very mindful of all the regulatory requirements, not just for FDA, but the human subjects, the IRBs, the informed consent, and subcontracts. Are they in place for your clinical sites? Now, I mentioned partnerships and the need to collaborate and build your infrastructure. One advantage I have found in rare diseases is that often our collaborators are already known to us. If you take cardiology, there are so many people out there doing the studies that it's very hard to network. But we network with our collaborators, and that actually makes linkages of INDs a lot easier because you know who's doing what, right? And again, we've heard today the importance of the patient advocacy community. It's not just the academic researchers anymore who are driving this, but the NIH has, since 2004, invested a lot of money in patient-reported outcomes, which the FDA calls clinical outcomes assessments, PROs versus COAs, but we're talking about drug development tools. And for NIH, we've developed some generic tools. FDA wants them disease-specific, validated in the population. We say, fine, thank you, FDA. We're not going to tell you how to conduct business. But this is important when you come in with an NIH application that you show that the FDA will be accepting, or you're in the process of communicating with the FDA about this drug development tool, whether it's biomarkers or, again, the PROs. Now, what's important about the PROs, they were purposely developed for the express purpose of putting the patient voice into research. And so there is a qualitative research phase that includes focus groups, key informant interviews, and the analysis of that qualitative data, by the way, is not soft science. It's psychometrics, which is a very specialized field. And it then leads to these quantitative assessment tools, whereby you get the full spectrum of severity. So in the SF-36, you could have had floor and ceiling effects. You get a truncated view of how seriously that patient is affected. With these PROs, because of computer adaptive testing and the latest in item response theory, not soft science, right? You get a better picture of the severity of how patients are affected. Finally, that really brings us to the third theme that I identified, the data integrity issues. And we heard today FDA might be looking for CDISC. At NIH, I can tell you, you come in with an application, you say, we've worked with the FDA, we're going to be doing mapping to CDISC. Our study section is not going to say, oh, no, we want you to do this. No, you have the orders from the FDA about what they want to get to that regulatory decision-making. But again, you need to document that you've had these ongoing relationships and interactions with your partners and the FDA. Now, there's also something very important, and Jerry mentioned harmonization. Pre-data collection standardization using common data elements and data standards, established codes, that's wonderful. But what are you going to do with extant databases, whereby, for example, in a natural history study, you have to harmonize data? That's going to be a challenge for your informaticians on your team. And you want to be able to say something about apples and apples, not in general about fruit. So that's an emerging issue that the data science and informatics field will need to further develop, but stay on top of it so that any kind of study section will have confidence in the data that you'll be generating. And finally, I do want to thank the FDA. I've taken advantage of many of these workshops and trainings, And one that I attended within the last year included a patient advocacy rep, a mom, who talked about collaborating finally with academic researchers. And at the end of the day, after the academic researchers had been working with the patient advocacy group, they generated data, and the academic researchers said, "Oh, look what we found." And the mom said, "Yeah, we told you that. We know that." So that's, I think, a take-home message for all of that. And I'll end.
Great, thanks for all of those comments. Lissa?
What else can I possibly have to say? All right, so we were asked to come up with takeaway messages. So I thought of 4 takeaway messages briefly. The first one is that this is a hard problem and there's no single best approach. But I think if you heard anything today, you should have heard that the FDA is open for business, open for innovation, and don't assume that something won't fly. Come and talk. And I think that you've heard that over and over again. And that was really the purpose for going to this public meeting after the many years of lead up to it that have been described. The second is, I would say that you should have heard that it takes a village. If you look at the DN2 as an example, we had an in-depth presentation there, but there are many others as well. You have academics working on this, Randall at WashU. You have the FDA working on this. Billy Dunn wasn't here today. Some of my staff worked on this. You have patient advocacy groups. You have industry statisticians and clinicians, and at least multiple sponsors. I'm not quite sure how many. These are big one way to have a solution is a big enterprise, but this collaboration can be quite effective, and you can make sure that all of the important voices are heard in those collaborative ways. The third, these are complex issues. We started with borrowing and Bayesian methods, and we talked about bias from external data sources and so forth, data quality and electronic health records. But we also have a lot of really smart people working on these topics, and many of them are here today. We heard about all that the CF folks are doing, building registries, great role model, I think, to plan from. We heard from industry statisticians working on very complex Bayesian problems, methods that they use internally for their own decision making and are willing to share with regulators and others to think about other types of decision making. We've got lots of smart people around us, and I think that there's no shortage of people to come up with these ideas, so I think that's very encouraging. And then the fourth is that we need another public meeting on endpoints in rare diseases. So we did take note of that.
Great. Thanks, Lissa. So next, we'll go to Cartier.
Thank you very much. And again, it's always a little daunting to be towards the end of all these excellent comments. So I'm going to do my best. For those not familiar with the Biotechnology Innovation Organization, we're a policy and advocacy organization located here in DC, but represent biotechnology companies across the globe, ranging from the entrepreneurial, those that don't yet have a product on the market yet, to the multinational companies. So just so you understand the perspective of which I'm bringing my comments. And so I thought I'd share sort of briefly share three buckets of comments. One sort of focusing on the reflections I had coming into this meeting. The second being, what did I garner from this meeting about things that I need to take back to my membership and our companies for further discussion. And three, how can we look at this in terms of developing actionable items from a policy point of view? So again, this is clearly an assembly of the best and brightest working on every time I come to one of these, I'm so impressed with everybody that is working on issues, identifying new issues, and promoting better ways to develop innovative medicines to treat rare diseases. I think every time I come to these, you get a better understanding of how we're all trying to appreciate the unique issues that come with developing drugs to treat rare diseases. And within that, the unique aspects of each rare disease itself. And so, every time I come here, I feel like I learn a little bit more about how complex this is, at the same time feel inspired about the solutions being brought forward to address those issues. I also, when I was coming here, I was reflecting upon, we had recently, it was Rare Disease Awareness Week, and I think appropriately, there is a lot of recognition presently about the tremendous scientific advances that have been made in diagnosing and identifying new rare diseases and developing treatments for those diseases. We've celebrated, record-breaking numbers of FDA approvals. But I think as we learned today, this is also, that is terrific, and we want to keep marching towards that, keep that trend going. But this is also clearly a golden age of learning, because with each approval, we had many failures. And somebody brought up electronic health records and sort of, that's taking a little bit longer. Maybe we haven't fully realized the potential that information from electronic health records could bring based on conversations we had 10 to 12 years ago. So how can we learn from those lessons and think about how we can do this better? And so I think this is requiring, this goes into the second bucket, is what do I want to take back from this meeting and create further discussions with my membership to develop consensus thinking around and or support efforts that are ongoing to better enable utilization of modern and maybe perhaps more effective approaches to drug development. So in that sense, I think we heard a lot of great comments that are both looking back as well as looking forward. So we also recently in Washington went through a whole exercise where we have 21st century cures. There's a lot of tools now available, particularly to the FDA, to promote modern approaches such as innovative clinical trial designs. We had a lot of conversations about adaptive and Bayesian. We're looking at master protocols, et cetera. And I think that from my perspective, I want to have discussions with our membership to make sure that there's nothing in the way of getting those, the ability to utilize those. And also working with FDA, NIH, and others to think about how do we share those learnings? How do we promote learnings, from one approval and turn that into information, one approval or one failure and turn that into information that will help the next 10 drugs in clinical development be effective in their development and approval journey. I think we also heard a lot about data, and I heard this, I think, in a lot of different ways. One, in looking about what is the state of data, and not all data, historical data can be treated equally. So what do we learn from that? How do we, what can we do to make sure that the data that exists is being utilized with maximum efficiency and reaching its full potential? But also, how do we look at data that maybe was collected or in a way that is not that helpful and ensure that we prevent against replication of those mistakes moving forward? And so I think that's something I want to explore a little bit further. The other, I'll end on this sort of reflection. As a policy person in Washington, D.C., we're always sort of pushing FDA to give us more clarity, give us greater understanding. We want, how can we ensure that there's consistent adoption of best practices across review divisions and therapeutic areas, and that's something that we still want to do. This is also an area, though, that we want to make sure that whatever we do on a policy standpoint ensures flexibility, because as Liz and others have said, there is no one-size-fits-all for the variety of needs, for the variety of rare diseases affecting millions of patients across the world. And so I think we need to find that right balance to make sure, again, we're leveraging learnings, understanding what flexibility means in this space, but removing any barriers to, again, enable approaches that make the most sense. Because the other thing that we heard about today, and I think a golden age of learning that we are now in the middle of, and that is a much better understanding of patient needs and the importance of that voice in drug development and review processes. So again, that gets to, that can mean a lot of things. It gets to understanding the needs of trying to run clinical trials for small patient populations across the world. It can mean how do we better understand the needs of patients within a rare disease that have different needs based on severity and progression of disease. So how do we address, how do we meet statistical standards and approval requirements, but at the same time make sure that the development process is reflective of those different needs. And I think many people have said it best, it does take a village. It is clear that engagements like this are going to be critical, and BIO is committed to continue working and participating in these conversations. And again, with an eye towards moving towards action that we can take to probably, my sense is mostly get out of the way and make sure that there's nothing getting in your way to bring forward some of these ideas into practice on a more regular basis. So with that, I'll end my comments and turn to my colleague, Rich.
Thanks, Rich.
So being last, right, I guess, I'll try to be brief. I promise I won't take more than an hour.
Okay. It keeps getting warm in here, so.
I heard you. So I'll try to make it briefer than that. You know, in full disclosure, I wasn't here all day. So, but I have participated in drug development for rare diseases for most of my career. Previously in a biopharmaceutical company, then I've spent and was lucky to spend 4.5 years at FDA just before I came here, where throughout all of this has been quite a passion for me. And so while at FDA, I had the opportunity to spend a fair amount of time in previous meetings leading up to today. So I think I'm pretty familiar both with the speakers and with much of the material that we brought forward. So my most important comment is the importance of what this is. I mean, we're potentially here with a landmark opportunity in front of us to really do something very important for this entire field. A change has been long needed in how we approach and analyze these things. And the necessity, with such small sample sizes now available to us, the necessity of having to move beyond our obsession with the p-value, I think is incredibly important to be stated. And so Bayesian approaches are not new. Lissa, when was it? How long have we had Bayesian thinking? Couple centuries. There you go. But here we are in the 21st century, and we're really having a serious discussion about bringing this into a regulatory decision-making. I also want to really thank the FDA, its leadership by having this conference, by much of the work that had been done leading up to its conference, and its openness and willingness to really consider these changes are so important because, yes, we can use them and when we make company decisions, but what we really need is to make sure that they do not become meaningless without regulatory acceptance. So to me, this is a really important step towards achieving that. I also think that I've spent a long time with registries in my prior careers. And so the wedding of Bayesian approaches with registries and natural history studies, I think are absolutely key to getting as much information as we can when the sample sizes are indeed so small. I will make a small editorial comment about master protocols, which I think are a great idea and certainly a wonderful way to try and conserve and get as much information as you can when you have a very small number of people to study. No question about it. I do think there are practical issues in the business model that have to be addressed in order to get companies to really sign on in a really good way to participate in master protocols, and I'll leave it at that for now. I also think that as we think about rare diseases, I often say that they are the tip of the spear. It is clearly an area where we can have some comfort regarding uncertainty, benefit, and risk because not only, well, basically because they are a small number of people that are impacted with the regulatory decisions that are often involved here. Not that that makes them less important, it just gives us a different framework for the thinking. And so as we begin to move forward into the era of personalized or precision medicine, whichever term you think about, we ultimately will come potentially to a point where everything is a rare disease. And so this gives us the wonderful opportunity to pilot this new way of thinking, or a 21st century, even if it's 2 centuries ago, thinking that allows us to incorporate and get experience with that within the regulatory framework before it moves on. At Pharma, where I'm now, we strongly support the implementation of PDUFA, FDARA, and all the other legislative actions of the last year. And we very much look forward to working with FDA to accomplish those. And one of the most important priorities for us among those is innovative clinical trial design. And so this is, again, a wonderful place to begin to move forward and to think about how to do this in the very best way. And then I will say that I think most of the things that have been talked about are really very practical. They really are doable. This isn't blue sky that we're really talking about. So let's do them.
Okay, thanks, Rich. So we have about 10 minutes left on this panel. And judging by all of you, I probably should turn right to audience Q&A because you guys have been pretty participatory. However, I will go ahead and try to summarize. We had 10 people. I didn't hear 10 different things. I heard sort of some common themes, and I'll just briefly summarize those in reverse order. So I heard, FDA, as evidenced by this and lots of ongoing work that FDA is doing, is to quote Lissa, open for business. And very open to sort of innovative ways to do rare disease drug development better in a way that gets to regulatory acceptance. So that was number 6. Number 5, we heard a lot in these comments around the role of Bayesian statistics and Bayesian methods and ways to sort of share best practices in doing that, because it's certainly an important role. 4 was innovative trial designs, including master protocols. I'd really like to hear the suggestions, Rich, from you on what the business model needs to be. So that might be a question for you guys to bring up. So that was number 4, is this master protocol designs, and how do we get investment in that, and what are the best practices for doing those better? Number 3, a lot about data, not just pre-market data collection and these trial infrastructure and how to share data better, but also incorporation in this model of registries and real-world data and real-world evidence, and the role of all of those different data sources to bring more certainty to drug benefit and risk, particularly in rare diseases. Number 2 and number 1, with number 1 being the most important from what I've heard. Number 2 is collaboration is key, not just among stakeholders, but global collaboration. And then number 1, I think what I heard most consistently through the comments is the role of patients in not just bringing their own data to bear in these registries, but actually being a catalyst and a driver to building these collaborations. This is the one area where I hear very strongly the role of patients and actually motivating companies and incentivizing companies to do things differently, and the need for patients to be at the center of all of this. We've seen so many examples throughout today that it's happening, that there are efforts to do that, but yet a lot of questions on how to do that better and how to help patient disease organizations be leaders in this area to help catalyze a lot of the collaborations that need to happen, the data collaborations, and improving the processes. So with that, since I don't have anybody, maybe I'll ask one question to the group, and then I'll turn to the audience. In terms of how do we get patient groups in a spot where they can be leaders and drivers? What can, from your perspective, I'm not going to go through all of you, but what can FDA do to encourage this? What can industry do to help do this? Or maybe it's just the patient groups themselves sort of stepping up, like in the examples that we've seen today. But just quick thoughts on how we can encourage patient groups to be at the center of this. Maybe I'll turn to Jerry, because you really hit that point very solidly.
Well, from my perspective, one thing that the FDA can do is, and I'm not sure legally or if the legislation supports it, but really insist that companies work together and work with the patients. What happens a lot is that a company will try to develop a drug and will work on their own. and develop a drug in sort of in a silo without consulting with other companies, possibly even without consulting with patients. So nowadays there's more effort to get the patient voice in, but the patient voice is a supporting role. And I think really what needs to happen, especially in the rare diseases where the patients are so few in number, that the patients should be at the center of drug development. And if the FDA encourages companies, especially in a rare population where multiple companies are developing multiple products for the same patient population, the FDA should, and this is where I don't know what the legislation is, but the FDA should insist that the companies work together with the patient groups rather than to work individually, so that they can make sure that some of these harmonization activities occur. And if the patient groups don't have enough funding or don't have the right business model, then there's an opportunity to fix that, but not to have each company work independently. Even developing separate endpoints and separate ways of evaluating the data and separate statistical methods. There should be collaboration from the very first study, and it should be put into the clinical development plan for the product. And also, we haven't really talked about it much, but the statistical analysis plan that companies write should be written early in the process and should follow the drug development. Shouldn't be something that's thrown in at the very end, but something that should be thought about very early in the process. And I think that if companies choose not to do it and choose to go independently, then maybe the FDA can say, but wait, there's other companies you should be talking to, and there's other patient groups that you should get involved into the process and make sure that the patient groups are really at the center of the development.
Thanks. Julie and then Abby.
So another thing that patient groups can do is write succinctly and thoughtful white papers and guidances for the agency's consideration. Some groups have already started doing this. We find we're getting some very scholarly materials in. 3 groups involved in atopic dermatitis came together and sent us in a 40-page synthesis of the issues from their perspective. These kinds of efforts are immensely helpful.
So on our case, I think what you said is exactly right. So when we first started working with the sponsors that have been the volunteers in this team, our collaborative interactions with the agency and their willingness to come and help us, and then we go back to them, has been very attractive for the sponsors. And even now our sponsor discussions, they can trust themselves, and we're starting to have really good interactions about what endpoints and what the agency has told them at one time and what they've told them at another time. I mean, we realize that endpoints, that's not their IP. Their assets are their IP. This stuff is the stuff that we should agree on. So, but that collaborative nature with the agency has been really, and that upfront agreement on certain things has been very attractive for sponsors.
Great, thanks. Question in the audience?
Been discussed tremendously, and we all know that there's a race in the industry to get to the market first, right? And if you're the second, then instead of superiority trial, you have to have a non-inferiority trial. Now, in rare space, that means impossibilities sometimes, because now the sample size goes through the roof and all that. And I'm wondering, in terms of incentives, Rich, that you talked about, perhaps some of these issues could be addressed. So if drug A and drug B and drug C from different companies utilize the same control arm under the master protocol, what are the incentives for them to participate and not be in that race for the market, but rather race for cure?
I don't know.
Is that to me?
No. You have
Yeah, I think master protocols require a certain critical mass to start with, right? You have to have, I think somebody else said it, you have to have, at the same time almost, give or take a year or something, you sort of have to have several companies with several products that can engage together in a protocol, a master protocol. Now, these are rare diseases, and it's a pretty rare event that there are several companies interested in that particular rare disease at that particular moment. So that gets complicated in terms of then do you create, and you're right, there is a competition here. You know, in the Orphan Drug Act, if you all have similar molecules, in fact, you might be the winner takes all. And timing is also a critical issue in all of that. And so you also have to give up control, right, to the master who's driving the master protocol generally. So you don't have control of the quality. You may not have control of the endpoint. You may think that your product will be able to show a dramatic difference with a different endpoint, where your product is more aimed at that endpoint rather than the endpoint that the master protocol has focused on. So these are all important elements that sometimes make it tough for companies, and often these are little companies in the rare disease space, you know, more bio than pharma. And for them, this is the gamble of their whole company. And to turn their baby over, you know, to a master protocol is daunting for them. So it has to be made really attractive in order for them to overcome all of those concerns and hurdles. And I think what is attractive is regulatory certainty, or as close to it as you can get, and how it's accepted. There has to be a sense of cost, that the cost differential between trying to go it alone and this is dramatic enough that you're willing to overcome all those other issues and give it a shot.
Also, someone from FDA respond.
Okay. Great. Thanks for your comments. We have time for 2 more questions. Why don't I go to this one here? Thanks.
Thank you. Claudia Morse from Emory. One of the challenges that I experienced as a clinician scientist, sort of navigating FDA regulatory and then trying to find my funding from NIH, is that a lot of the FDA recommendations are really geared towards industry. So I found myself in this really odd space of trying to figure out how to move the science forward and ensure that what I'm doing is the right thing to get this drug approved if it works. So do you guys from FDA have any recommendations for people like me who are in academia trying to make a difference?
Does anybody? It's a very easy question. I don't know what the.
So I would say that, you know, look at some of the innovative pathways. For example, the CPIM. These are the information meetings where you put a package together to say all the things that you know and put your best questions forward. These are non-binding meetings where you get the best of discussions on scientific path forward, what can be done, kind of, and you will also have the appropriate people engage in it. So if you need, you know, the clinician, statistician, clinical pharmacology, all the things that you may need, you know, may come together. So it will give a good idea about what are the things that is needed, and it might be a path that you may want to look into.
Great, thank you. Last question?
So CPIM is a critical part of innovation meetings. Because
Yes.
acronyms are not commonly understood. I'm Frank Weicott from FDA. Just as a suggestion in the discussion in context what Jerry brought up, namely the data ownership by patients, I think FDA can do more in terms of guiding the community what to discuss. Technically, theoretically, patients do co-own the data also in industry-sponsored trials, yet the interpretation thereof is not so clear, and I think we need to make a case for that. In particular, since that would change the whole picture of data access, data quality. So we need essentially health data liberation. Certainly there is value in it, and I think there also can be money made and certainly kickback to patients who contribute that information. But I think that's a critical step we have to make, because in many cases we are hampered. And you know, we can continue in the next 10 years to discuss it, what are the best ways. But FDA sits right on the button, because at the consent form, which we somehow do control, there's an opportunity to ask patients whether they want to contribute that information to the comments. So.
Thank you. Any comments from the? Okay. That brings us to the end of this session. So I'd like to thank our panelists for all of your great comments. Thank you. Thank you very much. And then as the panelists are leaving the stage, I'm going to go ahead and invite Laura Lee Johnson up to the podium to take a few moments to reflect on what you've heard today. We heard you at the very opening of this meeting, so we'd love to hear some of your key takeaways. Thank you.
So I don't have a fancy tablet, so I've got to not drop my laptop while I'm up here. Anyway, I want to thank everybody again for coming, and before I get into our summaries, I also want to thank everybody who's put a lot of work into these meetings. So I have Catherine, Frank, Morgan. I want to wave your hands and the whole Duke-Margolis staff, because I know that while we interact with the two of you, there are many, many others behind you. So Greg, they did a great job. Yeah. Make everyone know. Also to Rich, yes.
Yeah, I'm going to do that again.
To Rich, Jonathan, and Lissa for starting this up. So you just heard two of them, but Jonathan wasn't here today, but please send him our best. And to Lucas, Estelle, and Ye Feng for helping us continue this in CEDAR, and the many, many other people, Aloka and many others who have helped with this. And to Robin Bent, who had the joy of reading Lissa's mind and then my mind, and that's the nice way of saying she did the huge lift and really a large portion of this work. So thank you, Robin. She says no. So what I almost said this morning, and I didn't, which was to try to get a show of hands, kind of who's here from a patient group, you are a patient, otherwise patient perspective. All right, thank you. And any payers? Of course, we don't know who all is online. And people who call themselves sponsors. So the different companies or the CROs working with them, et cetera. All right. Academics, we've seen a few, a good portion. And feds of all flavors and types. And so I think it was really interesting in that public time, we didn't hear so much from patients or payers. But this meeting, we wanted to make sure, again, that everybody was included, because this has been brought up many times. It's really important to have people involved in the beginning, but as we think about many times the many sponsors that come in that are interested in drug programs, there are also a lot of different patient groups, and a couple of people brought that up. And so what is that collaboration that's going on? So summarizing some of our takeaways today, I think Lissa and Greg both did that very well earlier, so I wrote those down. That made my life a little easier too. But I'm going to take a few other thoughts, kind of like when to borrow. So we had a lot of discussion about this. And realistically, we've been borrowing for years. Like, how do you actually power a trial? Well, you have to have some clue from somewhere. If you don't, things usually go haywire. But I mean, we've been using phase 2 trial data for years to try to do this. But now we're thinking about, is it flexible borrowing? Is it fixed borrowing? Are we really talking about how many controls we need? How do you describe this and summarize the results? So ultimately, those clinical folks, we've got to write the labels. How do we actually describe this to patients? What is it that we're summarizing? And something that one of my colleagues sent to me that I think also is very interesting is, let's say you're using Bayesian method, you're thinking about this for your primary endpoints. Do you always do the same thing for your secondary endpoints? Are we going to also be mixing methods inside? Because if you think about it, I may do a logistic regression for one thing, time to event analysis for another. What other types of mixing can we do, and how does that impact our planning for the trial? In our natural history studies, there's a lot of great opportunity, but as many people brought up, there's a lot of quality issues that could come up, but also some quality solutions. And those, again, have been brought up, and how do we, moving forward, have better, stronger registries in natural history studies? How do you capture the treatments, the standard of care, and the fact that standard of care is not that standard, so you really do need to capture the information on a global level. A few people mentioned trying to do that initial cross-sectional study and then use that to design the good longitudinal study. But we also have a little bit on the expiration date of the data. So when you think about the reduce, reuse, recycle, eventually you have to renew and start over again too. So how do you figure that out? And that's where some of those borrowing practices, again, might be coming into play. 21st Century Cures also talks about the real-world evidence, and so think about some of the legislative parts. Mark and others also brought up other issues that have come up and topics that could really be strengthening us moving forward from a legislative standpoint. On platform trials, we talked a lot about the pluses and the minuses, the barriers of getting companies and other groups into the space. What's going to happen when a product gets approved? How are we leveraging this? But as many people pointed out, we got that problem anyway when we're in siloed trials. And so now, in fact, I've got a big group of people working together on this. What we didn't talk as much about was, what about if one international regulator approves it, but the FDA maybe hasn't yet, or FDA has, but EMA hasn't yet? How are we going to handle that issue? But again, now I've got multiple groups together thinking about it. And I think Ebola is actually, they didn't talk that much about that example, but I think that's also a good example of, okay, we've got a platform. We have now set up a way, and they've done off of this observational trials, a lot of good epi studies to move forward, but then they're kind of ready, because unfortunately there's probably a reason they've got to be ready for the future. So these are things to think about of how we're leveraging, but then of course what also came up many times is who's going to pay. But I think first and foremost, what I keep hearing again and again is also worry about the designs and the endpoints first. Try to think about what we need. I had tried very carefully to avoid the word multiplicity, type 1 error, p-values, and bias. The concepts aren't wrong, though. And I think that we always heard, like, the reason there is a concern is because we're concerned if the evidence isn't really telling us the truth. How much evidence is really evidence? So those focuses, though, on study rigor and on the evidence, really important. Talking to patients and caregivers to inform the choices. Kind of like what's really at risk. And it's very important and it matters a lot to say, I've gone and talked to all these different patient groups. Sometimes we call them in to FDA to talk to us. Find out kind of, okay, what matters, what's important. Or, as Julie mentioned also, saying like, here's our summary, here are the methods of how we collected this information, and we're going to give this information to you. The endpoints, what's measured, a lot of people brought this up. Responder definitions typically aren't based on really strong information. Like, unless they're completely cleared the disease, okay, we know what a responder is then. But many times people try to come up with kind of trivial mathematical way of defining it, but it may not really be a definition of response. And plus, there can be a lot of other mathematical and other reasons that may not be the best endpoint for your trial. So we have to think about what matters, what evidence do patients care about, and what changes do they want to see? And again, involve that as we're thinking about planning all of our studies and trials. And that, as Petra and others brought up, may not be in full alignment with what the clinicians and other folks have talked about for their registries and their natural history studies, et cetera. So in order to have the information necessary at the end of the day, involve all of the folks early on. It also may not be easy to get some of this information from the medical record, but again, if clinicians work this into, this is what I collect because my patients and we as a collective have decided we care about it and we want to track it, that information is then available later on. So there is, again, a large group consortia together. So what we're doing may not be new, but it may be vital. There are a lot of different ways to access the agency. It might be a pre-IND or that variant thereof, depending on the center you're going to. PACE, CPIM, there are a lot of ways to come and talk to FDA. And it's very important. And I also don't want to underestimate some of that ethnographic or qualitative work in order to figure out kind of what we'll do. So a small story off the measurement side, before we had a lot of computing could do the item response theory, there was one statistician who he spent years talking and doing a lot of qualitative work and came up with the weights for this fairly long instrument. And once we had the computing power and they actually took all of the data together and they ran it through item response theory, his weighting for the scores was almost identical to the very careful qualitative work that he had done to determine how to handle the scoring. That doesn't always happen, but I think it is important how these different ways and these different mixed methods can lend to each other. So I mentioned that we're always borrowing, that natural history designs have helped us drive designs of trials for years. It's how we understand what the disease process should be, but also has brought up bringing in more of that PK/PD information. And this is, again, also times you can talk to your patient partners, the nurses who are working with them, all these other folks. How many times a day will you really stick yourself with that needle? What type of mode of administration will work? There are a lot of different things to think about, but many times a lot of companies have been thinking, because that helps you plan the logistics for your drug and for other methods. Look at your old data for your accrual and retention rates. Kind of use that to help you plan your next study. There are a lot of things, a lot of exit interview information too that can help us work. But does all this take time? It's like, I don't have time to go talk to people. Well, it takes a lot less time than a failed study that you could have prevented. So, it's kind of like buying a little bit of extra insurance, doing a lot of other things. It helps you out later on. But we had a lot of talk about business proposition behind choices, and I think it's not really clear yet. It has been for some groups, but I think that is an area, and that's not something I get to work on, but it's something that as a collective we need to work on. So we're doing a good job every time, because if you're borrowing data from that early study, it better be pretty good. Or that might be why you like the non-dynamic, dynamic modeling. But there are a lot of sponsors, a lot of patient groups. It's not always easier and expensive, but again, it might be easier and less expensive, especially when you're in it together and you can bounce ideas off of each other. Now, one thing someone asked during the open session about how we could handle this, and were the international regulators talking. We actually do have a collaboration with EMA for rare diseases, and they're trying to expand that to other regulators. So we do talk. And we want to talk, and we want to try to give consistent advice. So the paradigm shifts we had, killing responder outcomes, I heard that one, rephrasing the concepts behind whether it's p-values, type 1 error, multiplicity, and that general gestalt of ideas, how we're going to handle bias and what we're expecting, and then also the paradigm shifts for borrowing priors or Bayes. So FDA is open for discussion. EMA and other regulators are open for discussion, and we all want to talk with you and with each other. So thank you, and thank you, Greg, for everything.
Great. Thank you to Laura Lee. I just have a few closing remarks. We went through a lot today. I want to thank all of you for sticking with us, particularly our panelists and presenters who did a phenomenal job of bringing up all of the issues that need to discuss, and thanks to all of you for active participation throughout the day, and to our web audience who continually emailed us questions. We didn't get to all of them. I'd like to thank particularly Laura Lee Johnson and Robin Dent for taking the lead at FDA to help make this meeting a success, and acknowledge and thank the Duke team who helped, we heard about earlier, I'll do it again, Morgan Romine, Catherine Frank, Elizabeth Murphy, Sarah Saprisi, and our newest member, Nicholas Harrison. Thank you very much to all of you, and have a wonderful rest of your evening. Thank you.